Good morning from Tampa and the 2024 Spastic Paraplegia Foundation Annual Conference. We're having a great time getting to know folks that are here for the first time and talking with doctors and researchers. A lot of great things going on. I want to share a little bit more about that with you in a couple of minutes. First, let me say we apologize for a little technical problem we had at the end of yesterday. The last thing I told you is that we would have Dr. Fakari and the child onset discussion on for you next, and we had some unexpected issues that occurred, not with our technical folks, but with some other issues here at the hotel. So to kind of get you up on that information, Dr. Fakari is going to join me mid-morning to talk a little bit with you about that. So we're going to try to catch up some things from yesterday. But this morning, I have a very beautiful young lady who's working hard helping us here in this conference, Miss Blair Qiang. Blair's the daughter of Jim and Melissa. And of course, you guys know Jim, a longtime member of the foundation and our fundraising chair. And I've asked Blair this morning to share with you just a little bit. You know, we get the impression so many times we think these diseases are just affecting those of us who are a little bit older. But Blair knows that's not the case, and she's fellowshipping with and working with our children and our youth. So, Blair, thank you for being here this weekend. Thank you for all your help. Share with me and with our folks listening virtually how your work has been with the children, with the youth, how you see this youth group coming together.
Okay, so I think it is so important because I know I've been coming to these meetings since I was little. Like, some of them I don't even remember coming to. I just know I was here. But I feel like there wasn't a lot of, like, connection with the younger kids, and I didn't meet that many people that also had this disability. It was always like, oh, my parents and my parents' friends here and stuff like that. And I know there was one conference we went to, Atlanta, I think it was, and we went to, like, the aquarium. But then it's just been, I feel like it's been a lot harder for us to, like, find each other and connect with each other. And I feel like that's so important. Like, we all sat, a bunch of the, like, young adults all sat together for dinner last night. And it was so fun. We were all talking. We exchanged numbers. And I feel like that's just so important because, like, yes, like, we're all connected from having this. But, like, I feel like it's so important for the younger kids to have each other and have friends in here that they can talk to because we're going through the same things, but younger than what some other people in this foundation are doing.
You raised a great point. In past years, we would take groups off-site to aquariums and festivities like that. And the reason we don't do that these days, it really two things. It's cost, but the other bigger issue is potential liability.
Yeah.
Finding folks who will move us because of our mobility issues that we all deal with. So that's made it difficult. So we try here to provide even a separate track that's focused on children activities and youth activities. And you've been so helpful in helping us manage that here at this conference. But you raise a great point that I hope the young people who are here I watched you guys have dinner together last night. I hope when you guys go home, you stay in touch from time to time. You can share just about life in general. But there are days we all have a more difficult day. The temperature's cold, there's pressure in our lives, something has caused our symptoms to be worse. And when you can talk to someone who understands that, it makes a difference.
Yes, for sure. And I know I talked, because there were a few people that it was their first time coming to these conferences last night, and they were like young adults, and they were like, how has it been like coming here like for a while, like since you were young, like knowing about this whole group and this environment and all these people that also have this disability? And I was like, I honestly think it has helped so much, like having these, like this group like surrounding me growing up, because I feel like I would have had like much more of a negative view if it wasn't for this group of people and like seeing that there were other kids affected too.
Well, Blair, we've known you for a number of years. You
Yes.
just have grown up on us. And again, thank you for being here, for being supportive, and for helping with the details of this weekend. And guys, in just a moment, we're going to turn over to Dr. Baas. He's going to have our first presentation here this morning. We'll have some breakout sessions later, but you'll have virtual feed from this room about all day. In between, we'll be talking, I hope, in just a few moments after the first session or maybe the second session both with Dr. Osdendler and with Dr. Fakari to catch up on some issues from yesterday. So thank you for joining us. We hope that you are learning some things about our conditions, about our foundation, that will be positive in our lives and our work together in the future. So with that, thank you for joining us. In just a moment, we're going to turn the camera toward Tim and get this day started. So thank you for being with us, and we'll talk to you later.
Good morning, everybody. Welcome to our Saturday program. We're glad that you're here. It's a great day. It's sunny and warm and humid outside here in Tampa. That's why we're inside, so where it's a little bit more comfortable. Welcome to Florida. You never know what you're going to get. We're really glad you guys are all here. We had a great day yesterday, a lot of great information. I want to point out a couple of quick things for you. Remember, tonight, we're having the dinner here this evening where we're going to have the raffle, ticket drawings, and all that kind of stuff. Make sure you get your tickets. Go see Josh at the main table. Take care of that. Also, we still got some T-shirts to purchase and stuff. Make sure you get those. My wife and I buy a ton of these every year. We hand them out at Christmas, birthdays, you name it. We got so many in our closet. We hand them out for things that are great to give to people. They're a great way to express what we do and how to spread the word. Okay? You never know where this shirt's going to end up somewhere. Okay? Another thing, caregivers yesterday had a great breakout session, and there was a moment where we had some caregiver tools, items to help our membership with. If you'd like to see some of those items that they showed in the caregivers meeting yesterday, we're going to have them over at the table for people to take a look at. Items you can purchase and buy, little things that just make your day a little bit better. Okay? Real quick, I want to make a quick shout out to a couple of our sponsors for U.S. Youth Soccer. Thanks for being here. It's a Florida group. They're really, really great. Canine Companions, who were here yesterday with our youth group, working with them yesterday. Glow Natural Wellness. I like those. I think I'm going to start using their products soon after this Florida heat, beats down my skin a little bit. Acava, they helped us with our welcome bags. The First Community Bank of the Heartland that also had some items that we gave you in your welcome bags. So I think I covered everybody on that. We're going to get this day started. It's going to be a nice, easy day. Oh, and real quick, if anybody in here is wanting to go to the PLS breakout, it's just down the hall, around the corner, just down the hall. I'm sure they're already there, but I had to mention it anyway. Here we go, ladies and gentlemen, Dr. Baas.
Thank you so much. It's such a wonderful pleasure being here. I started studying hereditary spastic paraplegia about 16 years ago, and at that time it was one small project in my lab, and due in large part to how this community has really embraced me and my research over the years. These days, hereditary spastic paraplegia is the primary topic of my research. And this foundation has been wonderful in providing 3 different grants, additional grants to my colleagues at Drexel University, and just an enormous amount of moral support. The community has been wonderful, and I'd just like to welcome everybody to email me, give me a phone call, whatever you like, and I'm happy to share with you any progress that I have. I'm not a medical doctor. I can't really give you a lot of medical advice, but if you're interested in how the research is going, I'm completely transparent and eager to speak with you. Let me start out with the first slide and talk a little bit in general about hereditary spastic paraplegia. As you know, hereditary spastic paraplegia mainly involves gait defects and spasticity, and this is mainly in the lower limbs, lower limb weakness. Oftentimes, individuals will wind up in a wheelchair, not always. There are other symptoms, and these symptoms can be complex. They can extend into the upper limbs. There can be some speech impediments. There can be some urinary bladder issues. Usually, it's the gait deficiencies that are the most common feature of the disease. It's usually adult onset, but not always, and we have the pleasure of having some children among us these days for the meeting, and I am in contact quite often with a couple of different smaller foundations that are dedicated to childhood cases of HSP. There is an enormous amount of variance. I think the age of onset, at least with SPG4, average age, is maybe about 40, but children of all ages, people of all ages display different symptoms. Some individuals may not display symptoms until they're 70, and other individuals in the same family may display symptoms during childhood, so it's highly variable. Could I grab the next slide, please? So hereditary spastic paraplegia, what's going on with this condition in terms of the degeneration of the nervous system? It's mainly degeneration of what we call cortical spinal tracts. So these are nerves that start out in the motor cortex of the brain. So the cell bodies are located in the motor cortex of the brain, and they extend their axons over long distances down your spinal cord, all the way down your spinal cord. And then within the spinal cord, those axons form connections called synapses on other neurons. It's those upper motor neurons, the cortical spinal motor neurons that start in the brain. They're the ones that are degenerating in hereditary spastic paraplegia. And by degeneration, what we mean is a pulling back. They pull back from their synapses, they lose their connections, and then they gradually pull back toward their cell bodies. They also display pathological swellings. That's a hallmark, that the axons become swollen. Complex cases could involve other populations of neurons. People who suffer some cognitive problems, for example, that would be other sorts of neurons in the brain. The cortical spinal tracts are the primary tracts, the ones that everybody suffers degeneration, but there can also be some other tracts that are affected. Next slide, please. Hereditary spastic paraplegia is a term that covers a constellation of different genes that are mutated. When I first started studying HSP about 16 years ago, I believe it was about 12 genes that were identified whose mutations cause this disease. Now it's up to, I believe, 92 last I checked. It could be more. So these are all different genes whose mutations result in cortical spinal degeneration and this constellation of symptoms. Are these the same disease? Do they have some common coalescence on a pathological mechanism, or are they different diseases that just have the same constellation of symptoms? We don't know. We don't know the answer to that, but I think by studying the various genes that give rise to hereditary spastic paraplegia, we're coming closer to being able to answer that question. The particular variant that I study is called SPG4 HSP. This is the most common variant, so maybe about 40% of the cases it's SPG4, and then the other cases make up the other percentages. There are cases that we still don't know what gene is mutated. Individuals have all of the symptoms of HSP, but it's not any of these 92. It's still something we haven't discovered yet. So it seems to be some vulnerability of the cortical spinal motor neurons to a lot of different mutations of a lot of different genes that give rise to these symptoms. And even within SPG4, which is mutations of the SPAST gene, there are over 200 different mutations of the SPAST gene that result in hereditary spastic paraplegia. So with some diseases, everybody who has the disease has the same mutation of the same gene. Here, people who have SPG4 HSP, you have that in common, but the particular mutation that you have could be quite different, over 200 different mutations. Next slide, please. Now, what do I mean by a mutation? As you know, humans have 23 pairs of chromosomes. We have 46 chromosomes in total. There are the XY and the XX, the chromosomes that code for sex, males versus females. And then the rest of them are genes that are in common with both males and females. And these chromosomes have a variety of different genes on them, and each of these genes encodes for a protein. The genes consist of bases. A, C, T, G are the 4 bases. So a variety of different combinations of A, C, T, G, AAA, CCC, TGA, that's what's encoding for the proteins. And what a mutation is just one of these bases has aberrantly changed. A C has become a G, or an A has become a C, and that's where all the trouble comes in. The vast majority of mutations that occur in the human species are benign. We undergo mutations all the time, and they don't matter. But every so often in certain genes, they matter. And apparently in the SPAS gene, it matters a lot because all sorts of different mutations create trouble. Now with SPG4 HSP, a few additional facts about it. This is autosomal dominant inheritance. That means all you need to do is have one mutated gene and you've got SPG4 HSP. And it's presumably 100% penetrant. We don't know for sure if there are people walking around who live their entire life with an SPG4 HSP mutation and never suffer any symptoms. Theoretically, it's 100% penetrant. But people vary so substantially in the severity of their symptoms. Again, some small children are already in a wheelchair, where some 70-year-old individuals are just starting to experience the symptoms. So there's a great deal of variability, but theoretically, if you've got the mutation, you've got the disease. There has been confusion with other diseases. Because HSP is such a rare disease, and each individual variant rarer yet, a lot of physicians are not familiar with HSP. So there are a lot of cases, and I've heard specifically of, say, for example, a teenager in Michigan who all his life he was diagnosed with cerebral palsy. That's what they thought he had, cerebral palsy. But no, he's got SPG4 HSP. I've even heard some stories here yesterday at the meeting about people who were diagnosed with cerebral palsy until that individual's children had the same symptoms, indicating that it has to be genetic. Now, there are de novo cases, and what do I mean by de novo cases? One might think if everybody with HSP stopped having children, would the disease be over with? No, because these mutations pop up fresh. Once you have the mutation in your genome, you can pass it on to your children. But any new child that's born could have a mutation that just popped up de novo, brand new. It's not in the family. And that individual, if he or she has children into the future, would presumably have the possibility of having that mutant gene. Interestingly, the de novo childhood cases seem to be a lot more severe in their symptoms than the inherited cases. And I think that's probably teaching us something interesting and important to do with genetic background. If your family has this mutation and passing it on to new generations, it probably means that the genetic background in the family is pretty resilient to the mutation. Otherwise, they wouldn't be having children. But the small children that are born and their families have never had it before, their genetic background doesn't seem to be able to tolerate it very well. So a small child with a de novo mutation that might be the very same as a 70-year-old man who's doing pretty well, that small child may already be very debilitated. So this is another reason why this foundation is so important, because the knowledge that we gain from communicating with the patient community is so important. Next slide, please. Now let's take a look at the SPAS gene. I know this gets a little bit complicated, and I'm going to try to make it as simple as possible. But genes will code for proteins. And what's very interesting about the SPAST gene is that it codes for two different proteins. They're pretty similar, except for one of them has a part that the other one doesn't. So the shorter version, which you see in the lower schematic, that's called M87. M87 spastin. And M87 is the predominant form. So if we look in your body for spastin protein, it's very easy to find M87. There's a lot of it in your nervous system, in every cell of your body there's spastin. Its function is to sever microtubules. So microtubules are what are called cytoskeletal elements in cells. They're particularly prominent in neurons because they're kind of like the skeletal elements that give a cell its shape. So there's a lot of M87. It's regulating the microtubules by cutting them into manageable pieces, and that's important. M1 is this little bit longer isoform, and it's the minor one. You can hardly detect it. There's so little of it. And yet it must have some important function because evolution has held on to it. This isoform called M1 relates to membranous structures. So if you remember Dr. Blackstone's talk from yesterday, he showed how mutant M1 is less mobile than normal M1. And that's in the endoplasmic reticulum, which is a membranous structure. So M87 and M1 might interrelate to one another. They may not be entirely different, but for the most part, I think the easiest way to think about them is they're two different proteins, each one doing a rather different thing. Next slide, please. This just schematically shows what I've been talking about. So you can see on one side of the slide, you can see a neuron, and it's got its long axon, so that rounded portion, that's gonna be in your motor cortex in your brain, and then that elongated portion called the axon, that's what extends down your spinal cord. And then blown up, you can see that colorful filamentous structure, that's a microtubule. And microtubules are really important for the architecture, for allowing the neuron to generate this very elaborate morphology. And then those other globular structures, those are motor proteins, and they're carrying nutrients and other sorts of cargo. They're carrying the mitochondria that Honda talked about. They're carrying all these cargoes up and down the length of the axon. So those are microtubules, and those are regulated by M87. Now over in the lower panel, you see the endoplasmic reticulum, and that's where M1 inserts into the endoplasmic reticulum and helps it to have its shape. Next slide, please. Now when you think about M1 and you think about M87, they're both going to bear the mutation. If you have a mutation, a pathogenic disease-causing mutation in your SPAS gene, both M87 and M1 will have the mutation. And really the question becomes, in a lot of diseases, is it a loss of function disease or a gain of function disease? And what do we mean by that? What we mean is with a loss of function, if you don't have enough functional, normal M87, or you don't have enough normal functional M1, you're in trouble because your microtubules aren't severed enough, your endoplasmic reticulum isn't shaped well enough. You're in trouble because you don't have enough functional normal protein. Gain of function is very different. And don't be fooled to think gain of function is something cool and it's a good thing. It's not. Gain of function means that that mutant protein is doing something it should not be doing. So it's gaining some kind of attribute. Maybe gain of toxicity might even be a better term, because the mutant protein is misfolded, different sites may be revealed that are normally hidden, and now it's doing crazy bad things. And in actual fact, in most neurodegenerative diseases, gain of function is more common. Next slide, please. So I've been funded by the SPF for quite a few years now, a couple different grants, to kind of resolve the normal functions of spastin, and then whether this is a gain of function or loss of function disease. And this slide kind of illustrates in another way what I mean by gain of function and loss of function. So loss of function is just that the kid didn't show up for class. Gain of function, oh, he's there all right, but he's making trouble. And gain of function gets progressively worse and worse and worse because your spinal cord, your corticospinal motor neurons, are accumulating that mutant protein. Usually, with a gain of function mechanism, the mutant protein becomes quite stable and it accumulates. Next slide, please. Now, over the years, I've wanted to answer a number of questions. And one of the questions is, if this is a gain of function disease, is it gain of function due to the mutant M87, or is it gain of function due to the mutant M1? And what we've been able to do with a few different model systems is to introduce into squid giant axon or introduce into Drosophila fruit flies or rodent neuron cultures either the mutant M1 or the mutant M87 just by itself and compare. And when we do that, it's always the mutant M1 that's highly toxic. Mutant M87 doesn't seem to be so bad. So say, for example, with the fly, if we introduce the mutant M1, that fly can no longer fly. You put it on its back, it can't right itself. It can't climb, it can't fly, it can't hold its wings properly. You introduce mutant M87, it doesn't really care too much. It's pretty normal. With the squid giant axon, we can actually look at cargoes moving on the microtubules, and the mutant M1 will be very deleterious, will really slow down the movement of these organelles. Mutant M87, no big deal. And the same is true with the rodent neuron cultures. So it seems to be that the mutant M1 is the problem, that it takes on the toxic properties. And remember that mutant M1 has something that the mutant M87 doesn't, and that's that extra little region. And that extra little region is very hydrophobic. And by hydrophobic, I mean that it shuns water, and by being hydrophobic, it can really promote misfolding and toxicity. So it makes sense. It makes sense. What is really important as a next step is we needed a vertebrate model because squid don't walk and cultured neurons don't walk. You know, we needed to be able to understand a model that has a spinal cord, a brain, and can walk. So what we did is we made a mouse model in which we introduced mutant human spastin. Next slide. Now this is a normal mouse, and it's walking on this rod, and it's perfectly normal. And let's see that one more time if you can. Now, there's something called a knockout mouse, and what the knockout mouse does is you get rid of both your spastin genes, and that mouse has no spastin. And curiously, that mouse would walk just about the same as the normal mouse, with no spastin at all. It's okay. It's sort of okay anyway. Now the next slide is when we knocked in the human mutant spastin, and this mouse has a lot of trouble walking. Now, it may appear that this mouse is kind of chubby, but it's not really chubby. It's really just scrunched up because it's trying to walk and it's having a hard time with it. So this mouse has all the normal mouse spastin. So there's no loss of function going on here. It's exclusively gain of function. So I think what this mouse does is really fortify the point that SPG4 HSP is primarily a gain of function mechanism underlying the disease. Interestingly, though, that if we take this mouse that you're looking at right now and we cross it with the knockout mouse, the offspring will be worse, So the way we think about it is that either loss of function or gain of function could potentially explain the disease. But at least with these mouse models, it appears that gain of function is necessary and sufficient for the disease, but loss of function exacerbates the disease. That's how we think about it. Next slide, please. So this mouse has certain limitations, though. We made this mouse about 15 years ago, and it's not been made to the most contemporary standards. Remember, it has 2 wild-type mouse SPAS genes. It's got the mutant SPAS gene. The mutation we used for this mouse is called the CY mutation, and it's only gain of function. So it's not quite a representation of the human patient. And this became a particular problem when we wanted to try antisense oligonucleotide therapy. This is what I was funded for last time by the SPF. Antisense is a technology that's really effective at handling treating certain diseases, because what the antisense can do is it can shut off the expression of the mutant gene. If you have what's called allele-specific ASOs, you introduce it into the brain, it'll leave the wild-type gene alone, and it'll simply shut off expression from the mutant gene. Interestingly, though, we couldn't do that with the current model. It's a little complicated, but the way that we made the current model is we introduced cDNA, and cDNA has what are called exons, but no introns. I know that's a little bit complicated, but there are certain parts of the gene that we did not introduce with the old technology. So now what we're doing is we're creating a brand new model, and this model is going to be remarkably similar to humans. We're going to have 1 wild-type human SPAS gene. We're going to have 1 mutant human SPAS gene. The mouse genes are gone. We're just simply adding the human genes. They have all the introns, all the exons, so it's just going to be very, very much like the human condition. And it'll have a different mutation, which is called the Lillian-Blair mutation. This is a mutation suffered by 2 little girls whose parents have formed a foundation called the Lillian-Blair Foundation, and they very generously provided financial support for the creation of this new model. But this model will have gain of function and loss of function all together in the same animal, and because we have the introns as well as the exons, we should be all set to try the antisense oligonucleotides. So we're very excited about doing that. New tools are very important in moving forward in studying this disease. Another really important new tool that we're working on, I really want to share with you today, is the creation of a brand new antibody, an M1 specific antibody. Remember when I said that M1 is not expressed very highly? It's very easy to detect M87 with a variety of techniques. You start looking for M1, and there's so little of it that it's very hard to find. And so M1 being what we think is the culprit, the cause of this disease, if it's invisible to us, if we can't see it to study it biochemically or using microscopes, if we can't see it, we're kind of in trouble because that's the target we're interested in. So we're working on developing an antibody that will enable us to see it. Next slide, please. So what is an antibody? Now, you all know that your body creates antibodies in response to foreign antigens. So in the research lab, we use antibodies all the time for a different reason. We kind of exploit what an antibody is to be able to study different proteins. So you take the antigen, in this case it's the spast protein, the M1 protein, and you inject it into a mouse or a rabbit, or sometimes they use chicken, and then that animal will make antibodies, and then you purify the antibodies, and now you've got a tool. You can recognize that protein on biochemical assays or microscope assays. You can visualize it in cells. And the problem with M1, again, is so little of it that with the general spastin antibodies, you could barely detect it. So we really wanted to develop an M1 antibody. It's kind of risky because we don't know it's going to work or not, but to create an antibody to enable us to study M1. The other thing that's really sometimes incredibly useful about antibodies is if the antibody binds the toxic site of the toxic protein, it could theoretically block the toxicity. So if M1 has a domain that's particularly toxic, then this antibody, if we introduced it, say, into your brain, it could block that site. So even if we can't get rid of the accumulated M1 protein, we can inhibit its toxicity, theoretically, with the antibody. Next slide, please. So we use a couple techniques. I know this gets a little technical, but Western blotting is a biochemical technique. I'll show you an example in a moment where you kind of see bands on a gel. You can visualize the different proteins. Then immunohistochemistry is when you use the microscope to visualize where the protein is. Next slide, please. So here's a Western blot. And this is a Western blot that I created in the year 2008. And this is using a general spastin antibody. And you can see all those big, heavy bands. That's all M87. So you have the cerebral cortex, you have the spinal cord, you have embryonic day 18. This is mouse tissue. You have P12, you have the adult mouse, you have E18. And those big, thick bands you see is all M87. Now notice in the adult spinal cord, you've got less total spastin, but you start to see M1. You can kind of see that it's there. And that is our first hint that maybe cortical spinal motor neurons are unique in normally generating more M1 than any other cell of your body. And that's a good clue as to why cortical spinal motor neurons may be particularly vulnerable to degeneration when there's a mutation in M1. But again, it's so hard to detect that we set forth to make a brand new antibody that would only detect M1. Next slide, please. There's a brand new technology making recombinant antibodies. Remember I said, you inject a rabbit or you inject a chick and you purify its antibody from its serum and all that. Well, these days, people are making what are called recombinant antibodies. Remember, an antibody is a protein too. So an antibody has a sequence. It has an A, C, T, G sequence. And if we develop the antibodies and we know that sequence, well, that puts us in a very good position. Because if we know the sequence of the antibody, then we can make unlimited amounts. You can just churn out as much as you'd ever need of these antibodies. You could also theoretically, if you wanted to use it therapeutically, instead of introducing the antibody into your brain, you could introduce a DNA construct that would compel your brain to make the antibody, kind of a gene therapy approach with the antibody. So it can be incredibly useful to have these. We have 5 different ones currently in hand, and they may all have different properties. We're just now testing them back at the lab. We're testing them even as we speak. And we hope that at least one of them is really, really good, but maybe all 5 will be good. We also have a rather adventurous idea that maybe M1 could be a biomarker. So I think you've all heard about the utility of biomarkers in disease. We'd like to be able to know, have an unbiased marker of how disease progression is underway in particular patients, and also to know if we apply a therapy, is it working or not? So to have something in your bloodstream or something in your cerebrospinal fluid that we can assess quantitatively, that's called a biomarker. And if it's really true that M1 is very much accumulated, mutant M1 is very much accumulated in your corticospinal motor neurons, maybe it's getting into your cerebrospinal fluid, or maybe it's getting into your blood. And if that's the case, then M1 could also serve the purpose of being a biomarker. We don't know that that's true. It's kind of a adventurous idea, but if it's true, that is one more just groundbreaking advance of having this antibody. Next slide, please. Now this gets kind of complicated, but what I want you to appreciate here is on the first side, the panel A, we have overexpressed, we've taken a DNA construct for either M1 or M87, and we've overexpressed it in cells. And if you look where it says M1 specific antibody, you can see that under the M1 column, you've got that green band. With the M87 column, you see nothing. Now shift your eye over to where it says spastin antibody. This is a generic spastin antibody. And you can see both the M1 and the M87. So a general spastin antibody sees both of them, but our new antibody only sees one. Now this is a preliminary version of the antibody. This is called a polyclonal antibody. So we're not quite seeing the final product yet, but the preliminary product is really good. So we're very, very hopeful about this. Now look at the mouse spinal cord. If you look at the wild type, that's just a normal mouse. It's not even diseased. The M1 specific antibody, you see that big fat green band? We can see M1 really well now. Remember I said it was so hard to see with the general spastin antibody. And if you kind of shift your eyes over to where it says spastin antibody, the general one, you see that red band, that's your M87, but where you see that little asterisk, you don't see M1. It's just so hard to see M1 with a general spastin antibody. But with our antibody, you see a very robust band. And then if you look down where it says merged, you see the green band is M1 and the red band is M87. So we can really see them equally well. Even though there's so much more M87 than M1, now we can see them both. So I think this is a really big advance. Next slide, please. A little bit later this morning, Emanuella will present to you her gene therapy results. So we're working on gene therapy in collaboration with Miguel Estevez of UMass. And one of the things that's really important for gene therapy is to know, is it working or not? Is it working? You want to be able to see that the M87 and the M1 bands on a Western, when you apply the vector and you remove the endogenous and you replace it with the healthy spastin, that both M1 and M87 are being fully replaced. So Emanuella's going to talk about this in a little while, but I just wanted to give you a peek ahead of time because it really illustrates how much we need this antibody for almost all of our projects to make substantial breakthroughs, and this is a really good example of that. Next slide, please. This is an example of an immunostain, and this is a cultured cell. This is just like a fibroblast. And if you look at the upper panels, what we've done there is to overexpress M1 and then stain it with our antibody. And where you see the M1, the healthy M1, is in those little blotches. It's probably the ER we're seeing, the endoplasmic reticulum, as Dr. Blackstone talked about. But look at the mutant. Look in the lower panels. See how the mutant looks so filamentous? I think the mutant is either accumulating in the ER or maybe accumulating on the microtubules. It's a little hard to tell which one it is because they look so similar. And I think Dr. Blackstone pointed that out too. But the mutant M1 is behaving in ways that we can now visualize. And say, for example, if we tried a therapy, say if we tried maybe the drug that Honda talked about, that we could see, well, is it making the mutant M1 behave the way it should behave? Is it working? So being able to see M1 this way, particularly the mutant M1, is really quite a breakthrough. Next slide, please. This is an immunostain of mouse brain. And this is very interesting, that if you look at the frontal cortex, so this is where the corticospinal motor neurons are located, The red that you see is called CTIP2, and that just confirms that you're looking at a corticospinal motor neuron. The blue is the nucleus. The green is M1. And notice that the corticospinal motor neurons stain really well positive for M1, as we might expect. But look over a hippocampus. First of all, you don't see any red because they're not corticospinal motor neurons, they're hippocampal neurons. And notice you really don't see that green stain at all. Now, I'm sure that hippocampal neurons have some M1, but apparently not very much because we can't see it. And what is this telling us? It's really confirming that finding from way back in 2008 that corticospinal motor neurons normally express a lot of M1. And this isn't just in the adult. These are cultured neurons that we've also looked at. And cultured neurons are the same thing. So even in a small child, throughout life, there's M1 enriched in corticospinal motor neurons. And it may be that that's really the elusive answer to why corticospinal motor neurons are really so vulnerable to mutations in spastin. But maybe that's also why corticospinal motor neurons are so vulnerable to mutations in those other 91 genes, because M1 spastin is there. And if anything goes wrong, M1 spastin is going to be that troublemaker that came to class and made the trouble. So maybe that's the common answer across HSPs. And we're eager to take this antibody once we're finished with it, and we can try it in animal models or human tissue, not just for SPG4, but for other HSPs too, maybe PLS too. And maybe that's a common factor that's really at the heart of all this trouble. Next slide, please. So let me just draw a few conclusions here. The corticospinal motor neurons express M1 more highly than any other cell type that we've been able to look at, even early in life. Now, these are tentative conclusions. We're still working on it, so don't take this as gospel, but this is what we think so far. Corticospinal motor neurons apparently need M1. There must be some reason why they're expressing M1. And so whether it's regulation of endoplasmic reticulum or endosomes, or for some reason, we'd like to understand why they need more of it. Because this may be another reason why corticospinal motor neurons are particularly sensitive to loss of function, of losing something that they need. We think the mutant M1 becomes more stable and accumulates in the corticospinal tracts, eliciting toxicity. But frankly, we don't know that because we haven't been able to look at human tissue. For that matter, we haven't been able to look at mouse tissue either without the antibody. So now we will. We'll be able to look at the mouse tissue, and it's another reason why it's just so important that we develop a biorepository of human tissue, because there are a lot of ideas that we have about these things that we just need confirmation in actual human tissue. So with the M1 antibody, we can now see the key protein in SPG4 that was previously almost invisible to us, and I think that's important. Next slide, please. Just a couple of other kind of speculations. Might the M1 antibody be introduced into patient corticospinal motor neurons to mitigate toxicity? The gene therapy, as Emanuella will talk about shortly, is fantastic because it turns off the expression of the mutant protein and replaces it with healthy protein. But if you turn off the expression of the mutant protein, what about all that mutant protein that's in your spinal cord that may have been in your spinal cord for 10 years, 20 years, 30 years, 60 years? Is that going to go away just because you turn off more? Maybe, but likely it's quite stable and it's not going to go away so fast. So we need combinatorial therapies. We need another tool to mitigate that toxicity. And the M1 antibody may be just the perfect tool for mitigating the toxicity of the mutant protein that's just not going to go away very fast. Might the accumulated M1 in the corticospinal tracts leak into the cerebrospinal fluid or blood, providing that biomarker? I think it's a long shot. I'm not saying I'm confident that's the case, but if it is the case, it could really be a fantastic missing piece of the puzzle of having this biomarker. And then again, kind of a big speculation, but might M1 be the common vulnerability that ties together everybody in this room? The common factor that ties together all variants of HSP. Maybe it represents the vulnerability of corticospinal motor neurons, regardless of whether we're talking about SPG4 or any of the others. Next slide, please. So I'd like to acknowledge the people who've done this work, Gerardo Morfini, who's worked with me on the SQUID model, and the members of our team at Drexel, Oscar and Emanuella, Korai, who's the guy back at home doing all the work on the antibody, and then other collaborators we've had. I have a short list of other collaborators and students I've had over the years. And I just want to point out here that we have a plan. And our plan involves gene therapy, it involves ASOs, it involves M1 antibody therapy. We're also interested in new drugs. We heard a little bit about new drugs yesterday, and we're trying to understand, why is mutant M1 toxic? What pathways is it tapping into that's creating the toxicity? And if we understand those pathways, then we can repurpose drugs that already exist. Maybe Oscar will talk a little bit about that later this morning, or create new drugs that specifically mimic the properties of the M1 antibody to block or inhibit that toxic site on M1. And the last thing I'll mention really quickly, and I know Emanuella wants to talk about this a little bit too, is to restore lost function. Because a lot of the things, the therapies that we talk about, would be quite ideal for preventing the degeneration of the corticospinal tracts. But what about people who are already in a wheelchair? How are these therapies going to help you if you've already got a degenerated spinal cord? And I just want to stress, at least from my perspective, that I have a nobody gets left behind kind of attitude about this. That it's wonderful to be able to diagnose a patient early and prevent the symptoms from happening. But if you're already symptomatic, I hope we can stop the progression of the symptoms, but I hope we can do even better. I hope we can find ways to fortify the healthy axons that you already have so that they can substitute for your lost axons. So we can talk more about that, and I know Emanuella wants to, but it's important to me to stress that everybody in the room is important, and regardless of your stage of degeneration, we have you in mind and we're working on it. Next slide, please. I also just want to thank again the Spastic Paraplegia Foundation for the financial support they provided this work, but also the inspiration. And I feel part of this community, and I feel embraced by the community, and I embrace this community. And so my heartfelt thanks to the foundation for what they've done for my research and really for all of you. Also, I want to acknowledge, as I mentioned, the Lillian Blair Foundation for funding the new mouse model, and I want to acknowledge the Cure SPG4 Foundation for funding the M1 antibody. And both of these foundations are made up of parents of small children who have the de novo mutations. Other donors who have generously sent me a couple $100 or whatever, is very important, and that's very appreciated too. And of course, NIH is where the big bucks come from, and they're also the hardest to get the money from, but we'll keep trying. And I really want to thank all of you for your very kind attention. What questions do you have for me? Back of the room, question.
Of course. Here you go.
Hi, and thanks for your time. You talked at the beginning, there's, for someone who has SPG4, there's over 200 mutations of this one diagnosis, which is wild. This is the first time I've heard of that. Has there been any sort of like longitudinal studies on, there's such variety of how symptoms are expressed within just SPG4? Has there been any like longitudinal studies on is there a correlation between like which gene mutation you have and severity of symptoms?
That's a key question in the community that we talk about a lot. For example, when we make a mouse model, we choose the mutation that's going to give the most severe phenotype, because then it's going to be easier to know if the therapy is working. So there are some studies, not very many, but some studies that suggest that if it's a truncating mutation, the symptoms might be a little different or a little earlier onset or later onset compared to if it's a non-truncating, a full-length mutation. By truncating, I mean due to the mutation, it's just a shorter region of the gene that will express. There's a premature stop codon. But there's really not enough data to know for sure. And really, as I said earlier, even people who have the same mutation can vary significantly in the age of onset and severity of symptoms. So these say, small children, the Lillian-Blair mutation, there may be an adult with the same mutation who doesn't have symptoms at all until age 60 or 70. So there may be, if we had an enormous amount of data, it's possible that different mutations may be more or less severe, or maybe different mutations are more prone to the complicated symptoms. But because there's such a variability from person to person to person, it's really hard to know the answer to that question. And I think what's probably more relevant is genetic background, is what else is going on in your genome that makes you more sensitive, more vulnerable, or more resistant to having that mutation. If there's 92 different genes whose mutations give rise to this disease, there's probably 92 others that don't give rise to this disease, but have implications for the vulnerability of the individual to those mutations. So maybe with big data analysis, we'll start to understand some of this.
Any other questions? Way over here to your right. Hang on, I'm going to pick this one up first over here. Hang on, we'll get there. Here we go.
My sister and I have HSP. I have number 7. She doesn't know hers, but we've searched in our mother's background and haven't found anybody, and our father's. Did I understand that we could be, say, ground zero? Or, how did this begin?
So your sister and you are not twins, right?
No, we're not twins.
You're not twins. And to the best of your knowledge, there's no background in your family of having HSP-related mutations, but you both do.
That's correct.
It's very interesting because I don't know the answer to your question. I mean, it's very unusual, but it may speak to exactly what I'm talking about, that maybe it's a combination of a couple of different genes. Maybe there's a mutation that you have, your mother has, that within itself is just not causing the disease. But combine it with something your father has, and you've got trouble. And maybe this is, there's a certain fraction of people who have undiagnosed, they don't know what the gene is. So that's the only possibility that I can think of besides de novo. I suppose the other possibility is it's just bad luck. I mean, you've each got a de novo mutation. Maybe you've got 7 and your sister's got 4 or something. And it's just, there's billions of people in the world, and maybe it's just unlucky that you each have a different de novo mutation. But I find it curious. It's a curious situation that maybe learning more about the variability, the variances in your genome might provide some clues. Because I do think there's a lot of stuff that we just don't know about the contributions, the sort of the totality of the genome that might cause HSP.
Now, our father died at 42, so maybe he didn't have the onset yet.
Yeah, so if your father died when he was age 42, if he had lived to be 50, he might have had symptoms. If he had lived to be 60, he might have symptoms. So that's a good point. You just don't know because many people don't suffer symptoms until much later in life. So unless he's had an actual genetic test to confirm that he didn't have the mutation, then he probably did. He probably did. That's my thought. He may have had de novo, and so no one in his family previously had it. But it could be that in some families you're just very resilient, that your father and maybe his father were just very resilient to it. You know, there's some people who have minor symptoms and they'll tell their doctor, I don't have any symptoms. I'm fine. I have no symptoms. And then the person walks across the room and, yeah, he's got symptoms. He's just kind of stubborn about it.
We've got one more question over here. Dr. Baas?
Yes, sir. Thank you. I was wondering, so you talk about gain of function. So would an example of that be something like if you get sudden shooting spasms where your knee flies up or something like that? Is that what you're referring to as an example of gain of function? And how common is something like that?
Well, we use the term gain of function more at a molecular level. So what I'm talking about with gain of function is you've got that mutant protein in your brain, in your spinal cord, and in some way that mutant protein is creating a problem situation. Maybe it's causing certain enzymes to be overactive or underactive. Maybe it's causing the mitochondria that we heard about yesterday. They're not moving. So that's why your energy levels are low. Your mitochondria aren't moving. So we talk about gain of function more at the molecular level of maybe it's stopping proteins from being modified the way they should. So things aren't working. Now that may lead to, and we think it does, that we think that may lead to the symptoms that you're suffering. But we use the term more at the molecular level.
I've got one more in your front, right here.
You got another one in the back, but let's see if we have time.
All right, thank you for that presentation. You answered all my questions that I had throughout.
Well, I guess we can go now, right?
Except for one last one. How far are you away from doing the M1 protein antibodies in the mice or whatever? How far are you from doing successful studies into that?
Well, we have just received a package that has the 5 antibodies in it. And Korai is back at the lab, and he's testing all 5 of those monoclonal antibodies. We were absolutely thrilled with how the preliminary polyclonals worked, as I showed you. And if those monoclonals work as well as the preliminary version, and all we need is 1, we've got 5, maybe all 5 will be great, or maybe 3 of the 5 will be great, or maybe they'll be different in different ways. Maybe some will be great for Western blotting, and others will be great for toxicity blocking. So we're testing these antibodies, and we should be able to have some answers soon as to how good they are. Once we have identified the ones that are good, we can make unlimited quantities. Not without money, we have to buy them. We can make unlimited quantities, and we're certainly eager to share them with the community. So we want to have human tissue, as we talked about. We want to take a look. I know that John's lab at Wisconsin has a rat model. We want to share the antibody with John's lab and with other collaborators. And I hope hope it's a transformational moment for the HSP field that we have the antibody, and I hope it has properties that will be useful. And we can also use the antibody for something called immunoprecipitation, in which we put the antibody together with a cellular extract. We pull down the M1 protein. Then anything that sticks to M1 will come down too. So we can get, I think someone yesterday used the word interactome, and we can find out, say, what proteins stick to mutant M1 versus normal M1. So it could be just a font of information, and we hope. It could also be that all 5 are garbage. We have to start over again, so, but we're hopeful.
Okay, everybody, we said we had one more question. We got one more, and then we're going to have to move on, okay? I'm going way left. It's okay. It's okay.
Thank you. I had a question. So in my family, it's hit every generation, and everyone in my generation has had this. And when you go get genetic testing done, they tell you it's like a coin toss. You have a 50% chance of having it. But in my family, it's been every generation, and everyone in that generation, has had SPG4. Is there an answer to that, as if the mutant gene is just strong and it's more effective? I'm just curious to know, because the people I've met have not had it as significant as I've had in my family.
Yeah, it's autosomal, so it's not on a sex gene, and it would seem 50-50. If one side of your family is passing it on, it seems like it would be 50-50. So it's probably just bad luck that everyone's had it. But it's possible that there's more than one gene involved. I suppose it's possible. One time I heard about a family, I think in India, that was from such a small community with interbreeding that there's a family that's homozygote for the mutation, meaning both genes are mutated. But that is highly unusual, very small kind of interbred population of people. But why that would be with such a rare disease in our country, it more likely is just bad luck, I would say.
Well, everybody, thank you for those wonderful questions. Dr. Baas, thank you very much for your wonderful, informative presentation.
Thank you.
We're going to take a quick break for a little bit, get ready for our next presentations, everybody. We'll see you in a little bit.
Well, good morning again. I have an exciting visitor with us that we all know and love, Dr. Hande Osdendler. And Doctor, thank you for all that you do for this community, the research that you're involved in. As you guys remember, last year, Dr. O, I'll call you Dr. O.
Sure.
Dr. O brought us a wonderful piece of artistry, a work that she did that we didn't know she had time or energy to do after she does all the research work that she does. But she brought that, it was auctioned off, and that afternoon we had anonymous donors say he would donate up to $75,000 if we could match it while at the meeting. We got to the Saturday evening process, we had auctioned everything off, and Dr. O turned to Jim Qiang and said, "Jim, how much do we like?" And Jim said, "We need $12,000 more." And Dr. O said, "Let's do that right now, and I'll do the first 1,000. And in the next few minutes, we raised the balance of that. So Dr. O's not only in this to help us get research done that makes a difference, but helping us figure out how to raise dollars in that direction. So Dr. O, thank you for all that you do,
Thank you
all
so
that
much.
you've done, and for being here with us this weekend. Dr. I guess you're involved in so many pieces of this research, and we have followed your work with the compound NU9. So let me just ask you a very wide open question. If you were an SPF or PLS individual or family at home listening now today or listening next week on YouTube, in the research field and as we live with what we live for, what's the most encouraging news and thought you can give us today based on what you're doing?
Of course. Thank you so much for the invitation. And I think for the people who stay at home and who cannot attend here, we are still part of the family. And this is like a family get-together. And I really feel that this foundation created a family for HSP and PLS patients, and not only the love of the family, but also hope for the future. And it's not just a filled word like hope, but it's really, truly meaningful hope. And I think that I have seen you generate that over the years. So I'm humbled to be part of this family, so thank you so much for including me. And I see research exponentially growing. First, for example, we did not really know about the genes, the mechanisms. Now we know more than 80 mutations, 150 total mutations. The genetics of the disease is better understood. And with that, the mechanism, the underlying mechanism of the disease is much better, precisely understood. So we begin to understand why each patient developed the disease. What is the underlying cause? Once we know that knowledge, then we can actually formulate effective long-term treatment strategies. And those are emerging with gene therapy approaches, ASOs, and with the gene therapy, you have seen that there's a clinical trial already initiated with children. And that's an amazing achievement. And the ASO is an important topic, and you know the ALS field, SMA fields, you know, they are making important strides in that. And I think HSP and PLS is prime targets for those because we know the mutation. And I think that is an open field that's coming up very fast, very quick. And I think that's why it's important to be involved with the foundation as early as possible and as much as possible, because when there is change, there is momentum, you need as many people in the game with you. So I think that's very important. In addition, for the first time now, we identified a small molecule, a compound, NU9, as you said, that improves the health of diseased upper motor neurons. I mean, that is game-changing. We did not have any compound that improves the health of upper motor neurons. Now we have it. And most importantly, now there's a company, Akava Therapeutics, which you can check on its website, says our primary target diseases are hereditary spastic paraplegia and primary lateral sclerosis. We did not have a company out there fighting for those patients before. Now there's a company, there's a small drug, and that NU9 actually got approval from FDA to start phase 1. I mean, can you imagine how exciting that is, that we are going into clinical trials, and that within a short period of time this drug will be tested on patients, on healthy volunteers first on phase 1. And you know, these are very exciting times. And plus, thanks to your foundation, Spastic Paraplegia Foundation, the clinical centers are forming a cohesive unit, and then they are getting ready for the clinical trials. And that is extremely important because this is a rare disease, and some patient is seen in this hospital, the other patient is seen in another hospital, but there was not really too much teamwork or working together between those centers, and now you establish that. There are at least 10 different centers working together. They're getting ready for the clinical trial. They're looking for the, they already initiated the clinical trial. So they are building a platform for the next clinical trials to happen. And I hope NU9 would be the first one or the second one coming up in the pipeline. But once you form the pipeline, you will have many solutions going through that pipeline. Forming that pipeline was the tough work, and that's what you achieved, and I thank you so much.
I appreciate everything you just said. Those of us who work with the foundation and work with foundation members and potential contributors, we get asked often, well, explain to us what has our research accomplished? What has the money we've put into our research accomplished? And those of us who are not scientists and not researchers have a very difficult time specifically answering that question. But what I think I just heard you say is all the research that we and others have paid for in the past have gotten us collectively to the point where these exciting things may be just around the corner as we talk about compounds like NU9, as we talk about additional research in those areas, as we talk about clinicians who are assisting in creating that platform for clinical trials. So all the elements seem to slowly be coming together. And I'm sure none of us like to hear what I'm about to say, but it's just the way it is. The way this happens at the next level and the next level, it's a matter of money. Whether that money comes from grant resources, and that this foundation is working diligently to try to access better roads to NIH and other sources that have substantial amounts of money. But in the meantime, the contributions that all of us as members of this foundation and others make continue to be important as we build those bridges between here and NIH and clinical trials. So it's tough to say we need to continue to raise more money, but we got to get across these bridges to get to the point you're talking about, correct?
That's correct. That's very correct. So I know, with the foundation, the research grants that you have given to me, to other labs, are exceptionally important. And I know for the donors and patients out there, they may think like, where is this money going and how much contribute is this making? But I am happy to tell you that this actually makes an immense contribution, because the knowledge that we generate with those foundation grants lays the foundation for us to go for NIH grants, for bigger grants. If we don't have that support at the very beginning, we have ideas, but then the ideas need to turn into preliminary results. And if those preliminary results do not come about, then you cannot go and ask for NIH grants, for bigger grants, and so forth. So that's why the foundations are extremely important for those great ideas to nourish. And, for the patients out there, for the friends of patients out there, I think every support that you show to the foundation helps the scientists and the doctors to eradicate this disease, because we have to put money towards knowledge. And the more knowledge we have, the less disease we have. And that's why I'm here to help as much as I can with my artwork, with my donation, with my time, because as I said in the beginning, we are family. And when family gets together, family solves the problems. And I think this is the time that we solve the problem and we see a better future for everyone.
And this family is larger than even SPF. I mean, for instance,
I think so.
It doesn't matter to me which foundation or which foundations fund and find a cure. Those of us who have these diseases just want a better tomorrow.
That's true.
And so SPF came into existence, and its primary purpose today is to help fund this research. So we appreciate any contribution folks can make to go toward that purpose. But there are contributions in other places, and if those contributions go toward your research to find these cures and these treatments, we're thankful for that as well. So we invite you to consider anything you can do, whether it's with SPF or with another foundation that directly supports this kind of research and work toward treatments and cures. Dr. O, we love you, we appreciate you, we thank you for being here, and we look forward to continuing to work down this process. One of these days, we're going to come to an annual convention, and we're going to have balloons and champagne like a New Year's Eve party, because we are going to have found one treatment that makes a difference in people's lives, and it's going to be because of folks who contribute and people like you who are doing this research. So thank you for being part of our team and part of our family. We love you and we thank you.
Thank you so much. Thank you.
Thanks for being with us this morning.
Thank you.
Guys, you heard a great explanation. Dr. O said it very well. She's on the front line of that research, and so thank you for listening to that. And Dr. O, I'm going to invite here in just a moment Dr. Fakari to join us, and we're going to get him to talk to us about that clinical platform that you were talking about just a moment ago. With that, I want to say thank you to our PLS folks who are here. I'm wearing, as you all know, I'm an SPF and I have HSP. But we have the largest group of PLS folks that we've ever had, and they came armed with PLS T-shirts. Norma and I were honored that they shared those with us, and so today we are supporting this PLS group that's here, and those of you that are home. And we want you to know that we're going to do all the research we can, both in the HSP and the PLS front, to find some answers.
Thank you.
So, Dr. O, thank you, and I'm going to call in Dr. Fakari. We're not even going to leave the set here, just let him come in and take the microphone. A very good transition, as Dr. O has talked about this process that moves in the direction of our SPF CERN and the common work that you guys are doing, Doctor.
Sure.
I shared with our group a little bit earlier this morning that we had a little bit of technical difficulty at the end of yesterday. I had just told them that we were going to turn to early child onset, and we didn't get to do that, so I appreciate your coming to visit today.
Very happy to. Thank you for having me.
We just ask you to share with our listeners at home, and of course those who may listen next week on YouTube. If you can just summarize the work and the discussions that you and the parents of the children were having yesterday, we would love to follow up on that.
Sure, absolutely. Very happy to. And I'll start out by saying that I think it's wonderful that the SPF is supporting this effort. I think there are a couple of topics, and this is what the discussion focused on yesterday, that are very, very important to parents with children with hereditary spastic paraplegia. So we talked about the link to cerebral palsy, the delay in diagnosis for many, many children. We talked about our approach to symptomatic therapy, and then we spend a lot of time talking about sort of community support, how parents support each other, and we talked a little bit about research. So those were the sort of the big areas we talked about.
Last year we celebrated as we created the CERN, and so share with us, if you would, just kind of a quick summary of how things have gone in year one and where you see that going in year two.
Yeah, very happy to. So a high-level summary, the Spastic Paraplegia Centers of Excellence Research Network, or CERN, really was created with the idea of building a national network that can support clinical trial readiness for hereditary spastic paraplegia and PLS. And year one very much focused on doing the foundational work for this. So I always think about these larger projects, if you want to use a metaphor, like building a house. You first build a foundation, and that's what we did in year one. So we got together, we organized a group of 10 centers around the country, 3 centers who focus exclusively on childhood onset HSP, 7 centers who see patients in the adult age group. And year one was really spent on organizing the network, developing a central research protocol, which is a key step, getting this approved and rolling this out. So we're very excited that just a few weeks ago we were able to enroll the first patient at the Boston site. We're up to 5 patients as of today. So slow start, but we're starting. And we hope that the other sites will come online very soon.
Well, we're very excited about that process, and for our listeners at home and in the future, as the CERN group meets monthly to talk about progress and coordinate activities, 3 of your board members, Norma and I, and John Cobb, our research chair, and Frank Davis, a past president, we've been invited to sit in and participate, and it's good to just listen, and every now and then we can provide a piece of information that is helpful. But it's good for us, Doctor, to know the foundation that you're laying, the plans that you're making, and how that's consistent across all 10 of those clinics.
Yeah.
And as we hope that foundation builds more clinics in the future.
Absolutely.
So that we create nationwide this opportunity for, we hope, a quicker diagnosis.
Yes.
And quicker access to clinical trials as funds become available and that process moves in that direction.
Absolutely, and I think if I can add to that, and also building, I think, expertise in clinical care. I think you're absolutely right. This is just the first round. We have 10 centers. Hopefully in the second round there will be many more, so we can really represent most parts of the country. And I think over time what I've seen is that these kind of initiatives, they lead to more research, more awareness, more funding. They lead to better clinical care because people specialize in the care for individuals with hereditary spastic paraplegia. So I think all of these benefits will take time to build, but they will become I think, available to the community very soon.
As Dr. O and I just were commenting a moment ago, you know, those of us who are on this board and we're not scientists or doctors, and we get asked often, how can you tell us that our research that we have funded in the past has made a difference in terms of where we are? And I would just ask you maybe for your response in that regard.
Sure, it's a great question. I think funding through the SPF has been instrumental, not just over the last year, but over many years in moving the field forward. And it's really difficult to give this a metric, right? But I think the things that I'm excited about are clinical trials coming along? Are we seeing the basic science that will lead to clinical trials eventually? Are we seeing earlier diagnoses? And I think all of this has been supported through research supported by the SPF. So in science, we use metrics. We use metrics like publications, metrics like patients enrolled in studies and things like that. And all of that has been trending in the right direction with the help of the SPF. So I think those are sort of the things I would point out. And then, you know, there are a couple of milestones achieved already, I think. You know, for us in Boston, it was a major milestone to have a natural history study for all types of hereditary spastic paraplegia, something that didn't exist a few years ago. We are able to provide patients and families with unknown types of hereditary spastic paraplegia with the latest in genetics. And all of these things are only possible because there's a strong community and funding provided to these research studies.
And in terms of past funding and future funding, as we were speaking a minute ago, this family is a big family, and there are a number of foundations and places where folks can contribute toward HSP and PLS research. SPF is certainly one that's been around for a long time, and we solicit and we ask for every contribution we can and ensure that that's going to go toward this research process. But we also put our arms around and cheer on other foundations because in the end, doctor, if you find a treatment that makes my life better, I don't care where it came from. You know, we just want to see people's lives made better, and we appreciate the work that the new CERN group is making in building that block, and the research that we look forward to with clinical trials, hopefully not far down the road. I'll ask you one last question, and then I'll let you go. But I asked Dr. O this question as we started. What's the most optimistic, enthusiastic encouragement that you might give people in terms of where we are and where we go next?
It's a great question. There's a lot of things to be excited about, I think. But when I meet with families in clinic every week, a lot of what we talk about is the fact that we're at an age where we have novel genetic therapies available, therapies that not only treat the symptoms, but really get to the root cause of the problem. And this is something that didn't exist when I first started going to medical school. So science is advancing fast. Some of these advances haven't made it to HSP yet, but will make it eventually. And that's what I'm very excited about.
Well, thank you for your work. Thank you for your partnership here with us.
Thank you.
And we look forward to continuing work together until we cross a finish line that'll make a difference.
I very much appreciate it. Thank you very much.
So thank you very much, Doctor.
Thanks a lot. Thank you.
Well, folks, we are about ready to turn back toward the podium. We thank Dr. Fakari and Dr. O for joining us. It's great to have their expert input on what they're doing and where we're going in the future. But at this moment, we're going to turn it back to Tim up front and proceed with the next part of this program. Thank you for joining us. We'll see you later.
Take a second, get everybody back into their seats so we can get ready for our next presentation. Hey, real quick, I wanted to point out, you know, we have these bracelets at the tables for you guys, and you got some in your bags and stuff. These are really great tools to hand out to people when you meet somebody new and somebody asks about what your condition is or how you're feeling or what's going on. You tell them, you know, here, peel off a bracelet, hand it to them. It's got information on it. You can tell them to go to the website to take a look and learn about HSP and PLS and the Spastic Paraplegia Foundation. This is a great little tool to spread the word about us and what we do and about you and how you're doing. So we're moving right along. Coming up next is Dr. Piermaria. Here she is.
Hello, everybody. I'm I'm Emanuella, and I'm very happy to be here this year as well. It's my second time attending the SPF meeting. So I want to first thank the organizers for inviting me and for putting together an amazing meeting. I've really enjoyed it so far. So today I'm going to talk about some of the data we gather on the gene therapy that we are working on for SPG4, as well as some new ideas on how to also promote functional recovery on people that are already affected. In the lab, we mainly study SPG4. Dr. Baas talked extensively about it, so I'm not going to repeat too much of what you already learned from him earlier today. I just want to point out a couple of things again, that SPG4 is one of the most abundant form of the autosomal dominant HSPs. It accounts for 40 to 60% of all the HSP known forms, and there are more than 200 mutations in the SPAS gene that cause SPG4. And the SPAS gene encodes for this protein you've heard from him that is called spastin. And what I want to point it out again here is this interesting thing that spastin encodes for two isoforms called M1 and M87, that they differ a little for their location in the cells as well as for their function. In the lab, we made a while ago, before I even joined the lab, this mouse model that we call the SPAS C448Y. And this model was made by Dr. Baas in order to study the gain-of-function toxicity. So we introduced the human mutated spastin in addition of the endogenous spastis on the purpose to test the gain-of-function hypothesis. So Dr. Baas already talked about it, and again, I'm not going to repeat myself. I just want to introduce the model that then we used for the gene therapy, as this model show behavioral defects and anatomical changes that are similar to the one that are described for the SPG4 patients. So that's why we decided to move with the gene therapy with this model in particular. So let's look a moment at the work we have done on a preclinical model for SPG4 using this animal. First, I want to point it out that the gene therapy is a concept that started earlier in the 70s, and throughout these last few decades, there were numerous attempts and numerous studies done on viral vector-based gene therapy approaches on a number of different diseases. All these studies has demonstrated us how effective the gene therapy is and clinically safe. So when we started to think to the SPG4, we thought that using a gene therapy approach to replace the mutated bad gene with the correct one was a therapeutical avenue that can be pursued. So we collaborate with the team at UMass. They are experts in gene therapy, so they help us in designing and in strategizing how to use this vector. We have a dual vector in which there is a component that can recognize the endogenous spastin and so remove it, silence it. And then the other component of this vector, it helps in re-expressing the correct spastin. So it's a classic gene therapy approach in silence and replace the bad gene with the correct one. How did we do the testing? So we started with pre-symptomatic animal at P0, P1, so they are very small pups. We injected this animal with our spastin vector. You can see here they were going under anesthesia. And then we let them grow, being in their home cage, and then at the 3 months and 6 months of age, we tested them on the catwalk because these are two of the timeline in which we start to see early symptomatic signs of gait alterations and later symptomatic. The catwalk is this, is this device that has been used for a number of studies on rodent, especially to assess gait alteration, and is an automatic device, so it's very unbiased, and you can use to draw a lot of different parameters. After the 6 months, we collected the tissue and we move on with further investigation. So first of all, we look at the spastin expression. I'm not going to stay too much on this slide because I just want to show you how our very first attempts in understanding the level of spastin in the disease animal as well as the animal that were injected with the vector were, and how difficult and hard it was for us to detect the famous M1 isoform. So thankfully, we have recently received this first batch of polyclonal M1 antibodies, and we were very excited to test on this animal because it's just so important for us to understand how much of this spastin we are replacing with the vector. So as you can see here, there is a much better Western blot showing you the M1 band, bright and crisp, and then we have the M87 band also present, and GAPDH is used to control the amount of protein we are introducing. Now, the interesting things here is, as you can see in the B and C graph, the M1 and M87 expression is enhanced in SPG4 animal, specifically, or actually particularly, the M1 expression is really high and significantly higher than the M87. But when we injected the animal pre-symptomatically with the spastin vector, what you can see here with the yellow bar graphs is a restoration of the spastin level to a normal condition, suggesting us that with this vector we are able to restore the level of spastin just as much as needed, so not too high or not too low. And it has been one of the most exciting results we have had so far, and it's also highlighting how important it is to have the correct and the right tools in order to study this disease. So axonal degeneration can be studied using immunohistochemical technique, and HSP is known to have cortical spinal degeneration as one of the main characteristics. So how can we assess axonal degeneration? We can use markers that helps you visualize what's going on in the tissues. And one of these markers are neurofilaments, which are architectural structure. And it has been shown in a numerous of studies on other neurodegenerative diseases that an increase in these phosphoform of the neurofilament is associated with degeneration of the neurons. So we decided to use these as a marker to assess if also in our animal we have degeneration of the neurons, as well as if what happened when we introduce the spastin vector on this animal. As you know, the main feature is degeneration of the cortical spinal tract. So in the mice, how can we look at the cortical spinal tract specifically? Here is a cartoon of the cross-section of the spinal cord of an animal, and as you can see in the red circle, there is where we focus our analysis, because in the dorsal column of the spinal cord of this animal is where the cortical spinal tract pass by. It's a little bit different than human people. So how do you visualize these? As you see, you have these green dots that are stained for the phosphoneurofilament, and in the SPG4 animal, these green dots that represents the axon are much brighter. So that means this phosphomarker is increased, and that is telling us that these axons are degenerating. And you can see here in the graph. But when we use our spastin vector, these bright spots are not anymore as bright and are actually more closer to what is the control condition and in the healthy animal. Okay. Then we assess using behavioral device, as I said earlier, if like we look at the behavior to understand the gait alteration that are in this animal. And one way to look at them is using the catwalk that I mentioned earlier, because the catwalk has all these different parameters that can help us in understanding how these animals are walking and if they have problem, right? So I'm not going to go into the specific of each of these parameters. I just want to point, we choose those four mainly because if you want to describe a way to how the animal walk or also how people walk is by looking at these parameters. And these parameters were actually also used to assess gait alteration in HSP patients. So one more reason for us to use this parameter and look specifically at this parameter when we use the catwalk to assess the behavioral phenotype of this animal. Again, all these graphs, I just want to point out a few things and not going too much into the all little details. But as you can see, the wild-type animal, they don't show much of a difference between their 3 months time point and the 6 month time points. They just walk fine, they are okay. When you look at the SPG4 animal, you start to see an increase in this bar graph that is telling you these animals are having problem in their walking condition, and this is represented by almost all these parameters. These issues, issue in the walking condition are like present at 3 months of age as early symptomatic stage, and they get worse at the 6 months time point. But happily, we saw that if we introduce the gene therapy vector on this animal as early as P0, P1, so when they are just born and they don't show any symptoms, we are able, as you can see by all these graphs, bar graphs appearing, that these animals, they don't show anymore these phenotypes. So that is telling us once more that this vector is helping in preventing the manifestation of the symptoms. So this is all very exciting, and we are still working quite hard in refine any other tool we need to look at to make sure of how this vector is working and how can help, and we still have some check mark to do to make sure to present a comprehensive work. But it's all very exciting in seeing that as early as P0, we are able to prevent the manifestation of some of the main symptoms that are recurring in the SPG4 patient. Last year, when I was here and I was talking about some of these data, I got a question that since then, I guess, it stuck a little bit in my head, and one of the questions was like, and this question was essentially, well, that's cool, interesting, but what this can do for the people that are already affecting, or how this can be used on people that have already symptoms. So we talk a lot in the lab in how we can address this for the community. And so that's why the last part of this talk is just few ideas that we got in the lab that we want to test on SPG4 to understand if we can promote functional recovery on symptomatic SPG4 patients. So at Drexel, we are very lucky because Drexel is also the home of the Marion Murray Spinal Cord Research Center, and we have some of the best researchers in the spinal cord injury there, so that help us in discussing in a more broader way our own project and actually gain knowledge from them and try to apply what they learn in the spinal cord injury field to HSP potentially. So, in the last decades, the exercise therapy is one of the has shown the most promising data to promote functional recovery after spinal cord injury. The exercise therapy induced the production also of neurotrophins that in the end promotes the sprouting of axonal branches, so you can restore the route to the tissue target. Moreover, here it is. Treadmill exercise has been long used as a therapy for spinal cord injury rehabilitation. It's a very easy assay and is like, and it has shown a lot of promising data in promotes and restore functional recovery in spinal cord injury as well as in other disorders, not disorders, as well as in other, like in traumatic brain injury or stroke. And it also showed that can promote cognitive recovery and reverse a sign of inflammation that in general some of the signs you see when you have degenerating neurons. So what we thought is that maybe we can combine the knowledge that we get from the spinal cord injury field and apply to the HSP by using as Dr. Baas said earlier, a combinatorial approach in which we can use the gene therapy on symptomatic animal, in this case, to stop the progression of the disease, because I envision that by just injecting the animals, in this case, at symptomatic age, it might not be possible to have a full recovery, but it is possible to stop the progression at that situation. But maybe what we can do, as I just said, is combine the gene therapy approach with something like the exercise therapy in order to have maybe a little bit more of just stopping the progression of the disease, and maybe we are able to restore some of this function. So here I just highlighted like a brief bullet points, although they are not bullet points, it's just to show you how we are thinking to address to that. So what we want to do for the next step while completing all the work on the pre-symptomatic animal is starting to inject the symptomatic animal and then have another cohort of animals that are also receiving exercise therapy. So we are going to look at the early symptomatic animals as well as late symptomatic animals. As you saw before, at 3 and 6 months is enough for us to address these two stages. And we are going to inject them with intracerebroventricular injection with the vector. What we will do next, we will use the treadmill exercise by using another device that is called DigiGate. And the DigiGate is similar to what I showed you before, because it can help us in assessing gait parameters, but it also can be used as a treadmill, so we can exercise the animal on these while getting also data, meaningful data on how their walking condition are over time while exercising. And then we are going to look at the morphology of the motor cortex on these animals. So we are going to collect CNS tissue and look at like the morphology in order to study using immunohistological technique again, plasticity and axonal sprouting, as well as we can apply tracers that allow us to visualize a specific tract and look at if there is any sprouting happening after exercise therapy, as well as we are going to investigate the expression of neurotrophic factors that are beneficial in the restoration of these functional recovery. And in particular, we are looking at some of these neurotrophic factors that have been shown in literature that promotes restoration and axonal sprouting, but like we will look even more deeper in, for example, also molecules that are called morphogens, so that they gives to the neuron some cues to understand, basically, they guide the neurons that are regrowing towards their target. So all of this is just our preliminary idea and attempt to try to understand how to help also the community that is already symptomatic, and it's just like a further step towards understanding how to give tools to the community and like get some beneficial effect. So with that, I would like to thanks all the people that work with me on the gene therapy project, our collaborators at UMass, especially that they help us in designing the vector, Dr. Miguel Esteves and also Dr. Heather Gray-Edwards. Obviously, none of this would be possible without the funding agencies, the Cures PG4, the SPF, and the fellowship that I got from Drexel University. And thank you all for your attention, and I'm happy to take any questions.
Pardon me if my question is elementary, but my, I guess, version of hereditary spastic paraplegia is I have SPG4 and I have like a deletion in my gene. Mm-hmm. But I hear a lot of you guys talking about like mutations. Is that different?
Yes and no, in the sense a deletion is anyway considered a mutation. It's different. So a deletion means that you have eventually a stop codon that is like not, let me see if I can find simple. So basically, you are not producing a full-length spastin, but just a piece of it.
Thank you.
No problem.
Any other questions? Okay, we're going to take a quick couple of second break here, and we're going to make a changeover for our next presenter. Dr. Piermaria, thank you very much.
Thank you, everyone.
Well, I got a couple of seconds to fill, which you know I can do. Remind everybody, remember, we still got the potato pants out there, so if you haven't had a chance to put those on and walk around in those, please do so, okay? Because it's a really, really cool thing to do to kind of feel and understand what it's like to have an upper motor neuron disease that makes it difficult for your ability to walk. Also, remember, there's still time for raffle tickets. I'm telling you now, later when you see what we're raffling off, you're going to wish you had more. I'm just saying, to increase those chances. Remember, you can't win if you don't play. We still got t-shirts out there for sale. We got some retro looking ones, which are really cool. We still got some of the Tampa ones for this year. So make sure you get out there and get those, okay? Also, we have the caregiver items that we talked about earlier out there at the table. You can take a look at that, things that can help people throughout their day. We brought those so you can browse those and find those out there on Amazon and stuff. So we're getting lined up here. We're getting ready to go. We're looking pretty good. Here we go. Are you guys ready? Here we go. Ladies and gentlemen, Dr. Qiang. Here he is.
Good morning, everyone. It's an honor to be invited to the SPF conference again and to present a small portion of my work in the lab. I study SPG4 in the lab, and that's just a small portfolio of my research projects. I wish I had more time to share more data with you guys, but unfortunately I have only 20 minutes to go through some really important things and updates I want to deliver to you guys. And would like to take more questions from you guys, and that could be very, very informative and inspirational for my continuous research on SPG4 and HSP. So I'm going to shift the gear a little bit today from animal models to human stem cell models. So my lab is dedicated to use human stem cells, particularly patient-derived stem cells, as models as well as a therapeutic sort of approaches to study the mechanism of the neurodegenerative diseases, as well as sometimes to treat neurodegenerative diseases. So what are the stem cells? I guess everybody here might have heard stem cells before. Stem cells has really two major characteristics. One is the stem cells can be reproducing itself. We call it self-renewal. So if you have stem cells, the stem cells can actually reproduce identically to daughter cells that represent exactly the same thing as a mother cell. The other really important characteristic about stem cells is it can differentiate it into functional and mature cells that carry out major functions in your body. For example, a stem cell can become a lung cell, can become a brain cell, can become a kidney cell. So these are the stem cells, the characteristics, and they're so important. And then kind of our body was built up from different stem cells when we were just a zygote, starting with a zygote, with the sperm and oocyte and combined together. The old dogma in the stem cell field is when your stem cells differentiate into the somatic cells, we call it functional and mature cells, there's no way they're coming back to the stemness. But the new trick is, for the past 20 years, we learned a lot from transcription factors in our nucleus that can reprogram our somatic cells. For example, our skin fibroblasts, or the blood cells you can draw from your peripheral blood, and then using some strategy to turn these cells into a stem cell stage. So it's a reverse your natural differentiation process to go back. And that really kind of is revolutionary in the biological field. Now we can turn, let's say, my skin cells into my embryonic stem cell stage, and then I can develop these cells into any type of the cell, compose my organs or tissues in the body. So technically, we call these stem cells induced pluripotent stem cells, and that's what I'm going to use this time as iPSCs. So why do we need human stem cell or HIPSC-based model for HSP or PLS? That's a good question. And for years, and you hear today and yesterday, a lot of talks from scientists using transgenic rodent models. They're really powerful and very, very useful to study disease mechanism, to screen drugs, to kind of carry out a lot of experiments to answer these questions for us. However, there are inevitable hurdles and limitations of the animal models, especially the commonly used transgenic rodent models. The first one is create each animal model, it's very costly. It takes like $20,000 to $30,000 for one strain of the animals. And that's just one mutation, let's say, if you want to put it in. The second thing is there is a big difference or nuance, I would say, wouldn't say big, a nuance difference between the genetics from the animals, especially rodents with the humans. And also there are physiological differences as well. When I say genetically, it means the proteins that compose our body, let's say spastin, elastin, or other SPG4 or SPG mutations, the protein you have, there are nuance difference in their sequence. And these nuance difference can be actually significant in the end to carry out the functions of the proteins. So the proteins in the rodents doesn't really equals the proteins in the human body. And also, particularly in the human cortical development, especially the motor cortex, it's way more complex than the rodent models. And last but not the least, the CST, the cortical spinal tract in rats and mice are very different than the humans. They innervated differently. They don't, in humans, most of the cortical spinal tract, they innervate lower motor neurons directly, but that's not the case in the rodents. And the third thing is there are a lot of clinical trials that failed on the drugs that showed really good efficacy in animal models. We still don't know why, but that's like the fact we have to face with all the drug development. The last but not the least, I think is a really important point here is you can't use animal models to study undiagnosed or sporadic disease. That means these disease cases are without known gene mutations. If you don't know the mutations, how do you model that in the animal model? You can't. But the human stem cells make it possible, especially from the patients, because you don't really need to know what the mutation you have. You just need the human cells from the patient. So more rationales to use the human stem cell-based models. Of course, I covered 50%, more than 50% HSP and PLS have unknown genetic mutations. That makes a perfect sense to use human-derived stem cell models to study the disease. The other thing is we have a really, really low amount of the post-mortem patient brain and spinal tissue to kind of study the disease. But that can be changed by the human stem cell technology because you can literally create your patient tissue, although it's probably more embryonic or developing than the mature and adult, but it's from the patient, as the brain-like tissue. The third one will be these taking the cells from the patient, really realize the personalized medicine and precision medicine that can be reflected on drug screening and toxicity test to see whether this drug is particularly effective on this patient versus the other patient. What to do with that, to use your own cells, to use your own cell-developed brain tissue to test these things. And how are we planning to use the human stem cell iPS cells to study HSP and PLS? So here we go. We generate human brain organoids enriched in upper motor neurons to study these diseases, particularly in my lab to study SPG4. What are human brain organoids? Those are miniature brain tissues that are small brain-like structures with essential brain cells. For example, upper motor neurons and the supporting cells like glial cells and different type of neurons in your body as well. And then they are not just mixed together, they actually are arranged in an orderly and a complex manner, really kind of give you some presentation of how the human cortical tissue developed. So we have two strategies in my lab actually to create two sets of the stem cell banks. So one we call this isogenic stem cell bank. These isogenic lines were developed from one healthy donor. And that could come from the blood and could come from skin biopsy or the skin fibroblast. We could grow them up, and then we can use our technology to turn this into embryonic stem cell-like stage, we call iPS cells. At this level, now we can introduce different mutations. by the Nobel Prize winning technology called CRISPR. So that is so powerful because from this one donor cell line, we can create as many different mutations as we can to represent all the people here, the mutations you have, using CRISPR technology, but with the same genetic background, which is we call identical genetic background, except your point mutation or other mutations you have. And then we can turn these iPS lines into human organoid, brain organoids enriched in upper motor neurons for disease modeling. This is so powerful because now it gives us the tool to study genotype-phenotype correlation. And what is the genotype-phenotype correlation? I heard a lot of people are talking, I have SPG4 mutation, but I have a deletion. I have SPG4 mutation, I have a missense. Are they the same? Are they presented in the same way as a disease mechanism? There used to be a lot of questions on studying these things, but you can't really put them into a very rigorous studies without having the same genetic background. Because as human, all of us have a different genetic background. You don't really know what causes the diseases from the mutation or something else, because we're all different. But in this technology, we can actually study the same thing, under the same genetic background, just that mutation you have. The other step we have, starting to develop in the lab, is we take patients' fibroblasts from different sources. And these are patient directly from the patient, the skin fibroblast. Now we turn into iPS, stem cells. Now we make organoids. And these organoids are actually just like similarly to your brain tissues, your own brain tissues. And then these are from different patients, from different type of mutations and different gene mutations, we can do that. And this really gives us to precision medicine or personalized medicine because these are directly from you patients. So here are, if someone can hit the first figure and then let it run. So these are how we generate the organoids in the lab. We have a bioreactors. You can see each of these little white dots here. They're representing the organoids and the brain tissue, brain-like tissues we're growing in the lab. The left one is a little bit younger. You can see they're smaller. The right one is a little bit more mature. We now can grow these organoids up to 12 months in the lab. But today we're going to talk about some of the organoids we used for analysis. These are from 3 months, 6 months, and from 12 months. So after we, develop this strategy to develop the human organoid, brain organoids or cortical organoids enriched in upper motor neurons, we want to characterize it to see whether these are having the characteristics of our human brain. And here is the first picture you see here. You see these little red loops. Those are neurogenesis site, and which means that your neural stem cells developed these little loops that give rise to neurons and or nerve cells and glial cells, which are supporting cells, and to kind of build up your brain. These are the little bit older organoids. You can see the mature nerve cells migrated out of the regenerative zone, we call this, and then just form this kind of cortical plate-like structures with axon like extended, like the cortical plate structures. And we really want to make sure our organoids are actually representing upper motor neurons here because that's the disease we're targeting. So we use a CTIP2 as the nuclear markers to see, do we get enough CTIP2 positive upper motor neuron-like cells? And we do. You can see here we have, we quantified it's probably 20% to 30% of the neurons we're getting are CTIP2 positive. And you can see these, another gene is SPG1, it's called L1CAM. That's also highly expressed in our brain organoids, which represented these are very close to what we have so far. We call this cortical organoids enriched with upper motor neuron region. And I'm going to present some of the studies we did with the disease phenotypes, and I don't have time to go through all of them, so I'm going to skip the set 2 and focus on set 1, which is isogenic HIPS lines, and we're not going to do any deletion today. We're only going to just focus on a truncated mutation S245X and a missense mutation C448Y. I want to remind you guys, these are isogenic ones, so they have identical gene background, genetic background, only the gene differences on spastin. So one has a truncated one has a missense one. So we analyze a 3-month-old organoids to start. You can see here are the CTIP2 stainings, which are reflecting our upper motor neurons in these organoids. You can see control have a robust production of CTIP2. However, in both truncated mutation and in CY mutation, the missense mutation, we are losing the CTIP2 cells, which are very unexpected in our previous, before we did experiment, we were not expecting to see there is a loss of the CTIP2 cells in our organoids. And the other difference between these, I call this genotype-phenotype distinctions, is we did see the difference between the truncated mutation and the missense mutation, as the missense mutation have enlarged the cell bodies, which another sign of the cells are not doing well, doing worse, not just to have a lower amount of the cells, but these cells are sort of dying. Not all of them, a portion of the CTIP2 positive cells are actually undergoing apoptosis. Which really surprised me is this figure here. The GFAP is a marker for astrocytes. I'm not sure how many people are knowing astrocytes in our brain. We have nerve cells which carry out a lot of functions, but consider astrocytes as a supporting cells. With them, you will not have functional nerve cells to do your job. So these astrocytes are actually having 2 different directions in our mutations. In the truncated mutation, we have very, very hypertrophic astrocytes, which means astrocytes are not developed well. However, in the missense mutation, the CY mutation, we have hypertrophy astrocytes, means these astrocytes are hyperactive. They're actually probably more inflammatory. So that makes probably the worst of the condition of the missense. So we are still investigating on these issues on astrocytes in SPG4, which is not really considered as sort of a big studying point for a lot of scientists here, but I would like to go deeper or dive deeper into the mechanism why the astrocytes are showing these 2 different directions in the 2 different mutations in SPG4. Last but not the least, for the genotype-phenotype distinction, we see there is a different level of degeneration happens in the 12-month-old organoids. When you see here in the middle is the truncated mutation, and then all the way to the right is the missense mutation. The truncated mutation, both of them, truncated mutation and missense mutation, shows a little bit increased markers for neurodegeneration. We call this SMI32. And these are our markers, or we consider that as a readout for our therapeutic interventions in the next slide I'm going to show you. So I hope I can deliver this message as in the truncated one and in the missense one, they both show the degeneration markers. However, the missense one again shows even worse degenerative markers than the truncated ones. So now we get into the therapies. So previously, collaborated with Dr. Baas, we find HDAC6, the histone deacetylase 6, is probably a therapeutic target for SPG4 specifically. And then I have this wonderful models, then I can test whether these HDAC6 can actually act as the therapeutic target with small molecules. So what I did is I validated this HDAC6 change in these mutated organoids. Interestingly, at 3 months old, only the missense mutation shows there is abnormal increased functions of HDAC6. However, in the 6 months old, the truncated one catching up. So you also see the abnormal increase of this HDAC6 activities. Very interestingly, we obtained human brain tissues from one male post-mortem brain and one control, age-matched control, the same region, and we run the HDAC6 activities in both. Interestingly, I want to point it out, these HSP patients we got from NIH, they actually don't have the SPG4 mutation. And we did our best to sequence this, the brain, and we find there is one very rare SPG4 mutation in this brain. It's called CTC1. So it's a newly found HSP gene, and actually it's mutated in this brain. However, no matter what, we do see an HDAC6 and increased activities in a non-SPG4 brain. HSP brain, which is quite exciting for us to point at HDAC6 might be a universal target for the whole HSP spectrum, but it's very preliminary. And we use our drug called tubistatin that's the drug that can inhibit activities of HDAC6. We want to validate whether this drug is doing what it's supposed to do, and we see, yes, it is. It inhibits HDAC6 dramatically. Now we look at the phenotypes. So we put this drug in different concentration, different durations of the preliminary data, but I'm not gonna bore you guys with all the experiment conditions I have. I just want to show you here with the one examples with the missense mutation. We put the drug on. You can see very obviously the degenerative axons marked by SMI32 was much reduced with tubistatin treatment, which is inhibit the HDAC6. So hopefully I convinced you guys the novel human models derived from patients are really essential to model the HSP and PLS and other neurodegenerative diseases. And particularly we find a way to culture these human stem cells and to upper motor neuron enrich the brain organoids, and that's unprecedented. The second thing is, I think I delivered some preliminary data to see there is a genotype-phenotype distinctions on the case of SPG4. We think missense mutations, especially what we're looking at, has a severe phenotype than the truncated mutations. The third, but not the least, is the HDAC6 we think is a very promising therapeutic target. And we actually not only see that in our SPG4 cases, but in the different HSP cases that's increased, and are really offering a promising therapy. So the ongoing project in the lab is to test new druggable targets to treat HSP and SPG4, and we have several novel targets now identified by our high throughput analysis in the lab. We're testing it actively. The second thing is we want to collect the SET2 IPS, which is from the patients, to really expand our portfolio or our reservoir of the IPS banks. The third one is we're actually collaborating with Dr. Baas and other people as a multi-PI collaboration to test the gene therapies using our human patient or patient-derived brain organoid models. I want to thank my lab. Everybody in my lab worked really hard on different projects, especially big shout out to Neha and Skanda, who are the pivotal personal force to push the project forward. I also want to thank all my collaborators at Drexel, around the world in UMass and Texas to help me. Without that collaboration, my research won't get this far. I really want to thank NIH and SPF Foundation to give me the resources to study SPG4 and HSP. And also a big shout out to the foundations led by the parents of the childhood onset SPG4, the Cure SPG4, the Lydia and Blair Foundation, and Maury Coterie Foundation. They connected me with the different scientists around the world to build up collaborations, to talk to companies, and then I can't thank them enough for me to connect with the different scientists. The last but not the least, I really want to thank Spastic Paraplegia Foundation to give scientists a platform to interact and meet patients. I think this is so essential. I have been to this meeting only from last year and this year. I can't tell you enough like how important that is, this interaction and meeting is, to meet with the patients and patient families. I had a lot of good conversations with the patients and patient families, and really kind of inspired me, on different levels about my research, about the questions I should answer, I should try to answer, and transforming sort of like what can feel like dry and routine bench work as a scientist in the lab to really motivating and impactful endeavors to try to make things better for you guys. I mean, absolutely, that's really, really essential to kind of like, to push the therapies, to push the disease mechanism studies towards what we really want to be. I can't thank enough about this platform provided by the scientists and to interact with the patients. So I stop here and I'll take questions, if any.
Okay, Oscar, I got one right over here.
Excellent presentation. Thank you so much. Every year you present very good data, and
thank you.
It's exponentially growing, so thank you. Maybe I didn't understand, so if you could clarify for me. So you said HDAC6 could be a therapeutic target. So do you want to reduce it? Do you want to upregulate it? Do you want to improve interactions? Like what kind of a target do you envision, and based on which result?
Yeah, based on, first of all, we find HDAC6 was hyperactive. And we did Western blot, look at the protein levels. The increase of the protein levels is moderate. So we don't have a lot of proteins in there to make it hyperactive. What I think, we don't have a good antibodies to look at phosphorylation and modification on this protein, but we have a really reliable kit to look at the functions of the protein, which means the activity of the protein. We see the activity is highly increased in our SPG4 animals. Now we also replicated that in our brain organoids derived from SPG4 mutations. So I think as a target, we need to turn it down. We need to turn it down. And also, as HDAC6 has been shown in other diseases, for example, the Parkinson's, as well as the Alzheimer's, the hyperactivity of the HDAC6 is also in those diseases. Okay. So there are the companies actually are developing HDAC6 inhibitor drugs to treat cancer, to treat Alzheimer's, and also I know there are a couple of companies, they also are trying to use HDAC6 or other HDACs to treat ALS. So we can probably connect with them to see whether we can collaborate with one of them.
Yeah, but then be careful, because the activity may be a compensatory mechanism. Because it's active doesn't mean it's toxic. So it could be that the cell or the system activates it to overcome some of the challenges that it has. So
that's
everything that goes up in a high expression doesn't mean it's bad. So it may be it may be that you actually may want to increase it even further to help. So, just because it's high may not mean that's bad.
Yeah, that's not the case in our experiments. When we treat with an inhibitor, which tubistatin we got from the company, and then we see the degenerative markers turn down. So.
Good.
Yeah, so that's we mark with the SMI32. So I think that's kind of promising. And also I didn't present it here. We have animal models collaborate with Dr. Baas and using his animal models, and we see when we inject IP with HDAC6 inhibitor, tubistatin, the same drug we use in organoids, the behavior of the animals were rescued, as what Emanuella was showing with all the parameters with the catwalk. So we think that's actually a positive side.
Excellent.
Thank you.
So for the organoids, I'm going to ask a time question. Let's say if someone walks in the clinic and then you said these are derived from fibroblasts. Some of them are fibroblasts. Okay. Yeah. So let's say you take a little bit of the fibroblast from a patient or from a person. So you develop organoids. What is the time frame? How long does it take going into clinic, giving a fibroblast?
So from the fibroblasts to the organoids? Mm-hmm. So we turn fibroblasts into iPS, the stem cells, within a month. And then we have to validate everything, no mutations introduced, the chromosomes are normal and things, just quality control, take another month and a half probably. And then after that, then we can expand it, and then we can put into our liquid nitrogen tank for unlimited use in the future. But now we can start to differentiate into organoids. So the organoid differentiation takes, the younger ones takes a month to get to the younger, really, really young organoids, very teeny tiny little things growing in the bioreactor. But we managed to grow them out for 12 months. So we can stop in whatever time you want to do longitudinal studies with a different age of the organoids. What I presented here mostly are 6 months, 3 months to 6 months old organoids. So that's the timeline we have for the human stem cell models.
And then from one fibroblast, it's just one organoid, or you have hundreds of organoids?
Oh, hundreds.
Hundreds of organoids.
So each generation with our bioreactor unofficially counted probably 60 to 90 little organoids growing on. You can see
and all those organoids are similar to each other?
Similar. They're very similar in the size. We have this really wonderful bioreactor in the lab that can consistently generate the size comparable organoids that kind of mitigate a little bit about variations between batch to batch.
Excellent. So then going from fibroblast to having your first young organoid is about 4 months.
Yes.
And then you can grow them for an extension of a year.
Yeah.
That's actually very nice because you can use all these individual organoids for drug discovery purposes, maybe.
Yeah, that's the goal is to generate as many as we can and to do a lot of screening on those.
Ding, ding, ding.
Well, there you go, guys. You get to see researcher to researcher interaction right there, live in person in this room. That was pretty cool. We're going to do another question back here.
Thank you so much for that presentation. I thought it was actually surprisingly easy to follow. So really, really appreciated all that. And also very exciting. I mean, it sounds like, significant progress in, SPG4. So I mean, I have a couple of questions, like you mentioned with this HD6. Well, first question is there anything else that you see in the cells, or have you studied, anything else in the cells that's showing up that could also be, causing the degeneration? And it sounds like this theory with HDAC6 did pan out, and you're seeing the results with this. You said it's a statin treatment?
Tubistatin.
Okay, tubistatin. So then, okay, so the first question is there anything else? The second question is, what are the next steps? Because it seems promising, and you found a drug that addresses it.
So yeah, love these two questions. The first one is we actually are doing very, very unbiased high throughput analysis on the organoids on our isogenic lines. Combining the proteomics and single cell RNA sequencing, we have several druggable targets we recently discovered. We're collaborating. I'm not an expert on some of the targets. I can't elaborate too much here because, with the time constraint. I collaborate with some people I can find around the country, like we're expert on these targets. We're actually doing more analysis. I submit some grants just to try to see whether these are actually promising targets for future testing. That's one thing. The second question I think you have is what's the moving forward? What's the strategy and things like that? I actually talked to two companies, and one of them I think the Liang and Blair Foundation, the founder Chris here connected me with the ALS the drug and the developed ALS drugs. They actually have HDAC drug. They actually doing that with the patients with the ALS. So we recently talked to them, and then there's some kind of nuanced things we need to take care of. But I think to talk to the pharmaceutical companies or biotech companies to repurposing whatever they are using for testing or for already approved is a way to go. And then that could be really fast and testing with the really, really early phase 1 studies on these compounds, whatever new compounds they have to inhibit HDAC6. The other thing is I also talked to big pharmas like the Merck and Pfizer just to see whether in their portfolio do they have drug of interest. Although these big pharmas, they're concentrated on big disease like Alzheimer's and Parkinson's, but they might have something they can also repurpose in terms of treating HSP, SPG4. So I'm kind of, anybody who has connections, feel free to contact me. I would like to talk to people who have, the information like that, just to moving forward. We want to put into clinical trials as soon as possible.
Great, everybody. Thank you very much. Dr. Qiang, thank you, sir. Good job. Okay, guys, we're going to take a couple of seconds here before we get ready to do stretching. That's our next segment. Hey, but anybody who is hanging out in room 529 in this hotel, are they in this room? Nobody? I guess I get to keep this. All right, well, somebody lost their key card, and we had it at the table. So somebody around here is in room 529. We'll make sure we get to them a little bit later. Okay, guys, we're moving on to our next segment. It's stretching with our, you know him, you love him, Zachary Lipson. Here he comes.
Can I bring this up with me? All right, everybody, so we're gonna start with some stretches sitting down, and I'm gonna meet you guys there with the chair, and then if you guys would like to stand up, we will. Is there anybody new here who wasn't here yesterday? Okay, so we're gonna start sitting down. Obviously, feel free to stand up if you'd like, but just to meet everybody where we're at. So we did a little bit of this yesterday. Seated cat-cow, this is just how we're going to. Guys, I'm sorry, can we just, with the talking, try to lower it a little bit so we can all focus? Okay. So we're going to start with a little bit of cat-cow. So we're going to start by rounding our backs. And if you have your arms on your knees, as you round your back, you might feel your arms start to slide forward. And then we're going to arch our back. And as you arch your back, you might find your hands moving up your knees. And let's go back into our cat. So we're going to round our back. And arch your back once again. So I want you guys to do a couple of these on your own breath, now that we know what we're doing. So as you round your back, I want you all to inhale. And as you stretch your back out, let's exhale that breath. All right, everybody, let's bring our arms up for a big good morning stretch. Yeah, I feel it in my back. Everything's tight. Let's bring our arms back down. And one more time, let's bring our arms all the way up to the sky. And if you can, we're going to bend forward. If not, you can just stay where you are. And we're just going to hold it here for a couple breaths. You could just feel your back let go. Take a little bit of the tightness out. Feel some relief. And let's return back to our neutral position where we started before. And we're going to try some of our cat-cow again. So we're going to round our back. as we inhale, and then exhale as we straighten our back out. And once again, we're just going to do a couple on our own breath here. And once again, let's bring our arms up for a big good morning stretch. Drop our arms down one more time. Sorry, not one more time, because we're going up with our hands. And let's release. And once again, we can return to our neutral position. And we're going to bring, can you guys hear me good? Okay, we're going to bring our right hand out, and you can grab your fingers for a nice stretch in your forearm, but if you're a little too tight, you can grab one finger, you can grab a thumb, whatever works best for you. Let's switch sides. Now we'll bring our left arm across our body for a nice stretch. Then switch sides. Okay, now we're going to bring both of our hands up to the sky. We'll bring our left arm behind our back if we can, however far back you can bring it. And if you can, we'll pull a little bit on our left hand or our left elbow. And let's switch sides. And from here, we will return back to our stretch with the arm straight out in front. We're just going to provide some relief for our forearms once again. And switch sides. Now we can cross our arm to our body one more. Let's switch sides. And now last time through these two exercises, let's bring our arms up one more time. And drop our left hand behind our neck and pull down with our right hand if you can. Let's switch sides. Okay, and let's reset. So now I want everybody to look all the way over their right shoulder. And then we'll turn our necks looking all the way over to our left. And every couple seconds, I want you guys to keep rotating. And now, whichever side you're on, let's bring our head all the way up to the sky and reset down on the other shoulder. Now we're going to do that same thing going the other way. Bring our head all the way up to the sky and then reset down to the other shoulder. Now we're going to do this once again for a couple of breaths. And now, whichever side you're on, we're going to go the opposite way. So we've been going up. This time, we will go down. So if you're starting on your left shoulder, we'll look straight down to the ground and end looking across our right shoulder. Okay, everybody. Now we're going to do some stretches for our legs. So let's bring our left leg out if we can, and our right leg forward if you're in a chair and it's a little uncomfortable or you can't really move them. You can keep both arms forward. We're going to be doing, or both legs forward. All we're going to be doing is trying to reach our toes. But for those of us who have our left foot forward and our right foot back, let's just go and grab our left foot. The goal here is to have your left foot as straight as you can, and we'll just slowly reach for our toe, or if that stretch is enough, you don't even have to reach. Because I know I feel it instantly. If you're doing both legs, you could stay where you are, but if you have your left leg out, let's just switch. Now with our left leg as straight out as, or with our right leg as straight out as we can, we'll just reach towards our right foot. So just to let you guys know, in about 5 minutes, they're going to start coming in to set up for lunch. Don't worry. We still have 15 extra minutes. At 11:30, they're going to have us all leave, but from 11:15 to 11:30, there might be some people in here. We still have the room. And one more time through, let's just switch. So we're going to bring our left leg back to the front. And if you've been down this whole time with hands on both foot, take a sec, reset, bring your body up. And then one more time, let's switch. Okay, so here's what we are going to do next. For those of you who cannot touch the ground, we will alternate our feet kicking straight out like this, if we can. If you have your feet on the ground, we are going to start with our right foot. Our right foot is planted right in front of us. Then we are going to take it, go as close to a 90 degree. I know we can't, but that's just the direction we're moving in. So for me, I can only go about 30 degrees, and I feel the stretch. So we're just opening up each hip. And if you're doing this with me, you can just keep bringing your right foot back, because it can get a little tense in your hips, so we don't want to overload anything. Now, if you guys are doing the right foot with me, let's switch and we'll do the left. Okay. Now we can return back to the front. So we're going to do a movement that has 3 different steps. Same thing, if you're sitting down, this will absolutely work. So like yesterday where we did a little bit of streamline, we're going to do that, except we're going to bend our body forward, or as forward as you can. So just be careful if you have people around you. Now from here, we're going to drive our arms back, and you should feel it in your back. And then from here, let's bring our body up, and we're just going to bring our hands straight up to the sky. And now from here, we'll reset, bend back down, drive our arms back, and bring your hands right up to the sky. Nice work, everybody. I want you to do about 5 to 6 of these, then we'll come back together. Nice work, everybody. Let's shake out our arms, shake out our legs. Doing a lot of movement so far. If you feel your hips are tight, try extending your feet out a little bit. Okay. Just want to make sure everybody has room. We're going to bring both hands up behind our head like we're taking a nap. And we're going to bend to each side. So maybe we can start with the right. And when you guys come up, I want you to try to tighten your core here. Tightening your abs will bring your body up. So every time we stop at the bend, we'll tighten our core and it'll bring us straight back up to the top. Okay, everybody. From here, if you are standing up, if you feel comfortable with this, we're just going to grab a knee and hold it. If you want to hold onto a chair, totally feel free. And we'll switch sides. And one more time through. Let's bring our right leg up. And now we will bring our left leg up. Okay, so with all of us sitting down, I'm going to give you guys an option. So it might be easiest for those of you who are sitting down to grab the chair here. So you could do one of two things. We're going to bring our foot. You can either do this with one or both feet. So whichever feels most comfortable for you. We'll bring our left foot up and then just try to extend it straight out. Yep. And if it's hard for you to actually bring your hip up, you can just kick. And if you'd like to do it together like this, you can. If not, you can bring one foot up at a time. Goal is to feel a little bit in our hips. Let's extend our arms all the way out, but make sure we're not hitting any neighbors. And with our thumbs up, let's try to extend our arms as far back as we can. Let's bring our arms forward. Let's retract them once again. Let's bring them in. We're going to do this 2 more times. All right, everybody, we have about 10-ish minutes. How do we feel about doing a little bit of boxing from yesterday? Yeah? We got a couple claps. Does that speak for the room? Okay, cool. So, as always, you can totally sit. Everything we're doing, standing, sitting, it works. So, little refresher. So, I'm going to say 1. I'm going to say the numbers 1 through 6. 1 is going to be our hand that is opposite from our dominant hand. So for me, that's my left. So this is a 1, and a 2 is the opposite hand from your left. So for me, my 1 is my left hand, and my 2 is my right hand. So we're just going to do a couple of those just to get everybody situated with the classic combination. So let's just throw a couple 1s and 2s. Nice work, everybody. And for those of us who remember from yesterday, let's do 1, 2, and 3. 3, if you don't remember, is that hook. So 3 is where we're going to take our non-dominant hand and almost move it like a hook to the middle of our body. So we're doing 1, 2, 3. Nice work in the front here. Nice work, everybody. Nice work. You got it. Now we're going to do 1, 2, 4. So the only difference here is we're going to hook with the opposite arm. So if my 1 is my left, then 4 will be a hook with our right hand. So we have 1, 2, and we're going to bring our 2 back, and it's the same hand, and we'll bring it out to a hook. This one is a little challenging because it's the same hand twice. So it might feel a little weird to get used to. Nice work, everybody. Okay. So now if you remember, 5 and 6 were our uppercuts. So 5 with our non-dominant hand is like a shovel coming straight up to our opponent, and then our 6 will just be our other side. So let's bring them all together. We'll slowly do 1 through 6. 1, 2, 3, 4, 5, and 6. Nice. Let's do it again. 1, 2, 3, 4, 5, and 6. Nice work, everybody. So another combo in there. We're going to do 1, 2, 5. Nice. See, we're all boxers. Nice work. And now let's try something different. Let's go 1, 2, 6. So it's just, we're uppercutting with the opposite hand. Nice work, everybody. Now we're just going to return back to the middle, and we're going to do 1 and 2. That is it. 1, 2, super simple. Okay, nice work, everybody. Let's shake out those arms. Now all together, we're just going to pump our hands all the way up in the air. Now let's keep our hands straight up in the air. Bring our right arm down. Bring the arm back up, and now we will switch. So we're essentially just switching which hand we're opening up with from the top. At whatever is most comfortable for you, you can bring your arm all the way down. If here's all you can go, or maybe halfway like a 90-degree angle. And let's pump out those arms one more time. I know we're starting to feel it, right? In those shoulders? Good. Won't last too long, though, I promise. Okay, now we're going to bring our arms out here and just hold them. Just hold them. It might sound easy, but now I know it's starting to get a little difficult, right? Just try to hold them out. Once again, let's open all the way up and bring those arms down. Nice work. Give them a little shake if you need to. And before we end off, we're just going to move those legs one more time. So if you're sitting down, we're going to do our stretch where we have our right foot forward and our left foot back. And with our left foot straight, we'll just reach down and try to grab. Now, if you're sitting, it might be easier for you to just grab both feet, but feel or do what works best for you. Okay, let's switch sides. And if you're standing up or sitting down, let's try to spread our feet a little wider than they actually are. If you're sitting down, you can just try little outward pulses. I want you to find a stretch that's outward enough where you can feel a little stretch in your hips, but nothing too crazy. If you'd like, we can go all the way down here. If you feel comfortable, just let go of the tension in your back. Let your legs support you. From here, everybody, we can reset. And that is our stretch. Thank you all very much.
Hey, everybody, I want to remind you that Zachary has his own breakout later this afternoon, okay? So if you want to do some more stretching, he's got his own room, and he's going to be doing his own show for you guys and getting you really going. So later this afternoon. So look at that. It's in your program in our last section of breakouts. Hey, while I got your attention, everybody, somebody left a really cool blue cane at the registration table. It's probably nobody in this room because nobody else wanted the key card earlier either. So, this was found in the parking lot. So, who knows? You guys, we're supposed to be sending home with you stuff, not bringing stuff back for us, okay? Okay, so just remember the model. But anyway, you're going to have a little bit of a break now, and then we're going to do lunch at 12 o'clock. So get outside, have some fun with everybody, get warmed up, and we'll see you in a little while. Thanks.
Well, as you guys see, we've completed our morning process here. I hope you're stretched out and loose and ready for an afternoon and a great day. It's great to watch Zach do that, and as you're watching, almost everyone in here is stretching out. And I just want to tell you, every year at the end of this convention or this conference, some of you give us, some of the people who attend, give us some constructive criticism back. They do a survey, let us know some of the ideas that they have about how we might make the conference better for them. And over the past year or two, a number of folks have said we do a lot of sitting and we get tight and we really need to move a little more. Well, moving from this room to others for sessions can be challenging, so we decided to try to find the opportunity to stand up, do some stretching here, and Zach has done a great job. And so just so you know, we do pay attention to those surveys and try to make this conference enjoyable for everyone. I want to tell you, as you listen this morning, it's encouraging to hear our researchers talk about the new areas they're researching, the new thoughts, ideas, premises that they're looking at. And for all of us, we're frustrated because this is not happening quick enough. We need treatments and cures today. But as you heard Dr. Ose and Dr. Fakari, all the research that's been done in the past has been the building block to get us to this point. And as we move forward with clinical trials out there being shaped with our CERN group and others, we are moving, we hope and believe, in the right direction. So thank you for being part of what we do. We need everyone doing all they can, whether it's financial contributions, whether it's involvement in our committee work, whether it's participating in SPF talks, whatever you do, whatever you can do, we need your partnership, your support, your involvement in this process. And this afternoon is going to be a great afternoon as well. with some of our researchers, Dr. Calame, Dr. Zuchner, doing some off-site work to us through Zoom. So you'll want to be here this afternoon. Before we leave here, we're about to break for lunch. We'll be back here at 1 o'clock. We've got a couple of other Tampa area supporters, helpers who've been part of helping us put on this conference, and I want to share some information about them to you so you know more about how that has happened and what they are doing and how they are helping us. But as we get ready to go to break here, I want to say a special thank you to a couple of guys officially known as Little Rock HD. But that's Jeff and Rick, our guys here. Just like Dr. O was saying, everybody here is part of the family. And these guys have been part of our family for many, many years, and we look forward to visiting with them. But most importantly, they do a great job helping us capture this information so that you can see it now and others can see it later on YouTube. So we thank you guys. And if you're out there and you need this kind of work, these kind of partners, give these guys a call because they are the best. With that, let me say goodbye to you just until 1 o'clock. Take a break, get some lunch. That's what we'll be doing here. And we will look forward to seeing you at 1. Take care. See you then. Well, good afternoon and welcome back to our 2024 SPF annual conference. I don't know about you guys, what kind of lunch you may have had at home, but this lunch here was good with a really rich chocolate dessert, and what I really need now, if I don't have a cup of coffee, is a good nap. But in any regard, everyone is still enjoying lunch here, and things are great here. I want to take this opportunity again to thank so many of our sponsors and supporters who have helped us put this conference on, and many of them right here from the Tampa and Tampa Bay community. And I want to introduce to you right now a young lady named Ansley Basham. And Ansley is with the Tampa Bay Butterfly Foundation. And you might be thinking, why would a Butterfly Foundation be interested in what we're doing with SPF? Well, when we reached out to a number of local groups and organizations about helping us with a children's program, something that children would enjoy, the Tampa Bay Butterfly Foundation stepped forward. And I think from what I can tell, in talking with Norma, you guys have spent a lot of time and effort preparing some things for our kids this afternoon. So first of all, thank you for doing that, Ansley.
Of course.
And then welcome to our annual conference, and tell us a little bit about the Tampa Bay Butterfly Foundation.
Of course, yeah. The Tampa Bay Butterfly Foundation is a local nonprofit. We focus on putting more wild Florida, like native plants, animals, back into Florida. And our main bottom line is getting rid of pesticides. So through planting natives and getting more species back into the area, a lot of people will cut out pesticides, which as we know is a huge health issue, causes a lot of issues for health. So we work with kids of all ages and adults from 3 to 103. We've had every single age in our facility. We talk about planting native to be able to attract specifically pollinators. So our main hook is butterflies, but that also includes all insects. And then because we have the insects, we also get some of the larger animals that people like more, like the cats and the birds, all that stuff. So our main focus is putting plants into Florida and then therefore helping everybody's health.
Well, you guys are the Tampa Bay Butterfly Foundation.
Mm-hmm.
Do you work just in the Tampa Bay area, or do you work all across the state? What's the area you work in?
So we work everywhere in the state. We've had places all the way up through Tallahassee. We've worked with the Wildlife Foundation on planting roadsides with native plants. And then people that come to our facility are from everywhere. So we get as far south as Miami all the way up to Tallahassee, almost Georgia. So we work with pretty much the whole state.
And when you talk about pollinator projects, those are getting more popular and more advantageous all over the country. Mm-hmm. Norma and I have worked for years with an organization that spans the 10 states along the Mississippi River, called the Mississippi River Parkway Commission. Mm-hmm. And one of the primary projects that one of our committees there, the Environmental Committee, has talked about for the past couple of years are pollinator projects all along the Great River Road corridor for a lot of different purposes. You mentioned one of the effect it could have on dealing with pesticides, those issues, but also preserving the bee population, the butterfly population, and they're attractive roadside kind of scenes for people to see. So a lot of good purposes in what you guys are doing, and I'm sure the folks in your area and all across the state are enjoying every one of those benefits.
Yeah, so a lot of people have started putting pollinator gardens around their houses too, despite HOA pushback. So they've kind of been fighting that. And one of the other goals, we work with a man named Doug Calame, who is a PhD, has a PhD in ecology or something like that. But his purpose, he wants to put as many pollinator gardens as possible in all these houses to make a giant wildlife corridor. So we not only work with like transportation networks, but we also are working with the homeowner to be able to get as much out there as possible and bring kind of the stuff that you can find out in nature to your own backyard.
Well, a very interesting project and process that you get to see a lot of activity and a lot of positive results as you see flowers and beautiful landscaping and so on, but knowing you're helping the environment and helping the animals, the butterflies, the bees as well. So great work. Well, I have to ask you this because I know, as you said, you're a nonprofit. Spastic Paraplegia Foundation is also a nonprofit, and we rely totally on contributions that come from folks who are interested in helping us get medical research done. What about Tampa Bay Foundation? Are you guys always willing to accept nice contributions that go toward your purpose?
Of course, yeah. If anybody wants to make a donation, you would go to butterflytampa.com, and you can click a link and make a donation there.
Very good. Ainsley, is there anything else about the Tampa Bay Butterfly Foundation or the projects and work that you guys do that I haven't asked you that we ought to hear?
We're always trying to reach out to as many places as possible, and we are very thankful for people that reach out with any questions that they may have. Even if you're not from Florida, we are wide-spanning in our knowledge. So we're happy to help anybody. You can just contact us whenever you feel like.
Well, thank you for being here.
Yeah.
Thank the Tampa Butterfly Foundation for spending time to put together some fun projects for our youth and our children this weekend. I know they're going to enjoy that this afternoon, so thank you guys for being here and for all that you do to make the world around us a better place.
Of course. Thank you for having us.
Thank you so much. Well, again, a lot of things going on here. We got folks finishing lunch. In just a moment, we're going to come back and talk with one of our other folks that we've gotten to know well. In fact, I see him standing nearby. So I'm just going to ask him to come on over and take the microphone, and we're going to talk a little bit more with someone. If you've been watching all day yesterday and this morning, we're going to introduce you just a little bit closer to this guy who's been helping us stretch and exercise. Zachary, thank you for joining us this weekend.
Absolutely.
And for the work that you're doing to help us think about moving and staying loose. Share with us a little bit about who you are, where were you born, why are you here doing what you're doing? I think your story is very interesting, particularly because you got HSP, you're SPG4 as well, and we happened to get hooked up as we were planning this conference.
Absolutely.
So let us know about you and where you've been, what you're doing.
So my name is Zachary Lipson. I'm originally from New York, but I've actually relocated down here to South Florida. So as you said, I have SPG4. And I've been here to help everybody stretch. For those of you who have been involved with us doing it at home, thank you very much. And I find it really funny because when I was younger, all the doctors, my parents would tell me to stretch. And I wouldn't. It was very hard for me to do it when I was younger. Not physically, just because I didn't want to do it. And I found it so, in my head, I almost wanted to laugh because it went from me being so hesitant to stretching to then all of a sudden being the one on stage bringing everybody through the stretches. And it's incredible. I realized that there was a lack of support in terms of physical exercise for people like us, because I would try personal trainers who didn't quite understand hereditary spastic paraplegia, and I got hurt every time. The first time I tried a trainer, I literally couldn't even walk, my hips hurt. So I'm really here to show everybody that there is a way to positively progress every day.
Well, Zachary, I'm SPG4 as well, but my family, all of us, our onset was somewhere between, say, 27 or 8 and the early 40s. You were born, and we're dealing with this from birth forward. But at your age, thank goodness your progression has not accelerated too quickly. But as you've grown up and you learned the stretching process, what difference does it make for you to do that every day? And why should all of our folks at home really focus on that? When people ask me what's the most important thing I can do, I tell them stretch as much as you can every day. But share with us why that's important to do it and do it every day as much as we can.
I think the important thing to keep on our mind, which it can be difficult, is positive progression. And positive progression is a principle that is always possible, even if there is no cure that exists for us. There are still things that we can do every day to help support ourselves. Now, for me, I partially do it out of fear, if I'm going to be honest with you guys, because I don't know what my future holds. And for me, I see stretching and fitness as a way for me to almost set up defenses for what can possibly come at my way. So stretching, working out, boxing, all of these things are meant to build supports for you. So if something does happen, we have that added defense and support in our body that will help us even just feel looser.
Well, Zachary, you, as I recall, you were a native New York. You grew up there in a different climate, colder a lot of the year. Here, you don't have to deal with that, and I'm sure that makes a little bit of a difference. But in terms of what you're doing, you're not just here doing this with us. You are trying to share these kind of stretching techniques, exercise techniques in other ways. Can you share that with us? And there might be some folks who might want to join you in that.
Yeah, absolutely. So after I graduated, or before I graduated college, my senior year, the way it worked was I had to be a participant in an internship. So through my minor disability studies, I got in contact with this organization called EPIC. And that is Endless Possibilities in the Community. And they were really the organization that helped me get my base in adaptive fitness. And what they are is a day program for people over 21 outside the school system. And they essentially are just providing support and something for people to do throughout the day. But this also includes exercise. So I was able to work with, my senior year of college, I was able to work with people who had anything from cerebral palsy to autism and Down syndrome. Now, 4 days before I graduated, my boss at the time, I was getting all ready to move back to New York. He says, "I want you to run our adaptive fitness program." I mean, obviously I said, yeah, it's everything I ever wanted. So I took over that program, and it gave me 15 clients. And over that year and a half, 2 years, we started to build our clientele. I'm sorry, I got so lost in this. What was the question you asked me?
Well, we were talking about what you do and whether there's a way that today for people to join you. Is there a social media spot? Is there a place people can keep up with what you're doing?
So over time, it gave me all of this experience in understanding how all different types of people move, walk, and act. So I started up in Delaware and New York, but the climate was very unforgiving for me. I hated the cold. It always made me feel tight, way tighter than I normally would, say, in the summer. So I relocated down here to South Florida. And this just helped me with my legs just feel looser. Now, it's not that I'm not tight. It's just they're looser than they would be in the cold, which just feels a little better for me. Now that I'm down here in Florida, I have started to virtually do what I was doing over in Delaware. So it's almost like a little bit of what we had over here that you guys were watching. It can be a mix of stretching, strength building, and even boxing. And we do this virtually on adapthletics.com or my Instagram, @adapthletics.
Well, very good. Zach, it's good to meet you, and I've told the folks at home that one of the things that's impressed me here with the stretching and exercise is I watch and observe nearly everyone in the audience is doing that with you. You know, many times when we've done this before, we've seen folks who just wanted to watch, they would not actively participate. But you've had virtually everyone in this room stretching, boxing, moving in ways to help them stay loose and stay refreshed for this meeting. So thank you so much for being with us, for what you've done here. We look forward to working with you in the future, and we hope that folks who are hearing about what you're doing can learn more about it in the future. And as they do that, stay loose and stay stretched.
Absolutely.
All right. Zachary, thank you so much for joining us. It's great to meet you.
Appreciate it.
Guys, this afternoon, we're about to move on in a few moments. This afternoon, you'll be hearing from Linda LaFontaine, who is from Canada, and we're going to be hearing about what she's doing in working with SPF issues in Canada and other organizations there. And as we said all morning, we all are brothers and sisters in this. There are international boundaries in places, but there are organizations in other countries who are raising money, working with researchers. And one of our goals here at SPF is to get all those people to talk together, to share information together, to work together, to cross that line where we do have a treatment, where we do have a cure, and collaboration is important. And so Linda's going to be talking to us about what happens in Canada that she's involved in there. And then Dr. Steven Zuchner this afternoon, Dr. Daniel Calame are just two of the speakers you'll hear from. You may have heard them before. They'll be joining us by Zoom, and you'll be able to see that as well. So a lot of good information coming this afternoon. We're going to take just another short break, as we kind of clear the room from lunch. We'll be back in just a few minutes with Tim introducing our afternoon program. We hope you guys stay with us, and I'll talk to you one more time before the day's over. See you in just a few.
And turn page. Okay. Okay, everybody, here we go. It's officially 1:30 in Tampa. It's 1:30 on a Saturday afternoon in steamy, warm Tampa, but we're in this nice ballroom getting ready for a presentation. So I want to introduce to you Linda LaFontaine. Come on, Linda.
Yes. Get up. Background. Good. Yep. All right.
Your advancer, just hit the right button over here to advance it and aim back there. And you're good to go.
Okay.
Ready? Let me give you a little tilt down. Just
Good.
there, and we'll be standing by.
Okay.
Enjoy.
All right, good stuff. So, let me introduce myself. I'm Linda LaFontaine. I'm a spastic paraplegia ambassador in Canada, and I'm a determined HSP patient. I advocate for HSP, I advocate for PLS, and other neurological diseases too. So thank you for having me here. In the room, hi everybody at home. It's fantastic. It's a wonderful opportunity for everybody. I'm going to read from my script today because I need to box myself in. I need to stay on time. I think I'm going to talk for about 25 to 28 minutes. So I don't know that I'm going to have a lot of time for questions, but that's why there's my email there. You want to know anything personal, you want to know anything about my science, I can nerd it up, I can talk about anything, okay? All right, I have a tendency to use way too many words. Yeah, so I have about 30 minutes. I'll talk to you in about 3 lumps or so, past, present, future. Now I'll tell you a little bit about who I am. I'll tell you a little about a timeline, how the SPF is involved with this, how I got to be here, where I am today with the cohort that I built or helped build with the CBIG in Montreal. Then I'll tell you a little bit about the biobank itself. I'll tell you about the specifics about the cohort that I've got there, that I've helped build. So I'll give you facts and numbers. Then I'm going to speak to you about the future. Now you've got to understand, I don't have a crystal ball. I'm kind of flying on the seat of my pants here. I don't have a blueprint. I don't have notes. I don't have rules, but whatever I do, you know, I always do whatever I can to advocate for HSP and PLS. Always, no matter what I do. Okay. I might do this in less than 25, 28 minutes, which would be good. Anyway, a little bit about me. I'm medically retired. What medically retired means is there's a medical reason why I'm retired. HSP. HSP is my medical reason. I'm a full-time wheelchair user. You saw me wheeling up here, but I stand all the time. My lower body, belly button down, awful. Weakness, spasticity, I lose my balance. Basically, the girdle muscles, the muscles that hold you up, they don't work. No. I've got serious bladder issues, kidney problems, stuff you don't see. It's not fun. But belly button up, I'm fine. If I could distribute the strength properly, it would be okay, but I can't. My SPG7 is mutated. I was diagnosed in 2007, working diagnosis in 2004, but I'm 49 now, so they didn't want to saddle me with a diagnosis of HSP when I was 29. I understand why now. I didn't understand then. But anyway, so I've been dealing with this for about 20 years. It's gotten progressively worse. It was a cane before, and then I went from cane to poles on either side. You see how I'm wobbly when I let go of the podium? Then I went to a walker. Now I'm in a wheelchair, and I'm in a wheelchair full time. In the house, outside the house, everywhere I go. But I have a do it anyway attitude. I just do it anyway. Doesn't matter. I don't much ask for forgiveness. No. I don't ask for permission. No. No. I just do it anyway. But I'm kind. I'm very kind. And I'm pretty much always smiley. Oh yeah. My mutation is pure and uncomplicated. Pure is that word means it's in the lower extremities. Uncomplicated meaning it's just HSP. It's not a touch of something else. It's just uncomplicated. That's what it means, uncomplicated. So far, there's no neuropathy, and because neuropathy typically means nerve degeneration. There's none of that, no pain. I don't know why. It's good. Just go with it. All right. So, a little bit of a timeline. I always wanted to do something impactful for all HSP patients. I just never knew what. I always heard throughout the last 20 years, throughout my whole HSP journey, I always heard, I can't wait for someone to do something, or I can't wait for someone to do something. I can't wait for someone to do something. I'm someone, and I'm doing something. So back in the summer of 2021, that's when I became an ambassador for the SPF. Being from Canada, there's two official languages. There's French and English. I've always seen all kinds of communications. They've always been in French and English. It was a no-brainer for me, right? And I'm French-Canadian. So, second nature, I just write in French. Well, through that simple act of writing in French, I communicated with a woman in Quebec. She had a charitable foundation, and she transferred it to me. It was really just that simple. Now, most of 2022, I spent it trying to figure out what it meant to run a charitable foundation, what it meant to write tax receipts, what it meant to all the administrative stuff that goes with running a foundation. And then where do my donations go? And that's when I found the Neuro in Montreal and Dr. Rouleau and all the cool stuff they do there. And I was introduced to their labs and to open science. And that just blew my mind. Everything that I found there was just fantastic. Okay. So I toured the labs. I was introduced to the concept of open science. I met Dr. Jason Karamchandani, and he's the neuropathologist and the scientific director of the CBIG Open Biobank at the Montreal, their neurology department. It's a hospital and institute. It's called the Neuro at McGill University in Montreal. Okay. In typical Linda fashion, I asked question after question after question. I just didn't stop. In November 2022, I personally contributed my RNA to CBIG. For me, it was just a simple blood draw. No big deal. I did that. I contributed to CBIG because I wanted to contribute to research. But only one contribution was certainly not enough. I mean, it wasn't going to affect HSPers, and that's what I wanted to do. So in June 2023, what I did is I started a cohort of HSP patients within CBIG at the Neuro in Montreal. at McGill University. So what we have now, I gotta aim it over here. Next slide. Can we do that? Yeah, okay. The details of what I've got right now. Can you tell I don't do technology? Okay, so we have 32 patients right now, HSP patients, that have done the same thing. They've donated their DNA. Of those, so far, there was 8 whole genome sequenced so far. Many patients had already enrolled in the CAN-HSP program, so they'd already been sequenced. There's scientific collaborations with Dr. Rouleau, Dr. Patrick Dion, Dr. Ziv Gan-Or, they're neurologists. Then Dr. Sally Farhan and, well, oh crap, I didn't put her on there. Dr. Alison Dilliot. Those are 2 geneticists. They're fantastic women. Oh, fabulous. Yeah, okay, we talked about that. So what is, I'll do it again. Next slide. No? Next slide. Can we, yes, okay, CAN-HSP. What is CAN-HSP? CAN-HSP is a nationwide coalition of doctors that study HSP, okay? Remember, I'm from Canada. That's what I mean, nationwide. It's collaboration between universities, between hospitals across the nation. The whole program was established in 2011. It was funded by the Canadian Institute for, no, not for, of Health Research, CIRHR. They found a novel gene in 2016. Look at my notes. CAPN1, and again in 2018 in AAAS. Now, the program never ceased to exist. It just kind of dwindled off. Now, with the new information that we, HSP patients, the 32 patients that were consented for this cohort, with the new information that we're putting into CBIG, now we're reviving CAN-HSP. So what basically, I understand that there's strength in numbers, okay? And what I'm trying to do with all of this is I'm trying to give researchers what they need. With what they need, they're going to be able to help us in turn, but what they need right now is information. You've heard it throughout the whole conference. They don't have a whole lot of information. We do. It's that simple. We're giving them information. The whole initiative that I'm putting together, it enables patients to play a greater role in research. It speeds up this research. It makes a link between patients, biobanks, and then researchers. And it's all done through open science, okay? And it's all in the field of neurological disorders, particularly with regard to HSP and PLS, and now other diseases as well. So specifically what I did for the 32 patients, they all contributed their DNA to CBIG Open Biobank. Okay? CBIG consented them, sent them saliva collection units via Canada Post. Patients sent it back. CBIG processed their samples. Now, what processing means is they de-identified it. So it is now raw scientific data. It is not about the patients anymore. It is about the disease. That's what I've got to share and collaborate. It's information about HSP. It's not about the patients. It's de-identified and extracted the DNA. That's what processing means. The DNA then was sequenced. We've only got 8 so far. And now the data is poised for research and collaboration. Now, sharing and collaboration is not about money. It's about advancing human knowledge through science. I understand that sharing and collaboration is difficult. It's a two-way street. I know I can do it, but I can only do it 50%. So what I mean by I mean the neuro can do it. They've done collaborations with Harvard. They've done it with Stanford University. They've done, I don't know, over 90 collaborations. They've done this before, okay? I can collaborate, not a problem. That's, so all of these 32 people that have been consented and are now my cohort, the 8 full genome, whole genome sequenced, that's up for grabs. I can share and collaborate with that. So what CBIG stands for, I'll go back a little bit. What CBIG is, it's a biobank and it's all open. CBIG stands, it's an acronym, it stands for, the C is for clinical, the B is for biospecimen, I for imaging, the G for genetic, okay? Okay, think of CBIG like a really specialized library, okay? And we're stocking it with truly rare and precious information or data about HSP. That's what we're doing. Like any library, all this information, the methodology used, and the results generated are all done via open science. It's all shareable. Open science is the concept of sharing and collaboration without patents, without silos. The concept of advancing human knowledge, all this information is completely usable. That's meaningful collaboration is a point of open science. Open science is not a new concept at all. It's only new to neuroscience. It's been around for years. You'll find it in all kinds of technology. It's even built into many advancements, like the technology that powers your cell phone, okay? All companies use the same science because it's advantageous to do it that way. In fact, NASA is a staunch defender of open science. It's a very efficient way to advance human capabilities. I'm nervous and I don't have any dexterity left anymore. I told you about that. Oh, yes, okay. So I was telling you that international collaboration is very complex. I understand that. I know that, but it's possible. It's complex, but it's possible. In fact, very close to the SPF home, Dr. Sabrina Paganoni in the Massachusetts General Hospital has often worked with Dr. Angela Genge at the Neuro. Boston has collaborated with Montreal. It's been done before. So we're going to do that too. Okay. Okay. Now I'm going to talk to you about the future of the cohort. That's the part where I make no promises. I don't have specifics. I don't have a crystal ball, but I know I'll always do whatever is best for HSPers. I truly, firmly believe that rare needs to stick together. HSP needs to stick to PLS. We need to stick together with other rare neurological conditions. The more doctors that we can attract attention, the more patients, just the more. Okay, so I started discussions with CPATH because CPATH is American. It's all new to this Canadian. But what I'm going to do, I know CPATH is very important to the SPF, so I'm going to keep Mr. Hart, we heard him yesterday, in the loop as much as possible. That's what I'm going to do. I tried reaching out to the Schill lab in Germany. Treat HSP has a very large cohort, and they've mentioned CAN HSP before. We're just going to see if I can connect those dots somehow. Okay, I was talking about how CBIG is a very specialized library. What we're going to do in the future, we're going to be contributing a lot of our images. The 32 consented HSP patients, we're going to be working on contributing our images, our MRIs. That feeds into Dr. Lapiana's white matter studies. I'm specifically mentioning Dr. Lapiana because she was able to get an SPF grant in 2022. By us contributing to her studies, what we're actually doing is giving the SPF fantastic return for investment. Their investment in 2022 still grows in 2024. You see what I mean? It's fantastic. It just grows human advancement through science. It's wonderful. So expansion in the future. I want to take it beyond 32 patients, and I will. I just don't have specifics. I don't have specifics of how or how much. We'll see. I think, is it Dr. Qiang was talking about organoids and cell lines? It's one of my, I'm going to do that in the future. Except I didn't, when I saw organoids the first time, they looked like spitballs to me. They look like mini spitballs. But they're little mini brains. They're a great way to study disease, right? But they still look like spitballs. Geneticists, Dr. Sally Farhan and Dr. Alison Dilliot that I mentioned before, they are studying, again, I don't have the details of this, but they are studying genes of uncertain significance to figure out what the significance is. We're able to give them information about that. So they're studying that. We'll go down that rabbit hole in the future. I want to expand the PLS cohort past the little, I'm sorry, I don't have details about the PLS cohort. I just want to expand it. So I want to be clear, I'm not talking about sharing patients. I'm talking about sharing information. Collaboration is key. We need to be able to give researchers, doctors, information. That's what this is all about. I know I can fortify your research. I know we can do that. I want you guys to be able to fortify my research. I'm very willing to jump through hoops to make that happen. How long did that take? 23? Okay. I have 7 minutes for questions. Do you want to do that?
One on your right, way over here. We're going to go start way over here.
Well, you know what? Here.
Hi. If I wanted to do a whole genome thing, analysis, and then give you all of my MRIs and the data that I've gotten so far from a lab, even though I'm not Canadian, could you use that?
I'm going to say yes.
Okay.
There's a different procedure for international consent, but the CBIG is, they're the experts in that. They've done that before.
Okay. Then you'd have 9, right?
Pardon me?
Then you'd have 9 people.
Yeah, it'd be great.
Cool. Okay, thank you.
All righty, do we have another one? They're always behind me. And now it's over there. And now I can't see anything. Okay, the room's gone dark. Do I see a hand anywhere? Please, you got to help me out. Okay, there's one up in the front row. Thank you. You got to be in my field of vision, guys. Here we go.
Thanks, Linda. That was a beautiful, personalized presentation.
Thank you.
My question for the whole genome sequencing,
Mm-hmm.
is that currently funded or subsidized partially by the Canadian government? Or do the patients pay for that themselves?
It's not funded by the Canadian government. It's funded by me, by my foundation.
Oh, okay. So your foundation provides and has funding to help potential candidates who volunteer for the whole genome sequencing?
We had, past tense, we had funding for the 32 people. On the neuro kicked in a lot too.
Okay, thank you.
All right, anybody, any more questions for Linda? Hey, I will say, if you want to participate and be with her, talk with her afterwards today. She's here. You can share that information a lot easier. I know her email was up there, but she's here today. Okay, so make these connections today. Get close to her. Come and talk with her. Ask these questions. Give her your email. She's very thorough. She's really good at email. I know. And so you guys can talk to her and make these connections and talk about this in a little greater detail. It's got my head spinning. Okay, and it's a really great thing, and it's nice to have this collaboration, across borders. This is pretty cool stuff. Okay, so this is just one more piece of that big puzzle. You are fantastic. Thank you very much for being with us today. Thank you. All righty, you want to come up and help her? Okay, guys, in a couple of minutes, we're going to move on to some Zoom presentations from a couple of doctors who can't be here with us. So give us a couple of seconds here to kind of glue all these pieces together, okay? But there is one caveat to their presentations. We are unable to ask questions of them. Okay, it's a little different format. They're zooming in, but we're not connected to ask questions up. A little different than we did with the presentation yesterday, because we had Tom here who was connected through a different device with his cohort, okay? So give us a couple seconds. We're gonna make sure we gotta get those guys in the right time zones, on the right Zoom call, at the right button, and make sure the internet is working, okay? Not for us, but for them. So we'll get on this. We're gonna have this in just a couple of seconds. Take just a quick little short break. Don't go too far. We'll see you in a second. Thanks, everybody.
All right. I'm assuming you can see and hear me. Hank, can you give me a thumb up? Super. Let me share my screen. Great. Okay. All right. So I hope you can hear me loud enough. It's great to be here, at least by Zoom. I'm really sad that I'm missing you there in person, and which would have been much more fun and much more productive to talk to you in person. I want to use the next 30 minutes to just sort of share some thoughts on genetics in general and in spastic paraplegia. So I hope you find this helpful. And let me start. So, obviously, the big goal here is that we get to therapies eventually. So you may ask yourself, so what is the role of genetics in the current times of spastic paraplegia? What role does it play? What role can it play in the future? Oops. Okay. And so here are sort of three main topics that are pretty obvious. One is, of course, diagnosis. This disease is an inherited disorder, so genes play a key role. And as you know, there have been many genes being found, and they have a major impact on families and patients who actually do know their diagnosis or don't know the diagnosis yet. So when we have a gene identified, this also means immediately there is a target, what we call a target, meaning there is a specific molecular mechanism or place where we can think very specifically, scientists, pharmacological companies can think about how to influence this target. So it's very important for any rational therapy development that we know the genes and the genes in each person. And the third point is certainly the biology. Much, if not most, of what we sort of know about the biology of HSP has started with identification of these disease genes. Because when we know the genes, then we know they must be important. And then other scientists can really go deep into this. And these are often studies that take a long time. But we learn about the biology, and knowing the biology, sort of the mechanics of the disease, gives further opportunities, of course, for therapy development. So, and at the bottom of the slide, sort of, we need to know the exact diagnosis, which is the gene, the HSP gene that you may have. But there are also other layers, what we call modifier genes. So there could be secondary genes in a person that determine if the onset is earlier or later, or if the disease, for instance, is more severe. So, and in all of this, ancestry can play a role. And of course, clinicians and also patients are very interested in this relationship between the actual mutations and the resulting phenotype. And this is a developing story. We're not even close to finishing this. And this hopefully will then lead especially to genetic therapy solutions. So the other thing to consider is we are here at the Spastic Paraplegia Foundation meeting, and we talk about spastic paraplegia or HSP. But that is not a disease in a silo. In fact, it is sort of embedded, if you will, in a spectrum of similar disorders that either look similar or they affect similar structures in the brain, or they are caused by the same genes sometimes. And they can sometimes, the same gene can cause HSP or one of these similar disorders. So here's sort of one sort of agreed on spectrum that is out there. And you can see that HSP falls right next to things like DHMN. What is this? This is distal hereditary motor neuropathy. or on the right side, there is ataxias, especially cerebellar ataxias. So each of these genes, they have their own spectrum of genes that they cause, but we are more and more aware of the overlap, and that HSP is not an island. It's connected with similar diseases. So benefit is, of course, we can learn from other disease fields. We can learn from them. We can learn from the biology that they identify. We can learn about the genetic targets and so on. So as most of you are well aware, HSP or SPG has turned out a disease which is not one disease, but many. And they are pretty much defined by the major underlying gene. So you can see here, this is not even up to date, that there are well over 50, the last number here is over 90 different SPG genes are known today. And some of these genes are quite related, so therapies might actually target a couple genes at once. But mostly right now, we probably would admit that each gene might need its own strategy for therapy, which is a daunting task, of course. Now, on top of this, unfortunately, many SPG patients don't know the genetic diagnosis yet. In fact, about 30 to 40%, it might even be a higher number of patients have not received a genetic diagnosis, even though they underwent the latest genetic testing available. So that is certainly something where we want to contribute more in the future. So genetics, is a lot in the media for many years now. Partially because it's so fascinating, of course, it's so impactful. Fortunately, there's a lot of attraction also in terms of technology development, and that has led to better and better technology to what we call sequence the human genome, a single human genome. Sequencing basically means measuring every single base and it's where it is and what it is. And that's the way how we find these mutations in HSP genes. And that's also the way how we find new HSP genes. And every year, there's a couple new HSP genes that are being discovered. So very important, the cost here, these are dollars, and this is what we call a logarithmic scale, the costs have declined immensely. So that today you can sequence what we call a regular genome for $500 or less. Here are some of the big company names. And you might be surprised, only a couple of years ago, only 2 years ago, the human genome was finally completely sequenced. There were always bits and pieces that were still not sequenced fully. And so in this paper here on the right side, they describe the final blueprint of the human genome. So why did it take so long? Because the technology available, which we call short read whole genome sequencing, is fantastic, but it had turned out it cannot sequence the entire genome. So we had to wait for another technology called long read whole genome sequencing, which is now only the last year to mature enough to be used more widely in research studies. And I expect this also to enter the clinical testing realm. So here's an example how discovery in genetics has been developing over the last 20, 30 years. And as you can see in these different colored bubbles here, it's not a linear situation necessarily. And each bubble represents a prevailing technological approach in that time. So in the 90s and early 2000s, there was this so-called linkage, golden age of linkage analysis that were mainly family studies. And then it actually slowed down, arguably it might have slowed down somewhat in the last few years. And there is a new technology now, the long-read genome sequencing, that is already showing much benefit. So another way to think about it is in this graph here on the left side, there is what we call the diagnostic gap, right? This is the clinical genome panels that are available that many of you have had. And if they're not successful, there is this gap. And then you may enroll in a research study, in a genetic research study, and if you're lucky, we find more and always find more, but it tends to be fairly small additions, any new gene. So the question really is in the genetics field, so where are the genes that fill this gap, right? And it has turned out that there are elements in the genome that we are not good at studying. And this has also to do with my first slide, or with an earlier slide where I showed you the whole genome wasn't even finished until 2 years ago. So we didn't have an opportunity even to look there. And here's another way to think about this. What clinical tests do and what most of we do in research today is, frankly speaking, only looking at about 2% of the genome. When we do whole genome sequencing, like some of you have undergone, of course, we look at the remaining 98% as well. But it has turned out we just, maybe the most important pieces of these 98% we weren't good at looking at. So again, coming back to the long read sequencing, and especially what we call the repetitive regions, they make up about half of the entire genomes, and only now we start beginning to see more of what is going on there. So, so what do I mean by all of this, and what does long read sequencing mean anyway? So the human genome, as you know, consists of these 4 bases that are arranged in a specific pattern. But it turns out there are regions in the genome that are, we call, repetitive. For instance, there could be a CAG. Imagine 3 letters, CAG, and then CAG. So there's a long stretch of CAGs. And when we measure this, the machines we use, they get confused, right? And if this is a very long CAG stretch, the read maps we what we call align to the genome, these short reads, we don't quite know where to align them correctly on these repetitive regions. And that has been, simply speaking, the problem. So certain genes like this FGF14 gene, you could simply not read out with current technology. There are some with certain tricks we can apply. There are certain things we can read, these repeat elements, like in Huntington disease, but not in all of them. And straightforward, really, these long read sequence technologies, they don't produce 150 letter or base pair long reads. They can produce reads that are over 10,000 base pairs. And suddenly you can really align them quite well and with certainty across these repetitive regions. So now we can read all these genes. So that's sort of breaking it down somewhat. And there's some more detail here on what kind of technologies are there. I'm not probably going to skip this. It's very technical, fascinating, however. And what we also observe is in the last 5 years, even though the interest in the 98% in the non-coding genome is high, has always been high, but mainly through some very sophisticated software approaches and improvements, there have been some really nice discoveries. For instance, on the left side is a gene called RFC1, which is a non-coding change. In the middle is a gene I think we talked about last year, possibly. It's a gene called CMT for CMT, and on the right side is a gene for ataxia. So patients with these genes, they can have spasticity, but it's not really the main idea here. I'm showing you this because all 3 of them are either non-coding, or the one in the middle is something where there is a gene that's almost identical. It's like a mirror gene that confuses us. And these gene pseudogenes, we will call them also, they are another source of confusion in genetics that we probably gonna solve much more in the coming years. So again, the main point really here is these three examples, they turned out to be not another rare cause of these disorders, these rare diseases. They turned out to explain a lot of patients, like thousands of patients worldwide. And so my point is, there is good hope that the 30 or 40% of patients who don't have a diagnosis today, it might sometimes go faster, so that we might reach much better diagnostic rates faster when we study these non-coding regions. So as you can imagine, as geneticists, we try to be at the cutting edge. So we work pretty heavily in this area right now. And we've been involved in a pilot project with the National Institutes of Health, where we study a few thousand of these long-read genomes and see what's in there in general. And we see, let's say, signatures that we haven't been able to see before. And it's a working group, not just the Miami group. And everybody's really quite excited and surprised what else is out there in the human genome. It's not like we know the human genomes that well yet, it seems. My student, Sarah Fassal, she just graduated last year, but she developed an AI, an artificial intelligence-driven program to go faster through these new findings. So these things also happen, that we're trying to use these machine learning approaches. And then here's another, hopefully not too confusing aspect of these sort of new genetics. Some of it is something geneticists have known for a long time, but now we can study it in much greater detail. So this is also not spastic paraplegia, it's another gene recently identified. And we were able to sequence the parents and the affected child. And what we see is for this particular gene here, which is a non-coding change, is one of these repeat changes. When you inherit this chromosome with let's say the mutation, what happens is this repeat always gets smaller. It always shrinks. We don't quite know why, but that is the case. When you get it from your father, it shrinks. When you inherit it from your mother, it always grows, it expands. And when it grows, it's not good, because when it grows over a certain size, these repeats, then they cause disease. So, but when you imagine this, that you study a genetic disease where every generation changes, and so, depending on where you get it, from your dad or from your mom, the pedigrees and the family history becomes quite confusing for everybody involved. And that makes it, of course, complicated, especially if you don't know what you're looking for. So this is the kind of genetics we have to really think harder about going forward. And this is another detail out of these pilot studies on these long-read genome collections. And these are all from, or mostly from controls at this point, actually. They're probably less than a few thousand long-read genomes produced from patients worldwide so far, at least at high quality. So this is all fairly new. So when we look at these repeat loci, which we were not really able to sequence so well up until now, everybody who sees these data is amazed, because it's not like CAG, CAG, CAG. When you look closer and with great detail, you see these motifs are interrupted. So each color is sort of a different motif, and you see down here the motifs. And it can get quite complicated. They are stacked into each other. So each line here, each row, is a different chromosome. So there are many patients stacked up here, or many individuals, and you can see everyone has a somewhat different type of of repeat at this place. It's conceivable that one of these actually is linked to disease, but finding these, you can imagine, is even harder maybe than what we have done so far in genetics. But now at least we are able to see that, and we can start really thinking about it. Another very important topic is, of course, you may get back a genetic report, and with 90 HSP genes currently known, so the test may have tested 90 different genes at once. Chances are that there are changes, I'm not saying mutations, just changes, that where the clinical laboratory cannot be sure, is this a benign variant that is there by chance, or is this actually the one pathogenic, the one mutation we really look for here? And when the clinical lab is not sure what to do with that, they call it a variant of uncertain significance, or VOS, or VUS. And this has become quite a problem. The correct classification of variants in known HSP genes can be quite tricky. And there are many, many patients out there who don't quite know what their gene is. They have a VUS in their report, and their doctor doesn't know either. So how can we better classify, basically sort into the benign bin or in the pathogenic bin these many variants? That's the task, and that is turned out not to be an easy task. So this is another thing we work on, and the field is working on and thinking about a lot. There are traditional ways, like this is the schema that clinical labs use. It's quite detailed and can be confusing, how to classify variants. There are newer approaches or alternative approaches that are being tried out. They're not really fully accepted yet, I would say. And many have to do with machine learning or artificial intelligence, a bit like ChatGPT, frankly. And the judges are still out there if that will contribute to a better diagnosis, a better classification. I think there will be some sort of convergence of all these different approaches in the future. Here in Miami, we have published one of such AI or neural network approaches to classify variation, and we were surprised how well it actually can work. So I don't want to go into detail. This is a published work, and we use it on a regular basis. In fact, it helps us to identify novel genes. There are about a handful of genes that where the computer basically told us, you should look at this gene. And then we look at this gene, and in a handful of cases, it turned out to be, yes, that was a good idea to look at this gene. This is, in fact, a novel gene. So another thing you can do when you start trusting your machine learning more, you can actually go out to a gene like spastin or SPG4, and you can say every conceivable missense change in spastin, predict right now, is this pathogenic or is it not pathogenic? Is it benign? And we did this for spastin, and this is what you get. So this is the gene here, and each dot is a different missense change that is theoretically possible. And all the red dots are the ones that are known mutations, missense mutations in spastin. And where these known changes, when they were considered disease-causing, they end up high here and low here and also low here. And you can see that the red dots, they correlate pretty well with the gray dots. So if we start trusting, oh, this works pretty well for the known spastin genes, then maybe the other gray ones are not so bad either. So that's really where I'm going here. And finally, I'm sure Darius has told you already about our new consortium, the SP-CERN. And the basic idea really is to bring people together, experts, make them work together to create common resources like clinical databases, DNA biobank samples that are shared in the group, but also genetic data. And so for genetic data, bringing genetic data together, that makes it easier and more successful to combine them and to find important things in these data. So we're going to use the Genesis database, which in Miami we have used for over 10 years now with great success. And it already contains a lot of data where the SP-CERN can sort of add on to that. In fact, right now there are over 2,000 genome data sets in this Genesis database. And they're not in some hidden hard drive. They actually de-identified in a cloud, and they highly protected, but we can share them easily, and we do share them easily among scientists in SP-CERN, but also beyond with colleagues in other countries who are researchers. So only researchers have access to these data. And this is one example how this has worked already in the past. This UBAP1 gene is a HSP gene, and it was found through really collaboration with different countries because they found each other very much so on this Genesis database. So it's very powerful. And finally, a word on other alternative approaches. Once we have a lot of genetic data, we can also start using statistics. We know from other diseases who have been successful in creating large genetic data sets that you can use statistics to find signals for disease genes. And this is sort of a pilot study we have performed a couple of years ago, which also included HSP. And you clearly start seeing sort of differences based on statistical measures. It's still a jump from there going to individual genes, but it is a very worthwhile approach. I think it's just hard in rare disease because you need to really build up a large genetic database. And another topic that every year comes up and we are very aware is the fact that, for instance, in spastin, which is a more common HSP gene, even patients have realized that there are large differences from patient to patient. And doctors have always asked, why is that? What explains this difference? Even in the same family, which carries the same mutation, you see differences, later onset, earlier onset, and so on. And the idea is there could, of course, absolutely be environmental modifiers. And environment really means everything from infectious diseases to food to climate, you name it. But there could also be secondary genetic reasons, so-called modifier genes. And the group in Paris has, and collaborators, they have performed the first SPG for genetic modifier study and published this. And in fact, they did find a signal here also, again, with these statistical methods. They found a signal in this gene called SARS2 that may contain genetic elements that modify how SPG4 is expressed in people. But I'm convinced this is not the only gene. This may not even be a main gene, but it's a great first find and needs to be followed up on. So we are in the SP-CERN, there's definitely interest to do that, and there will be resources to do that. A group in Germany, Rebecca Schüler with Treat HSP, she's working on this and she's collaborating with us in Miami on that topic as well. So with this, I want to stop and not sure how we're going to do it, Norma. I would be very happy to answer any questions if there are any.
Hey, Harper, can you type in the chat box for him to put up his email for people to send questions to him? Can you do that for me?
I can't hear.
You can open your mic if you want to talk to him.
Works.
Harper, you can probably speak to
I
him
will send you
directly.
an email. Yes.
Okay, real quick, can you capture, before you flip over, Harper, can you guys see that down there, his email? Let me read that out loud for you guys. All right. SZUCHNER at med.miami.edu. Okay, guys, if you have any questions for Dr. Zuchner, you can email him at that address. Okay? So he is going to unzoom, and we are going to move on to Dr. Calame, who's next in the bullpen. So everybody hang on while we make the switch, and he knows that you guys are all clapping loudly for him because he did such a great presentation. So thanks, Dr. Zuchner. Okay, coming up, and there he is, Dr. Calame. Here we go.
Can you all hear me? Should I get started? That a yes? Okay. All right. All right. Well, hi, everyone. It's really a pleasure to be able to speak to you all today. My name is Daniel Calame. I'm a pediatric neurologist at Baylor College of Medicine, Texas Children's Hospital. And today I'll be talking about my work on the genetic mechanisms underlying undiagnosed pediatric HSP. I'm sorry I couldn't be there with you in person today. I'd love to be there in Tampa. But I'm coming to you today from Houston, Texas. I work at Baylor College of Medicine, and I'm an attending physician at Texas Children's Hospital, which you can see here. TCH is the largest children's hospital in the United States. It sits within the Texas Medical Center, the largest medical center in the world. TCH is consistently one of the top children's hospitals in the United States, and our neurology group in particular is consistently one of the top 3, top 4 programs. I focus on neurogenetics. In particular, I focus on movement disorders and hereditary spastic paraplegia. And I'm very pleased to be a part of the SP-CERN natural history study. I work with Jim Lupsky. I suspect many of you in the room are familiar with Jim. Jim is kind of a big figure in human genetics. He really created the field of genomic disorders with his discovery of the PMP22 duplication as the cause of Charcot-Marie-Tooth type 1A, the most common type of Charcot-Marie-Tooth or hereditary neuropathy. And that really really changed everything, and of course has had a significant relevance to HSP, since deletions and duplications of SPAST are one of the most common causes of HSP. And the the SPF previously supported Dr. Lupsky some years ago, and they published great work with Phil Boone, showing that there's some unique features of the genomic architecture around SPAST that predispose it to have frequent deletions and duplications, these repetitive elements in the genome called ALUs that help mediate recombination. And so Jim thinks very, very fondly of the SPF. And so our focus is really on undiagnosed patients. And in HSPs, like Dr. Zuchner mentioned earlier, we see a similar story to many other genetic conditions. There are many HSP genes. I'm aware of at least 83. Dr. Zuchner mentioned 90. But despite this large number, the reality is that with our best clinical genetic testing, we still don't get a diagnosis in many patients, if not most. The figure on the right comes from a group in France. They applied a gene panel approach to over 800 patients of HSP, and they were only able to get a diagnosis there in about 30%. When you look at the breakdown of the genes that are involved, there's just a couple of genes that really make up the lion's share of diagnoses. Of course, SPAS being the biggest one for dominant HSP, and then for recessive SPG7. And the reason for that is for SPAS, of course, again, there's features of the genomic architecture that predispose to deletions and duplications of it. For SPG7, there's just simply certain pathogenic variants that are more common in European populations that make it to be one of the most common recessive ones. But when you get outside of those couple of genes, you have many ultra rare causes of hereditary spastic paraplegia, and it's clear that there are many more that remain to be identified. So as we think about how to get answers for undiagnosed patients, it really comes down to two areas. I think Dr. Zuchner talked already a lot about this first area, the idea that we're missing certain genetic variants, things deep in the introns, in the non-coding parts of the genome, the structural variants, the repeat expansions. And we believe that new technologies like whole genome sequencing, especially long-read whole genome sequencing, RNA sequencing, and refinement of our ability to analyze the data, it's going to make some significant breakthroughs in this area. But it turns out that in many cases, the answer is actually already there. It has already been identified in the exome or genome, clinical exome or genome that was already done, and it just simply wasn't recognized or wasn't understood at the time. And it because of the three kind of phenomenon that I list here, the HSP mimics, we call phenotypic expansions, as well as novel disease genes. And to get to the bottom of these, we really just need to get into the data, to reanalyze it with a more critical eye and with additional analytical tools, data sharing that Dr. Zuchner mentioned, and then a lot of experimental biology. And so I'm going to give some examples that have come out of our work in recent years of these three things. This first one is an HSP mimic. This was a story that began when I was a resident in the Bluebird Circle Clinic at TCH. I saw a young woman, 17 years old, who had a history of developmental delay and progressive spastic paraplegia. She first walked around 2 years of age with a walker. Her first words came around 4 years of age. And when I saw her at 17 years old, she had a diagnosis of mild intellectual disability, but her spasticity and her gait problems had really progressed, and she could hardly stand or walk without considerable assistance. And so she came to me already with the clinical diagnosis of a complex, complicated form of HSP. This figure shows the typical pipeline where we go from a DNA sample to sequencing to the computational pipeline by which that sequencing data gets kind of put back together again to yield the sequence of the individual's DNA. And then from that, we pull out the variants that are then analyzed and interpreted. And ultimately, whatever is thought to be significant of that goes into the medical report that goes to the patient and their doctors. But in cases like this patient, she had clinical whole exome sequencing actually very early on in the course of when exome sequencing was available, and her report was negative. And so, at this point, we kind of have a few options available to us. We can go back and sequence them again using a more advanced technology, like perhaps now long read sequencing, or we can just get the existing data and reprocess it in the lab to take a closer look at it and again apply new analytical tools. And what happened in this case was our reanalysis led us to focus in on this missense variant in this gene ATP1A3. This particular missense variant had never been seen before in any other patients in the medical scientific literature. It had not been identified in any healthy individuals and population databases. it was predicted damaging. It was a very conserved nucleotide and amino acid. And it fell in a part of this gene that was kind of a hotspot for other pathogenic variants. So it seemed to be a good fit from a genetic perspective. But the problem was that it didn't seem to be a clinical fit, that it didn't fit the diseases that were currently linked to this gene at that time. And that's why it didn't make it onto the clinical report. For the neurologists in the room, especially the movement disorder neurologists, this gene is very well known to us. It's a common cause of very striking paroxysmal movement disorder syndromes. On the left, you see an example of the most common one, alternating hemiplegia of childhood. This is a patient that I see in my clinic, and I share these videos with the permission of the family. What happens in this condition is in the first year of life, children will have hemiplegia. It'll come on, weakness on one side of the body. It looks like a stroke. That's kind of what you can see in the video on the far left. If you look real carefully, she's having a hard time using the left side of her body. And that will last for minutes to hours or even days. And then it goes away, or it moves to the other side of the body. It's a very striking condition. There also are many other abnormal movements. I'm also highlighting abnormal movements of the eyes that come and go. But this is what we see with ATP1A3, these very striking episodic movement disorder phenotypes. ATP1A3 encodes a pump, a sodium-potassium pump that's found within our nervous system. Maybe you see a figure of it on the right side of the slide. And this is important for maintaining electrochemical gradients in the brain. And so, again, the issue here was that it just didn't seem like a good match with the patient's condition, which looked more like a slowly progressive hereditary spastic paraplegia. And so the way we approached this was to then analyze data from clinical diagnostic lab databases, as well as to reach out to research collaborators from around the world. And it turns out that this particular variant is a fairly, I'd say somewhat common recurrent variant that causes this sporadic complex HSP-like disease. We found it in a total of 9 families. In several cases, we were able to show that it was a de novo mutation, a new mutation in the patient. And when we look at the diseases in these patients, it really looks like complex HSP. They all have developmental problems. They all have spasticity. Most of them have spastic paraplegia. And then in the patients who have spastic paraplegia, it's pretty evenly split between a progressive form and a more static form. And really, we don't see very much in the way of these episodic paroxysmal movement disorders like in the other ATP1A3 related conditions. We worked with another pediatric neurologist and physician scientist, Sho Yano, at the University of Chicago. Sho is very interested in this gene and is a great electrophysiologist. He was able to show that this variant completely kills the pump activity of the gene, similar to other pathogenic variants in ATP1A3. But what was really fascinating was he also found that the variant does something that hasn't been seen before. It results in an inward leak of cations, of ions, across the plasma membrane. Again, this has not been seen before in any other ATP1A3 variants. And in theory, this should counteract the activity of the other normal copy of the gene, and it may explain how this variant causes disease. So to summarize the HSP mimics, I think it's important to recognize that other genetic disorders can be clinically almost indistinguishable from pediatric simple and complex HSP. And because of this, there's really a big need to use untargeted genomic sequencing like exome and genome sequencing for clinical diagnostics in pediatric HSPs. I'll next move on to an example of a phenotypic expansion. This one focuses in on a gene called FLVCR1. FLVCR1 is a transporter. It's found in most of the cells in our body, most tissues, and it acts to transport 2 metabolites, choline and ethanolamine, into our cells. Choline and ethanolamine are critical for many processes. In particular, they are the substrate for what's called the Kennedy pathway, a pathway that generates phosphatidylcholine and phosphatidylethanolamine to critical components of cell membranes. Choline is also the substrate precursor for acetylcholine, a neurotransmitter. And choline also plays a broad role in many other metabolic processes as well. So very, very important gene and protein. We've known for some time that this is essential for life, at least in the mouse. When you knock this gene out in mice, you don't find any mice that are knockouts that don't have the gene at all. So if you look earlier during embryonic development, you can find some embryos that lack the gene. But as you can see here, they're very small. They have severe skeletal malformations, and they have very severe anemia. You can see that embryo is very pale compared to the normal one on the other side. In contrast, in humans, this gene was linked 2 years later to something very, very different, to a childhood or adult onset syndrome, where patients develop ataxia, progressive vision loss, blindness, and sensory neuropathies. It's a recessive disease, as shown in this pedigree on the left. And since this initial paper back in 2010, there's been at least in their 30 families identified, probably 40 or 50 patients with this condition, but very different from what we see in the mouse. And so we became interested in this because in the course of our work, both on HSP as well as in other severe neurological disorders, we kept finding patients who had really impressive variants in this gene, but with very different diseases. And so what I'm illustrating here is all the families where you see the red boxes or circles. These are families with very, very severe neurological disorders. I'll show you what I mean by that in a little bit, but you can see, unfortunately, that many of the children had passed away in the first years of life. We also found some other families that had the more typical disease, the ataxia and sensory neuropathy, but even in the kind of the milder side of the spectrum, we were finding some diseases that hadn't been associated with this gene yet. This patient in particular has HSP, has simple HSP, began in childhood, slowly progressive, none of the other features associated with this gene so far. And these two families have more of a classic kind of Charcot-Marie-Tooth motor predominant distal motor neuropathy. And so I'll now explain to you what I meant by the severe end of the spectrum. So these are some pictures of affected children as well as their brain MRIs. These children are very developmentally impaired. They have, almost all have very severe spasticity, very severe seizures. And when you look at their brain MRIs, you can see they all have evidence of brain atrophy. There's quite a bit of variability in the appearance of the brain MRIs, but on the most severe end, like you see on the bottom right, it can be very, very severe. Like this child just had a very thin rim of brain matter, kind of like what we call hydranencephaly in neurology. We also found the most severe disease, what we believe is the most severe disease from this gene, in two families. There were two families where there were children who were stillborn, who had significant skeletal malformations, anemia, and severe brain malformations as well. And in these families, they had mutations that caused them to have a complete lack of this gene, so analogous to what we see in the knockout mice. And we studied the impact of the missense variants that we had found, the mutations on the protein function and choline and ethanolamine transport. You're seeing on the far left that actually these missense variants don't seem to impact the stability of the protein or its localization into the plasma membrane, but nearly all of them significantly reduce choline and ethanolamine transport. And some of them, I don't show the data here, but they also impact the splicing of the gene and therefore either result in a truncated protein or perhaps no protein at all. And so kind of in summary, we think that the severity of disease from this gene is a consequence of the severity of the mutations that the patient has, with more severe ones causing more severe disease and the milder phenotypes from the milder missense variants. And so I think the summary here is that these phenotypic expansions, where a gene that was linked to one disease is then found to cause other diseases, this is really it really seems to be the rule in genetics rather than the exception. Probably most genes are going to cause more than one disease, more than one inheritance model, both dominant and recessive. And so this is something we need to be on the lookout for in HSP, because I think HSP may be, in some cases, a rare manifestation of other genetic disorders like FLVCR1. And then finally, I'll wrap up with a gene discovery, and this is still a ongoing story. So I'm happy to share our preliminary data. This is about a kind of continuing on a similar theme of transporter disorders. This is a gene called SLC7A1. This is another transporter. It's found all throughout our body, especially in the endothelial cells that line our blood vessels. This one, though, transports a few amino acids, the cationic amino acids like arginine, lysine, and ornithine. It's a high affinity transporter. In this figure, I focus on arginine because there's a lot of important biology involving arginine that probably ties into this disease. Arginine is an amino acid, so of course it's also important in just building proteins, but it plays a particularly important role. It feeds into the urea cycle, which some defects in the urea cycle are known to cause spastic paraplegias. It also is a substrate for nitric oxide synthesis, nitric oxide being an important vasodilator, as well as the synthesis of creatine and polyamines. This gene, the mouse story is very similar to FLVCR1. When you knock this one out, mice die. They do survive till birth, but they die on the first day of life. And strikingly, they also have very, very severe anemia. This paper was published well over 20 years ago, back in '97. But really, since then, there hasn't been much work on the role of this gene in the nervous system. And so, in the course of our work of reanalyzing genomic data from undiagnosed patients of HSP, as well as doing genome and exome sequencing in new patients, we came across this, and we think this is a really strong candidate gene for recessive HSP. We've identified a total of 7 families so far. There's 11 affected individuals. Mostly they have homozygous missense variants. There is one compound heterozygote with a missense variant and a loss of function variant in trans. And the phenotypic spectrum, like in many of the HSP genes, is broad. It ranges from, in a couple of families, from simple HSP to another couple, more of a complex picture. And then there is one family, one patient, the one shown here on the bottom right, that has a very severe spastic neurodevelopmental disorder. I have a few pictures here just to show of some of the affected individuals. This child here is, cognitively, developmentally is pretty much where he should be, but just has progressive spastic paraplegia. His brother is similarly affected. They both have, on their brain imaging, they do have very prominent ventricles and some degree of Vermean atrophy. And then I mentioned this more severely affected child. She has microcephaly and pretty severe developmental problems and more severe brain appearance. When you look at these missense variants, we haven't seen any recurrent ones yet. We think this is a very rare cause of recessive HSP, But we do see, curiously, that there are recurrently affected amino acids, valine 291 and arginine 588 in particular. The missense variants all fall within transmembrane domains. They're all very well conserved across different organisms, as well as different genes within this gene family, the SLC7 family. They're all predicted to be very, very damaging. When you look at their distribution within the actual structure of the protein, this is from AlphaFold, and this also shows alpha missense predictions of how damaging each changes in each amino acid are, where the blue or white, blue is benign and red is predicted pathogenic. You see again that all the variants fall in areas that are predicted very, very damaging, very likely to be pathogenic, and they tend to be near the inner pore of this channel, which suggests that they're probably impacting the flow of arginine and other amino acids through the transporter. And so right now we've generated both constructs for all these missense variants, and we've also made knock-in cell lines, and we're doing arginine transport assays and other studies to understand the biological consequences of these variants. So in conclusion, I hope I showed that HSP mimics and phenotypic expansions are a real issue in undiagnosed HSP patients, and because of this, we need to be thinking about genome-wide sequencing over single gene and gene panels, especially in pediatric HSP. We and many other groups have shown that the reanalysis of existing exome and genome sequencing data can significantly boost the diagnostic yield and facilitate discoveries. And like Dr. Zuchner mentioned earlier, gene discovery in HSP remains brisk, one to three genes, I would say, just about every year. And in the process, we're discovering novel disease mechanisms and hopefully opportunities for new therapeutics. And so I want to conclude by acknowledging, of course, my mentor, Jim Lupsky, and all members of the lab. We have great leadership at Baylor Child Neurology, Gary Clark and Huda Zogby. We're a part of a large NIH rare disease consortium called the Gregor Program. If you have undiagnosed patients, or if you are an undiagnosed patient, feel free to reach out to us. We have ways to help get you research genomes, long read, short read, transcriptomics, et cetera. We have many collaborators I have to acknowledge, and especially the patients and families that participated in the studies. And then finally, I really want to thank the Spastic Paraplegia Foundation. Jim was awarded a grant from you all a couple years ago, and it was instrumental in this work and getting me involved in HSP. But thank you very much. And I will drop my email in the chat. Please feel free to reach out to me.
All righty, everybody.
All right. Thank you all.
Thank
Bye-bye.
you. Dr. Calame, great job. I know that's going to go up there pretty quick, but Dr. Calame's email is in your program under his bio, okay? So you can look in that if you guys want to reach out to him specifically, okay? And we do have Dr. Zuchner's email, but I have it in the back of the room. So anybody need that, come back and see us, and we can give you that, because that went by pretty quickly. Hey, I want to do a quick couple of things. Remember, everybody, 5:15, the 5K photo outside with Jim. Also, the PLS community, when they do their breakout session at 4 o'clock, inside of that time frame, they're also going to have their group photo done during that breakout session. Just wanted to remind you that. Hey, and something really cute and really great that I wanted to tell everybody about while we're making a transition to our next thing is it's Andrew Hendrickson's birthday today. Andrew, where are you? Stand up. There you go. Andrew has been doing a great job helping us throughout the conference, and he's been taking 1 million pictures, which will show up somewhere on our Facebook page and Instagram pages and our website, and next year's picture PowerPoint of our historical review of conferences. You'll see yourself in some of those in that too. So we're switching over right now to get ready for our next presentation, which is going to be, looking at the wrong day, Dr. Schierbaum, and we're working on that, getting her laptop hooked up right now, and then she's going to work her way this way, and then we're going to get her going on her program. Okay, and then after that, we're going to have mini stretching with Zachary again. Okay, so you guys are getting a lot of workout. And he, by the way, is also having his own breakout at 4 o'clock, between 4 and 5. So if you really want to go and get a nice workout with him, we're looking forward to that a little bit later. So once we get everything set here, we're switching between laptops and everything. Remember also, if you want some raffle tickets and t-shirts and bracelets, you have till 5:15. So make sure you stop by the registration table and get all that good stuff, okay? Because we have it and it's here and available for you. And then later this evening, during our dinner, we're gonna have our raffle and our auctions and other kind of fun stuff. So that's gonna get everybody really excited, and it's gonna be a lot of fun. So it looks right now that I see we have the presentation up, and here we go. We're ready for Dr. Schierbaum. Here she comes.
Hello everybody, my name is Luca Schierbaum. I'm a research fellow and pediatrician working with Dr. Darius at Boston Children's Hospital, and I'm here today to talk to you about one of our research programs, the HSP Sequencing Initiative. And with this program, we aim to close the diagnostic gap in childhood onset HSP. Some of the things I'm going to be talking about have already been mentioned by Dr. Zuchner and by Dr. Calame, but I'm hoping to show you some new perspectives. Next slide, please. So to give you an overview over my talk today, I will first talk about the evolving role of genetics in HSP. Then briefly talk about HSP as a differential diagnosis in cerebral palsy. Then I will present you our HSP sequencing initiative, introduce you to our cohort, and I brought 2 of the cases we worked on this year. And then I will talk briefly about some future directions. Next slide. And next slide again. There is. Perfect. So with this slide, I would like to show you how the research on hereditary spastic paraplegia and on genetics has been evolving in parallel. So we see that the timeline starts on the left in 1880 and 1888 with the work from Stümpel and Lorraine, who first described and recognized the hereditary causes in spastic paraplegia. Stümpel described 2 brothers with hereditary spastic paraplegia, and Lorraine described the first 29 families with HSP. Let me continue with the description of the first HSP gene, SPG1. In the genomic research, we have the Human Genome Project in 2003, which sequenced the first human genome, almost completely, 92%, as Stefan noticed in his talk. In 2009, we established whole exome sequencing. And then in 2022, SPG91 was established, the SPARTAN1 gene to cause HSP. And so we see that over the course of 30 years, 90 disease genes for HSP have been discovered. And at the same year, the Telomere-to-Telomere Consortium managed to completely sequence the first human genome. And we see that the research has been accelerating towards the last few years. Pedro, next slide, please. Thank you. So with these 2 graphs, I want to show you how this acceleration in genomic research also leads us to new discoveries in HSP. So we see on the left side the cost of genome sequencing from Illumina, one of the big laboratories that researchers work with. So we see that 10 years ago, the cost for one genome was over $5,000, and right now it's approximately $200 per genome. And mirroring that on the right side, we see the PubMed research on HSP genetics. So 30 years ago, or 50 years ago, we had almost only like 5 papers that were published per year, and now we have over 300 publications per year on HSP genetics, which is amazing, I think. Next slide. To talk about hereditary spastic paraplegia as a differential diagnosis of CP, I brought you this meta-analysis from González Mantilla et al. They looked at 13 different studies that looked at the diagnostic yield of exome and genome sequencing in patients with cerebral palsy, and they found that indeed 30% of the patients that were diagnosed with cerebral palsy had a genetic diagnosis. But we also see that there's a broad range of the diagnostic yield between 10 and 40 to 60%. And it is important to notice that the diagnostic yield in a subcohort analysis is higher in pediatric patients with CP. And we also have to keep in mind that the most common genes where disease-causing variants were identified were the SPAS gene and ATL1. Next slide, please. So if we talk about the role of genomic or genetics and diagnosis, we often start with a really targeted approach, single gene sequencing or gene panel, and what we are often implementing now as the gold standard is the clinical exome sequencing. But the diagnostic yield of all these approaches right now is between 60 and 70%, meaning that 30 to 40% of the patients with clinically HSP don't have a diagnosis. Next slide. And this work of Cao et al. Shows us in more detail what this means. So in the patients that have a positive whole exome sequencing, most of the mutations are attributed to SPG4. If we look at CNVs, also most of the mutations are found in the SPAS gene, but 40% of the patient population that were looked at in this paper didn't have a genetic diagnosis. And the question is, what are we missing? There are several options. So one option is that we don't identify the mutations in genes that are already known to cause HSP. And the second option is that there are more genes out there to be discovered that cause HSP, or possibly as well a combination of those two things. Next slide. And to address this issue, to address the 40% of unsolved cases, we started the HSP Genomic Sequencing Initiative. In this initiative, we include all patients that are younger than 30 years old that have clinically suspected HSP but no genetic diagnosis. And how do we recruit out patients? So one contributor is the Movement Disorders Clinic at Boston Children's that is led by Dr. Darius. Then we have our natural history study. We have some patients and clinicians that contact us directly. And one big support for this are patient foundations that are really essential in connecting researchers and patients and their families. Next slide. Thank you. So what's the aim of this initiative? First, we want to establish the diagnostic yield in exome-level reanalysis in our well-characterized cohort with children and young adults with a clinical diagnosis of HSP and that don't have a genetic diagnosis. And the second aim, next slide, is to establish the diagnostic yield of short-read and or long-read whole genome sequencing in said cohort. Next slide. So this is an overview over the patients that we have enrolled so far. We have 95 patients enrolled. 68 of those are trios. 8 of those are trio plus, which means that we not only have parental DNA available, but also another family member enrolled. 10 singletons, 1 singleton plus, 5 duos, 2 duo plus. And important to note that 80% of the patients we have enrolled, we have samples from both parents available, which is really important to increase the quality of our analysis. Next slide. Thank you. And what I briefly mentioned before, we are implementing short-read whole genome sequencing. In short-read whole genome sequencing, the first step is a library preparation where the DNA is fragmented, adapter sequences are added, then we have a cluster generation by in situ amplification, and then we have in parallel sequencing by synthesis, which means that while the DNA is synthesized, we also get the sequencing data. And there are several strengths to this approach. So one is that it's an established approach. We have a low error rate from 0.1 to 0.5%. We have relatively low costs, and we can detect CNVs and non-coding variants. As a disadvantage, we know that we still have a diagnostic gap in detecting repeat expansions and complex structural variants. Next slide. And next slide. Right now we have DNA available for 51 trios, 7 trio plus, so we see that this mirrors the enrollment of the families in our cohort. Next slide. 70% of our patients have had previous genetic testing that was non-diagnostic. 13 of our patients did not have genetic testing, and from 10 patients, these informations are still pending. Next slide. So this is an overview over our analysis workflow. It's a little bit busy, but I'm going to walk you through this. So on the first up left corner, we have the data and sample collection. This means that we get the blood drawn from patients and parents. Then we walk through, take a short questionnaire to get a better phenotypical understanding of the symptoms of the patient. The phenotypical data will be stored on a REDCap project. The blood will be shipped to GeneDx, our collaboration partner. They will do the DNA extraction, and they will do the sequencing for us. Then the data will be stored on a Google Shared Drive, and then all the data will be moved to our research computing department. They do the quality control, they do the alignment and the variant calling with the Dragon pipeline. And those data, to analyze those, we have different tools. One tool is imaging that we use for a specific type of variants, for small nucleotide variants called SNVs, and for short indels. And then we have Genuity that we often work with. It's a tool for different types of variants, for structural variants and larger CNVs, and we also can do a cohort-level analysis. Then on the right side, you see the Genesis project. It's one of our collaborators, collaborations with Steven Zuchner's group in Miami. They have built a platform for variant calling and analysis, and also for data sharing, as he previously mentioned. And we use all these tools in combination to increase the diagnostic yield as a complementary approach. Next slide. So how do we approach if we see a variant that might be interesting? We start with strict filter criteria you can see in red, and then we slowly move forward to more permissive criteria you see in green. And we assess the variant starting with all red filter criteria for different qualities, for zygosity, allele frequency, combined check, and then also for associated disease. And in the end of this process, we tag the variant. If we decide this is not a variant of interest, we loosen our filter criteria step by step to more and more permissive. If at the end of all permissive filter criteria we still don't have any variant of interest, we will reanalyze this data set within the next year. If there is a variant of interest, we will follow up, which is mostly done through research collaborations. Next slide. And one critical part of this process is our monthly discussion as an interdisciplinary team with neuropediatric clinicians, genetic counselors, and clinical geneticists, and we discussed the genetic and phenotypic finding, always with the question, is this finding our diagnosis? And if we think and come to the conclusion, yes, this finding, research finding, might be the explanation for the patient's symptoms, we ask for clinical confirmation, called clear confirmation. If we think, yes, it's a possibility, but we're not quite sure yet, we determine which steps have to be done to confirm the diagnosis. This can mean submission to GeneMatcher, which allows us to collaborate with other researchers. We can do additional testing like muscle or skin biopsies. We ask for an ophthalmology consult, or we do additional imaging. And if we don't think this is a good genetic explanation, we will reanalyze the case in a year. Next slide. So this brings us to the first case that I brought for you today. It's about a 5-year-old girl that had a normal birth and prenatal history, but the parents noticed that she had some neurodevelopmental delay in motor, speech, and language. She had progressive lower limb spasticity with hyperreflexia, clonus, positive Babinski. She had toe walking and a spastic gait with a GMFCS of 3. She had some clumsiness, poor fine motor coordination, and also weak voice and dysarthria. Some workup had already been done. We had a normal brain and spine MRI. There were no abnormal findings in the ophthalmology consults. And she is right now treated with baclofen for her spasticity. Next slide. So I think you might have to hit play on the video to start. Perfect. Let's see. So this is her at the age of 1 year, and we see that she is indeed toe walking. She has some slight scissoring, and she needs a little bit of support for her unstable gait. At the age of 2 months, we see that she the disease is a little bit progressing. She still has toe walking. She has the slight scissoring and the instability in her gait. See this more right now when she's rocking and we see her from behind. This is her at the age of 3 years. She is wearing AFOs and she needs to hold on to the handrails and needs quite some support by her upper extremities to be able to walk the stairs. This is at the age of 4 years. We see that the spasticity is progressing. She is wearing the AFOs. The scissoring is becoming more prominent. And this video demonstrates her troubles with fine motor coordination. And I think we can skip that one for time reasons. A little bit shorter. Okay, perfect. So this is her family tree. We see that there is no significant family history. She's the patient in red. Next slide. And when we were looking for an autosomal recessive mode of inheritance, we found these 2 compound heterozygous variants. The first variant is a missense variant inherited by the mom with a low MAF, damaging computational prediction scores, and previous research have reported this variant as likely pathogenic. The other variant is paternally inherited. It's in the splice region, also with a low MAF. The prediction scores showed a more possible benign effect of the variant, and it was previously reported as a variant of uncertain significance. Next slide. We then looked for phenotypical associations, and we found 2 reports on OMIM, a big database showing that there are 2 syndromes reported with this gene, both autosomal recessive. One is primary coenzyme Q10 deficiency type 7, one is spastic ataxia type 7. Both conditions have a variable penetrance of symptoms, variable age of onset, and we saw a certain phenotypic overlap. On PubMed, there were some reports of neonatal encephalomyopathy, cardiomyopathy, and respiratory distress syndrome. Next slide. Then we looked a little bit more in detail, and we saw that there were 2 recent reports from the beginning of this year for biallelic variants in COQ4 causing indeed hereditary spastic paraplegia. The patients described in this cohort were a little bit different than our patient. They had more complex symptoms. They were also a little bit older at the age of onset, and this led us to writing a case report about this. You can see all the variants that have been reported on this gene in the graph. The variants in red are all variants reported for spasticity, in patients with spasticity, the ones with that are underlined with ataxia, and the ones with the green square around. These are the variants that we found in our patient. And this shows the broad phenotypic spectrum in this disease gene. Next slide. And why is this finding so important? So one is it has direct therapeutic implications. The papers have found that there were fibroblast studies in patients with biallelic COQ4, mutations in COQ4, and they showed that the patients have a deficiency in COQ10, so we started supplementation of this of COQ10. And then we also started screening our patient for cardiomyopathy as a matter of prevention. Next slide. And this brings us to our second case. It's also a girl, 3 years old, with a normal birth and neonatal history. She has progressive spasticity in the lower limbs, which started showing at the age of 18 months. She now has hyperreflexia in the lower limbs, clonus, positive Babinski sign, some toe walking, spastic gait, ataxia, delay in motor and speech. The workup in the brain and spine MRI was completely normal, as well as the neurometabolic screening. Prior genetic testing was negative, and the girl gets baclofen for spasticity. Next slide. This is the family tree. We see some history of depression in her family on the paternal side, on the maternal side, some intellectual disability, but no one in the family showed similar symptoms like our patient did. Next slide. This is her at the age of 14 months, and we see that she is walking quite normally for a girl in her age. We don't see any evident signs of spasticity. And this changes now just a couple of months later. She's 21 months old. We see the spasticity, we see the toe walking, we see the unstable gait and a broad-based gait pattern. This is 3 months later. And we do see that the disease progresses when she starts walking. Pedro, maybe you can move a little bit forward in the video. Yes. She is experiencing more frequent falls throughout the year. But overall, at this age, her symptoms are more stable with the spasticity we can clearly see right now. Okay, next slide. And when we assessed her genome for autosomal recessive variants, we found those 2 compound heterozygous variants in WNT1, one nonsense maternal mutation with a low MAF predicted to undergo non-mediated decay, one paternal variant in the splice site region, also low MAF, with predicted impact on the splicing. Next slide. And looking at phenotypic associations, there were some associations with infantile onset recurrent acute liver failure and skeletal abnormalities. Then there was also a recent paper that described 3 patients from 2 families that have indeed early onset progressive spastic paraplegia and ataxia, but also additional symptoms like dysmorphic features and optic nerve hypoplasia. And interestingly, the variants that were reported, one variant is the one that was inherited by the mom in our patient, and the second one is really close to the variant that was inherited by the father. Next slide. And we then decided to take a closer look at the splice variant to better understand the function of this variant. And what we did is we started at the DNA level, the 2 up in the left corner, and this variant was predicted to abolish the splice site. We then moved over to the right part where we performed RNA sequencing, and this showed that it didn't affect the splicing, but effectively led to a deletion of the last amino acid of the exome. And to confirm our suspicions, we performed some lipidomic study. I'm just going to talk briefly about it, which showed that our patient behaved just like the patient that have been previously reported. Next slide. And also this diagnosis was really important for our patient because it now allows us to screen for his liver functions, especially in times of disease, and monitor him for ophthalmologic abnormalities. One other important thing to keep in mind, the patients that I show you here, they were really young, so also the questions of family planning are really important for the families. Next slide. To talk about the interim results of our study. Right now, of the patients that we have analyzed, 49 patients, we could find a diagnosis for 20 patients, could identify candidate gene for 17 patients, but for 12 patients, we could unfortunately not identify any variants of interest. Next slide. To talk a little bit about the additional diagnostic yield, so we could identify a diagnosis in 40% of our patients. And if we look now in candidate genes and in solved genes, most of those genes and variants were indeed identified on the exome level and not on the genome level. So the additional diagnostic yield of whole genome sequencing in our cohort is 15%. Next slide. To talk about the outlook of our project, so we are part of a pilot project for long-read whole genome sequencing with the Broad Institute. You already heard a lot about long-read whole genome sequencing, so I'm going to keep it brief. Inclusion criteria at this time point is that we don't have a genetic diagnosis despite short-read whole genome sequencing. For this specific problem, we also need parental DNA, but we will expand this in the future for all patients, even if parental DNA is not available. And the aim of this project is to determine the diagnostic yield of long-read whole genome sequencing in rare diseases. Next slide. So long-read whole genome sequencing, we get reads from 10 to 100 kilobase pairs up to 1 million. There are two big different technologies, PacBio and UNT. I'm just going to keep this brief. Different strengths and disadvantages of this approach. So one is we can detect repeat expansions and complex structural variants. And we can also do something that is called de novo assembly. So the first step when we make all these puzzle pieces of the DNA, the fragments, we have to reassemble them. And we often do it versus a reference. And this reference can have systemic errors. And long-read whole genome sequencing allows us to do a de novo assembly. Disadvantages of this approach right now are relatively high costs and also higher error rates of 10 to 15%. Next slide. And why is it so important to actually know your diagnosis? So we always want to go from disease to know what to target in a therapeutic item. And for this, we need to understand what's the mechanism of disease. This is something that Darius had also talked about. So there are different disease mechanisms in the genome, molecular mechanisms, that allow us for different techniques in gene therapy or amendable therapies. Next slide, please. And apart from optimizing treatment and new treatment options, there are other advantages of knowing your diagnosis. Next slide. This allows us to start with, to be an entry point for research studies. We can, next slide. We can also improve, help with family planning, next slide, help with counseling, better genetic counseling, and the two cases that I brought you here really show how important it is to know your diagnosis for monitoring and early intervention of possible consequences of your diagnosis that might not display right now. Next slide. And it also helps to build communities, which are really the foundation for a lot of research that is happening right now. Next slide. So to summarize my talk, the preliminary results are that we can see an increase of the diagnostic yield in 40.8% in whole genome sequencing, which is mainly attributable to exome-level reanalysis. We have a 15% additional diagnostic yield of whole genome sequencing. Genetic diagnosis can yield actionable clinical findings, and additional sequencing and analysis methods are warranted. Next slide. So to close my talk, I would like to thank all the patients and their families that are really the foundation for and driver for all our research, and also the big team that you can see on the pictures there, especially Dr. Darius and Amy Tam, who are also here in Tampa with me. Next slide. And I would also like to mention that we have the two studies where we are still recruiting patients. If you have any interest, don't hesitate to reach out, and that we have the Movement Disorders program at Boston Children's. Thank you very much.
Great job. Hey guys, if you want to, because we need to move on to get to our next segments of stuff we have to do, remember, her email is in your program. If you want to email her directly, she'll give you all of the answers. And
I'd be happy to.
yes, she will, because she's so great. And also, remember, all of these presentations are being recorded, and they're going to be on our YouTube channel in a couple of weeks once we put them all together and be uploaded through the website, okay? All these will be out there, so we'll get to look at them, and you can go at them at your own speed. You can speed them up and slow them down and look at all the quality research, and you can blow up the slides real big in your home, because we know you all have 90-inch TVs in your living rooms, right? So you can put it on your big 90-inch and see all the graphs and the data up real close. Okay, so remember that, couple of weeks, all of these will be out on the SPF YouTube channels. Okay, got it? We're going to take a quick couple of seconds here and get ready for some more stretching. Here we go.
I hope not too many people are leaving us.
There he is. All righty, Zachary, here we go again. You're on.
Yeah, I have this. Okay, so just give me 1 second. For everybody who can, let's stand up. If not, totally cool to sit down. And I'd like for you guys to grab at the back of your chair if you are one who's standing up with us. What? Too far?
Sorry, that's all bounce on. There we go. A little bit better.
Perfect.
There you go.
Okay. All right, guys, we're in. So if you need the balance, feel free to put one or both hands on your chair. And whichever, or the length which is most comfortable, we're just going to do some small kicks with our left foot. Now, if you can bring your leg up, totally cool. If not, tiny wiggles. And it really doesn't matter which leg you start with, just getting some leg swings in. I hope you're here. I spoke to somebody, gave me a recommendation for today. Thank you. You're welcome. And let's switch. I was hoping you were here. Good. Okay, how many of us have tried a squat before? Okay, so let's see. So we're going to grab a chair if we're comfortable. If not, classic, we'll just squat down here. Now, if you are not new, or if you are new to squatting, let's take it slow. So I don't want you to try to touch the ground with your butt. Let's make slow, it might look or feel weird. Slow squats until you find a depth that's good. And totally grab the chair if you'd like. If not, just keep it here. Something I said last time that I just want to push into everybody's brains again is simple concept that positive progress is possible, even if there are no cures. There are things that we can do every day to make sure that we're safe. So let's stop squatting. We're going to stand up. And now we're going to move our hips a little bit. So you could start with your left leg, and we're just going to swing it all the way out. Now, same as our squats, maybe you might be a little uncomfortable going all the way up, so you could try moving your leg up a little bit, and even if that's too hard, you can do little circles right in front of you. But if you are holding onto a chair and you have good balance, I want you guys to try to lift that leg up. You should feel it in your hips. It might hurt a little bit, be a little tense, but it's supposed to. And let's switch. Okay, and one more time through. We're just going to do some kicks like we did in the beginning. And let's switch sides. Okay. Now everybody sitting or standing, take a deep breath in, bring our arms all the way up to the sky. Just going to exhale, bringing our arms back down. Let's do one big inhale, bringing our arms all the way up to the sky. And like we did this morning, let's just release and hold. If you can. If not, you can totally just stay up and keep doing what we had before. Let's take one more inhale, bringing our body all the way up. And let's reset. Nice work, everybody. Nice work. So let's bring our arms straight out in front. Might be a dance, I'm not really sure. Forgot the name of this, so we'll pretend it's a dance. Get our arms moving a little bit. And let's switch. Let's pump our arms up in the air. I don't know about you guys, but I'm already feeling this. My shoulders are starting to hurt. And release. How does that feel? Does that feel nice so far? Okay. So I want you guys to follow this movement. We're going to bring our arms up here and then go straight up in the air. When you get up here, I want you guys to try to extend those shoulders up as high as you can. When you're bringing down, release your shoulders. Okay, so for this one, to start off, I'd like for people to grab the chair, and if it turns out being a little easier, then let's go without the chair. So we're going to work on our hips a little bit. Let me come out to the front. I have HSP, so I'll hopefully have my balance while I show this to you guys. So this is another exercise to open up our hips. So we're going to start, any hip. We're going to bring our leg up, and we'll rotate it to the outside. We did it this afternoon, where we were rotating our feet in and out, except this time we're actually going to do the motion. So we bring our leg up, try to turn it to like a 90-degree angle with the bottom of our foot. And if you do feel like you have balance, totally try it without the chair. I just wanted to make sure everybody was good. And we can switch sides. You guys feel that one in your hips? Yeah. Good, because I do as well. Okay. So once again, we can do the sitting or standing. We did this afternoon as well. We're going to bring one foot forward. So I'm starting with my left. You can choose whichever. We're going to have our right foot back. Our right foot is going to stay, or our right leg is going to stay a little bent. And we're going to keep our left leg as straight as we can. And from there, we're just going to slowly try to reach down for our foot. Grab a chair if you need, please. When I was up here last time, I could not stop laughing in my head because my parents used to bug me about stretching all the time when I was a kid, and I never stretched until I got older and I realized that I had to. So I find it very funny that out of everybody, I'm the person talking to you guys about stretching, considering my parents bugged me every day of my life to stretch. And let's just switch sides. We got one more. And just like that, thank you everybody for our stretches. It is 3:45. We have about 15 minutes until our small groups.
Way to go. Thanks, Zachary. You're the best.
Thank you.
Okay, everybody, you know where you got to go. You've got your...
Thank you for all the wonderful participants.
You know where your breakouts are and where you got to go. PLSers are down in this hallway again. The child onset in the front of the building on the second floor. Okay, and we got podcaster Kim Sanchez in here in just a few minutes. We'll see you soon. And Zachary, by the way, is also in the front of the building on the second floor for his breakout session. So thanks, Zach. You're the best.
Well, good afternoon, and we're getting toward the end of this second day with a lot of good information you're hearing from speakers, and we know some of it may be a little more difficult to see, and we can't ask questions of some of the folks who are getting with us by Zoom, but there are speakers with research that we felt like, Jim, we needed to hear from and be updated on, and so it's good to hear from them, Dr. Zuchner and Dr. Calame, and as we do this virtual each year, we're learning a little bit more about how we might do things differently, and we're going to try to find a way, working with our team, that maybe we can figure out a way next year to have you all be able to call in as we do that. That would be our goal. We'll be working in that direction and see what the possibilities are, but we're glad that you've joined us, glad that you've been able to get this information with all those things going on research-wise. With me now is, you guys all know, our fundraising chair and past president and whatever her title goes, with Jim Qiang. And I've asked Jim, we've talked a lot, obviously, about all the different functions of the foundation, Jim, awareness and outreach, and the fact that 60% of our people are first-time attendees, and all those things are important, but we know at the very top of our list of priorities is to raise money to move this research to a point that hopefully you and I one of these days have a treatment. So I wanted to get you on to talk with us a little bit and share where we are. It's almost halfway through the calendar year. How are we doing fundraising? What are we looking forward to the rest of the year? So let me just hand it to you to talk to us about that.
Great, thank you. And the good news is we're doing very well. We are right at halfway through the year. To date, we have raised just over $525,000, which is significant. That's actually about 5% up higher from last year at this time, so that's good. We've got some events coming up in the near future. I'll go over those in just a second, but in your spare time, if you can, go to the webpage and look at all the different types of events that we do throughout the year. Look at something that works for you, pick it, and make it happen, because we need everyone out there to participate, make personal donations, reach out to family and friends, share your story, make sure everyone knows the difficulties you deal with, and ask them to contribute, because this is going to hopefully bring treatment options and cures in the near future. What we have left this year so far with fundraising is we're actually kicked off the 5K run, walk, and roll. It's available now through the end of October. You can go to the website and look under events, and you'll see the list of events. Go to the 5K page. It'll list all the information on how it works, and it'll allow you to take a link to the peer-to-peer fundraising page. It takes just a couple minutes to upload a picture, set up your own personal page, and then you can share that with all your family and friends. You can do that through Facebook, emails, whatever you want to do. We're hoping to raise $80,000 this year, and again, this will go through the end of October. Shirts this year is what I have on, this dark blue shirt. That's actually for the folks, anyone that goes on and sets up a peer-to-peer page and raises $100 or more will receive this shirt complimentary. So look forward to that if you want. You don't have to be a superstar athlete to participate. Find your activity. The main purpose of this is to become physically active or be physically active and to raise money for the foundation. So swim, golf, canoe, tennis, golf, run, walk, roll, whatever it is you can do. Find something to be active for an hour or so. If an hour is too long, do something for 15 minutes, 3 or 4 times a week, or take 2 weeks, whatever. Make it your event, but be safe about that. After that, we'll be sending out the annual report. Greg and Norma and some other folks will be working on that. That's really just to show our community members how how well we've done in the previous year. So this will be results from 2023. That will be available here in the next month or so. And that's a great opportunity. A lot of people get excited about what we announced, about how much we raised and about research. So a lot of times folks give through that way. We'll be wrapping up the year with our year-end giving, including Giving Tuesday. So the first part of November, you'll be getting a letter in the mail, emails and stuff like that to promote our year-end. Usually we raise a half or even better than half of our annual money through the year-end campaign. So if you haven't given yet, please do if you can. Please reach out to your family and friends. And if now is not a great opportunity, please do so this year if you can. And we really need to step up and try to raise as much money as we can, because again, I think we've got some treatment options and maybe some cures in the near future. Lots of positive information here at the conference. We had at least 8 speakers that talked about positive results. Gene therapy, one researcher has a list of medications he's going to start doing patient trials on. It's just exciting, and they're working together. So it's a great opportunity to get involved and make sure you support the foundation, which will hopefully bring some good answers for us in the near future.
Jim, as soon as we get home from this event, one of the things on our top priority at our home is our annual Golfing for Rare Disease event that we do the Saturday after Labor Day every year. A number of golfers, sponsors will participate in that. And a half a day of golf, we normally are able to raise about $20,000 to $25,000. And if there are other golfing families out there, folks who know how to make those things happen, we've got a very simple model we'd be glad to share with you that maybe can turn your love for a sport into some money for the foundation where you're located. If you'll get in touch with us by email or in whatever way is easiest for you, we'll be glad to share those ideas about how to do that. And it does take some work prior to the event. I mean, you don't just wake up and go do the event. There's a lot of preparation, but it's worth the time and worth the effort, because $20,000 to $25,000 for an event's not a bad day. Right. So we'd invite you to do that. Jim, as we go through this process, you know, you as fundraising chair and Norman and I often get questions about, well, what's happened with the money we've donated in the past? How is research going? What do we know? What can you tell us? And I tell folks, we're not scientists and we're not researchers. It's hard to answer that question. But both to Dr. O and Dr. Fakari today clearly said all the research that we and other foundations and contributors have funded in the past have brought the research arena to the point now that we really are at the proximity, we hope, of finding some things that can make a difference with a treatment. You know, the CERN that we established last year, creating the 10 centers at this point, that clinical trials might be easier and quicker to do with the way they're putting that together. So a lot of good things happening. We assure you that a treatment and a cure is not happening fast enough for either of us either, but that's why we keep working as hard as we are at this to try to get that to happen soon. We have family members, we have friends, and we want to see their lives made better in the future as we work to do this. So that's what's going on this year with fundraising. We ask you to continue to doing what you have done well in the past, and let's have the best year this year. And I'm confident, Jim, I really believe, based on how things are going now, that come December 31, January 15, we're going to be able to say 2024 was the best year ever, and that we can get more research done than we've ever been able to get done before.
$1.5 million. Let's do it.
Thank you for your leadership with our fundraising committee and our fundraising effort.
Thank you.
Let me share with you guys, this will be the last time I'll talk with you today. In just a few moments, Kim Sanchez is going to come and share with you and folks here who want to learn about how to use social media, how to talk to folks. You may think, I can't do that, I'm too shy, I don't have the personality. Well, I want to tell you, if you look at social media very long, you'll find that you've got as pretty a smile, you've got as much personality as a lot of the folks that are out there doing that. It just takes some confidence and some willingness to do that. And that's why we have Kim here. She has HSP, as she shared with you yesterday. She wants to help us become better at getting our word out through different social media platforms and in other ways. So we hope you'll hang on and listen to her, and that you might think about doing that somewhere you are, so that your friends and family will know more about your condition and about the opportunity to raise resources to help us find these treatments and cures. We'll be back in the morning tomorrow. Two things happen tomorrow. We'll hear from all of our committee chairs about the work of their committees, and we always invite you to pick one of those committees that you might have a talent or gift or experience in, because we can use everybody's time and everybody's work as we work on these agendas and these items of work. Then following that is what's always kind of the highlight for a lot of people. Dr. Fink will be the last doctor who will speak, and he'll be answering questions, and that will be in the morning as well. So we hope you plan tomorrow to be with us and participate in watching that as well. In the morning, I'll come on at about 8:25 just to kick the day off and tell you again how things will go tomorrow. But I won't be seeing you anymore today, but hang on for a few minutes, listen to Kim, and let's get this day done. You guys enjoy that presentation, enjoy this evening, and we'll look forward to seeing you at 8:25 in the morning. Have a great evening.
Ready for our 4 o'clock breakout session with podcaster Kim Sanchez. This is not the official open, by the way, but if you want to move a little closer, you're welcome to. It happens in these late afternoon events when everybody kind of gets pushed away because of other events and things that are going on. So feel free to come closer, okay? But she's also performing to those cameras back there so that everybody will be able to see these later on our YouTube channel and stuff. So you'll look real good and up close and really nice because these guys are great videographers.
Awesome.
Okay? So, but for our audience, feel free to come up. Everybody has moved and taken some of their stuff and whatever. Okay? So don't you worry about a thing. It's okay. We don't bite up here. Okay?
I do.
So Kim does. That's a good thing to know. So, all right, we'll give everybody a couple of seconds to get organized and get kind of settled in, and you're going to learn a lot of good information and great stuff. So come on up, move a little closer. It's okay. Remember, these seats cost $500 a piece at the ballpark. I'm just telling you, you want to sit in the front row? It's 5 big ones right here, okay? Yeah, everybody says, oh, I don't like to sit in the cheap seats. These are the big money dollar ones. Come on up here. Get where the seating is good, right? This is what it's all about. All right, come on in. Get on down here. All right. All right. Now we're going to do our official open.
Okay.
Ladies and gentlemen, this is Kim Sanchez. She is a podcaster who has a lot of followers on Instagram. She has PLS, and she is a member of the Spastic Paraplegia family. And she's now going to be helping us help you tell your stories out to the internet and to the world so that you can do it on our platform, the Spastic Paraplegia website, and all those other magical places that live out there in the internet universe. And she is brilliant and excited and energized, and now it's her time to tell you all about it.
Awesome. Thank you.
Good job, Kim.
All right, thanks. So, like you said, I'm Kim Sanchez. I was diagnosed with PLS about 2 years ago. Actually, end of this month will be my official 2-year diagnosis. I have a TikTok and an Instagram called My Spastic Life. And it's where I share a lot of stories about just not specifically PLS, but just disability in general, right? Because I think it's something that it's new for me. I know some people were born with certain things, but for me it's new, and learning how to navigate life with a disability is definitely a challenge, right? Would you guys all agree? This world was not made for people who, have disability. And so a common question that we get a lot here is, how can I find other people like me, right? And a lot of times, people think, well, the doctor should be introducing us and connecting us, but the Spastic Paraplegia Foundation is really a good way for us to find people who have the same illness or disease that we do, right? There's different variants, but at the end of the day, we all have a very similar experience. And so using the foundation is a great way to learn about other people who have the same issues as you, to reach out and find community. But also social media is another great place. And so I have found that myself. I know I've been out in the hall doing podcasts this, past couple days. And when I talk about social media, I kind of get that I can't do social media, right? That's not what I want to do. And I can tell you that's not something that I ever thought that I would do either, because I'm definitely a very introverted person by nature. And so I really started as a way to almost therapy, right? To kind of get my thoughts out. What's my experience? How do I feel? How can I show people the good side and the bad side of living with a disability? So I'm gonna ask you guys a question. So how many people here have met someone new who you can relate to? Everybody, right? So all of you guys have been here. You've related to somebody on some level, right? Whether it be a shared experience through disability, it could be something about, your kids. You've met people here, and you've networked, and you found other people, right, who are like you. How did that make you feel? Good. What else? Did it make you feel more comfortable, included? Did it make you feel, better about it? I mean, that's something that I think we all struggle with, right? Trying to find your tribe and trying to find people who are like you. And the great thing about the internet and social media is, PLS, HSP, you're not going to find very many people down the street, right, who have that same disease as you. So social media really opens it up. And I know after my diagnosis, I was Googling like crazy, trying to figure out what is this? How do I fix it, right? Because initially that's what you're thinking is how do I fix it? What medications do I take? What mobility aids should I be using? Things like that. And so it really helped me to find a community. And I found a group on Facebook, which is a PLS community. And unfortunately, the PLS community is having their meeting right now. So all my people aren't here, but they're definitely, I can tell you without a doubt that they really saved me. Because after diagnosis, I felt like I had lost my identity. And I think, a lot of you can probably relate. Because, if you've, I was very active and I had dreams for the future, and then you think about it and something happens and you're like, now what? Now what do I do? And so finding that group of people who I could relate to, it really, really saved me. And so one thing I hear a lot is there's not enough people online who really are sharing the positive sides of things. And, I know there's folks in here who I recognize from social media who show working out, who show, how can they still accomplish their dreams and their goals, even if you have something that's holding you back. And so sharing your story on social media, I think, is really important, not only for you, for your mental health, which sometimes doesn't get talked about a lot. That's one of the biggest challenges. Sure. Is it hard for me to walk from point A to point B? Absolutely. But I can tell you the mental part of it, right? The how do I get through this day is sometimes the hardest part of it. And to see how someone else can get through the day and make it positive is definitely something that needs to be shared. And so I think, disability social media can have a negative connotation, right? You think about people who are, talking about the sad part of it and the demotivating parts of it. And so being someone who shares, are there bad days? Of course, right? That's life. There's good days, there's bad days, there's okay days, but at the end of it, every day that you're here is a good day. And so I always try to find the positive part of it and share that with folks online. So what I encourage you guys to do, is definitely, even if you are not someone who feels comfortable to share or start social media account, right, and share your story, because not everybody feels comfortable to do that, go online, find your people, right? So find PLS groups or HSP groups or local groups. Use the foundation to find folks who, you can reach out to, ambassadors in your state, things like that, because you have to be a part of a community and you have to find people who are like you so that you can feel comfortable and you can, live your life to the fullest. So does anybody have questions? Oh, yes. I don't have my, go ahead.
Do you have a list of social media accounts that are active out there that you could circulate?
Yeah.
Twitter accounts and TikTok?
Yeah, I could. Yeah, I've got mine, and when I end up, so I can tell you there's a lot of, having rare disease, there's a smaller group of folks out there, but there are a lot of really great disability accounts that I follow, so I can share mine with you guys, or you can find it in the packet, but yeah, I would love to share that. You have my information, and then I think you can take that and kind of look at who I follow on Instagram and TikTok if you on those. I don't have Twitter, but you can look at those and see kind of folks that I follow. But usually what you can do to find it is if you look for the hashtag, right? So hashtag disability, hashtag mobility aids, hashtag HSP and PLS. I find a lot of folks, MS is one of the bigger ones, right? And so spasticity is one of the symptoms of MS. And so, that's one that I tend to follow and find other people who, that I can relate to that just talk about the general experience of being disabled, right? Not necessarily specifically PLS or HSP. but that's a great question. There's a lot out there. Some are good. Some of them I followed, and it's not the content for me, or it's not relatable. But you can find people out there who have similar situations, and that you enjoy their content. Yeah.
Hi, Kim. I would like to ask you to concentrate on or focus on your podcast and with SPF, and I wanted to make sure that there was like a place on the podcast where you address, certain aspects of PLS and HSP and maybe age groups, because we have a lot of younger adults. I think that the podcast is a perfect segue to reach some groups that are not part of SPF right now. So I commend you for that. But I was wanting you to focus on your podcast and tell us a little bit more about what we can do to help you.
Yeah, that's a great question. So the first version of the podcast is here. So I've been in the hall and I've been talking to people, and I can tell you it's been both a lot of joy, but also a lot of hard stories, right? Because people are sharing their stories and things that they might be struggling with, or things that, newly diagnosed, right? And you're lost and you're trying to figure out what to do. So it's been, for me, it's been great, but it's also been difficult, right? Kind of hearing stories, and it brings back those memories of my own journey and things like that. So this is our kind of inaugural podcast initially, but we do want to focus on specific things because HSP and PLS are similar, but there are differences between the two. PLS specifically, sometimes one of the challenges is you're kind of in that middle between HSP and ALS. And so you don't, for me specifically, I go to ALS clinic, but I don't really fit in there, in some ways. But then there's not another clinic or another place for me to go. So, you're kind of in limbo and you're not really sure where to go. So, focusing on the differences, like you said, between HSP, PLS, even some the different subtypes could be something to focus on. But I think one of the things definitely that we want to focus on with the podcast is reaching people who are, in that younger generation, right? Who are, they are online and they are trying to figure out, like I said, I Googled the heck out of PLS because I had never heard of it. I mean, how would you know, right? So when I got the diagnosis, I Googled it and nothing came up. I mean, nothing. And so having that out there, I think is great, and getting the opinion. So what I'll do is, on the website, we'll have information to contact me, and we could do a virtual if you wanted to participate. We could do virtual through Zoom, things like that, and record it and be able to share your story with others. So things that you've learned, if you've, have years of experience, things like that, different ways that, I know traveling, things like that, or things that people, how do I travel? If I have a wheelchair or a scooter, or, how do I, what cities are disability friendly, right? And learning from other people's experience. So we can do a variety of topics. And so that can be done through Zoom. So you can do it virtually. So we'll put it out on the website and share it out there so that we can do interviews with folks and share just different perspectives, like you said, different ages. Because I know from just kind of talking to people, my experience of becoming disabled in my 40s, and I've already got, my kids are sitting right here. I've already, I've got my kiddos. But if I was 20 and facing this, right, it's a completely different experience, right? How, what does my future look like? Or if I'm a parent or caregiver or family member of somebody, that's a completely different experience than someone who's living it. So that's a great, Tina, that's a great topic to talk about and just give all the different, because it does. I mean, as much as you don't want it to affect your family, it does. I mean, my kids would tell you that in a heartbeat, right? It changes everything, sometimes for the better. And I think about that too, sometimes where, if I could change it, would I change it? And I don't think I would, because I think it's made me focus on the things in life that matter more so than just, you know, the day-to-day grind, right? You go to work, you come home, you have dinner, you go, you know, sports, things like that. And now I focus on the things that matter, the purpose, right? Enjoying every day, because I don't know what tomorrow looks like. And so I think we are blessed in a way that we don't often consider, but we're blessed in a way where every day feels big, right? Where someone who, you know, maybe isn't facing that same challenge, isn't really, you're kind of just going through life, maybe numb, right? Or maybe just not really experiencing it. What other questions or comments?
Hold on, if you got a question, I'm bringing you a microphone.
Oh, awesome, thank you.
I don't follow you yet, but I do follow a bunch of other disabled content creators, and one thing that upsets me is seeing comments from trolls, I guess, would be how do you cope with that? I don't know if you get that on your accounts, but I imagine given the way many people in the world respond to disability, you might get a few. So I'm curious how you keep going with that sort of nonsense.
That is such a great question. And so kind of the backstory about me going on TikTok initially, because I didn't have TikTok before. I did have an Instagram, I had my Facebook, mostly it was, you know, talking about my family and life, just like everyone else, right? Like the highlight reel of life. And I switched to Instagram. I created a separate one, and it was really, like I said, kind of more therapy, right? Like me putting thoughts. I was going through a lot of changes in life. It wasn't just the disability. I was changing jobs, and I was going through a divorce, and my kids were getting older. So I had a bunch of things happening at the same time, and it was during COVID too. And so I thought, you know, let me kind of create this anonymous Instagram where I shared my thoughts about things. And then when this happened, I switched gears to it, you know, to be disability content. And it was actually my brother who encouraged me to start making videos, because he said, you know, you're good at speaking, you're good at talking to people. And I was like, no, I don't want to put myself out there, right? I don't want my face, like, I want it to be anonymous. And so he had encouraged me to do it, and I was scared to death. And the reason why is because I thought, well, how am I? I'm already, you know, struggling with so much, and I'm already feeling very vulnerable. What if somebody says something? And I'll be honest, like, for me, I am surprised. I've only had probably 2 bad experiences, which there's other content creators that I've talked to who I think the key is, if you are real and you are vulnerable, people do tend to not come after you as much. I think it's the people who maybe aren't authentic or, you know, braggy, kind of, you know what I mean? Those are the people who get it. But I'll tell you, so one story, there's one person who has PLS, and she had gone through all of my videos, which I've, at this point, since I've been doing it a year, I mean, I don't know how many I have, probably hundreds. She had gone through video after video and kept making negative comments, like, you seem so happy you have this disease. Like, why are you so happy? Your life is over. You shouldn't be happy. And at first I thought, well, this is an opportunity. You know, it's clearly she's hurting, right? Because she's saying this to me, so she's obviously feeling something. And I believe that, you know, how people treat you is a reflection of how they feel about themselves more than you. And so I thought, well, maybe this is my opportunity to reach out to her, right? And help her feel better. So she would post negative comments, and I would respond, you know, well, you know, my life's not over. I have a lot of good things in my life, you know, but it just kept escalating. So at one point I did block her, because I was having a bad day anyway, and she commented something. I thought, oh my gosh, I can't take this. So I blocked her for a little while. And then what I ended up doing, and this is kind of funny, is, and it's kind of happened recently, I was having a bad day and I was posting a frustrated video about something. I can't remember what happened, but I was frustrated about something, or it could be, I don't remember exactly what it was, but I posted something and I actually unblocked her and I tagged her in it. And I said, this video is for you. And it was me sad. And ever since then, she hasn't said anything to me. She didn't even respond to that video. But that's the one thing that I could think about. But I do feel like, you know, there are some people who are trolls, right? And are just mean. But I also feel like there are people who are going through the grieving process, and maybe the way that they're manifesting that is to, you know, say things. And so what I try to do is use it as a tool to try to help that person. Does it work always? No. But in this case, you know, I thought, let me show the good and the bad, right? So I show the days when I'm happy and I show the days when I'm sad, because I'm real and I don't want to just show the good things. I want to show the parts where it's hard. So that's how I deal with it, but I have been blessed. I thought for sure I was going to get a lot of hate and a lot of, you know, some people get things like you're faking it, right? And things like that, or you're doing it for attention. I haven't experienced that, and I don't know, I don't know if it's luck or just how I approach things, but I haven't. I've had a few, but I try to give them grace. I think there was another one. Oh, in the back.
Hi. I don't have any social media. I'm not on any social media. How do you get started? My neuroemotional specialist, he knows about some of the things I'm doing. They're kind of off the wall. I really take a lot of adaptive things to try and stay active. And he thinks that a lot of things that I'm doing that other people should be aware of, that your life doesn't have to stop. And for your podcast, like, how did you know you had enough content to go ahead and start doing podcasts? Like, because I know you started, like, I guess on social media first. So yep. When did you decide to start doing podcasts?
Yeah, it's a gradual process. You know, like I said, I started with photos and written content. Then I went to short videos. At the time when I started about a year ago, TikTok had a 3-minute limit. And so that was, I'd have to speed talk through those. But I also knew that I could keep it very concise, right? So I had to think about what's my message, and I had to be very concise. Now TikTok has a 10-minute limit, and I think they just increased it to 60 minutes. So you can upload longer content. The podcast, so actually my daughter did one of the podcasts with me. The first 2 I did, I've only published 3 right now, and I want to do more, but it is a bit time consuming, and I still work full time, and you know, I've got a lot of things. But what I ended up, the reason why I went to the podcast was time, right? Because some things you want to go deeper, and also I wanted to have the ability to it not just be my experience, but to share the experience with others. So what I love, I mean, here I've been able to talk to so many people and what I've realized through that, because that is the first time that I've interviewed other people. I have a lot of friends, like my best friends now are in the disability community, which, you know, if you'd asked me 10 years ago if that was going to be the case, absolutely not. I would have never considered that, but they are some of the best people, my best friends. So we talk about things, and so what I've learned through interviewing people here is that no matter where you are in your process, no matter what age you are, no matter what your circumstances are, we all go through that same human experience, right? So it's grief, it's overcome, adapt, right? All those things that you have to learn how to do. And so to be able to, so the podcast part of it really has become, how can I not only give my perspective, because there's only so many things I can talk about, right, where it becomes redundant, but also how can I bring that, share people's stories. And when I say share your story, it's not just about telling people what happened to you, but it's about sharing how do you feel about it. Because I think people, common thing I hear is, I don't like asking for help, which I'm sure a lot of us in this room think about, gosh, that's the hardest thing, right? To ask for help when people mean well. They just want to be there for you, and you want to be independent and you want to do it yourself. And so things like that shared experience and being able to hear it from different people, I think is really important, because we are, all the same. And, if you can share your experience with someone, hopefully get them through that process faster, and give them the wisdom, or, if you have different things that you're trying, as far as, some people go holistic, some people go, different exercises, sharing those things. Because with a rare disease, one thing that we've all kind of figured out is that the doctors don't always know, right, the things that are going to help us. We have tried the different things, and we share with each other. And I have learned more about how to manage my disease from my peers who have it than a doctor has ever shared with me. Because typically, I could tell you my diagnosis experience was kind of, this is what you have, there's no cure, there's no treatment, see you in 6 months. And you go home going, I don't know what I'm supposed to do. And so learning from each other, I think, is really what's important. And so if you do a podcast, you can share other people's experience, and also you get a lot more time. So, and trying to find people who, they just find you. I can't explain it. There's been no, I don't have a secret. I'm definitely not, I don't have 1 million followers or anything. I have a few thousand. But, people will find you. And probably twice a week, I will get a direct message from somebody who either is trying to get a diagnosis, just got diagnosed, or a family member who doesn't know how to support someone through the process. And so we'll talk, or they'll text me, or we'll have a conversation about it. But if you put it out there, people will find you. And it's, to me, it gives purpose to what's happening, right? Because you can't explain it, because you think, why me, right? Why am I the one? But when you have purpose behind it, it really helps to accept it and to, make it a lot easier, is what I find.
Is it possible to collaborate, especially if we're beginning, say she wants to share, could she maybe hook up with you and you guys could do a joint podcast to start?
Yeah, and definitely. And so we'll have my information on the SPF website. And then what we can do is just schedule some time to do a Zoom and just record it and put it out there. You can do voice podcasts. I think sometimes people like to see, faces along with it. So I prefer to do the video. It's been really, really good. But yeah, my information will be out there. So if anybody wants to share, they can just go on there and reach out, and then we can schedule some time.
Thank you.
Yeah. So the SPF podcast is going to be on Spotify. There's a link. There's nothing posted yet, so the link is not going to take you there. Mine is also on Spotify. So the question was, where does it live? Yeah.
I just asked the question where the podcast was.
Yeah. So it's going to be on Spotify, but the link to it for the foundation will be on the foundation website. So where you found the agenda, you'll see a link to the podcast there. And then my personal social media is My Spastic Life everywhere. So it's TikTok, Instagram, and also Spotify. And if you do have a chance, my daughter, she's great. She volunteered to do a session with me where she talks about what it's like to have a parent with a disability. And I learned so much from her. And I think as a parent, that's a common worry, right? How is this going to affect my kids? And what I've seen is that there's so much more compassion that they have for other people. This is where I'm going to start crying. But they're so compassionate towards others. And I think without this experience, it's changed them. And I'll tell you a quick story, because I remember in the beginning, we were at a water park. Y'all probably remember this. We were at the Gaylord. And I was trying to get on and off the bus to go to the water park. And my son was, 15 maybe at the time. And he was helping me. And there was a woman, and she said, when I was growing up, my dad had a disability. And I completely, like, I'm watching you guys, and, I can really, I can see the frustration that you have, right? Because I want to do it myself. And he was helping me, and I felt kind of, it felt awkward because it was so new to me. And she said, what's going to happen is someday you're going to realize it's going to be so good for their hearts, right? And they're going to be the most compassionate people. And at the time, I was like, no way, no. I was like, this is, no. But, I can sit here probably 4 years later from that point and know that their hearts are changed forever. And it's made them so much more compassionate. So gave me a thumbs up, so it's true. Love you guys.
Kim, so I'm curious. Now, admittedly, I'm not huge on social media, but so you have a better perspective than I do, I'm sure. But obviously, for spreading awareness, social media is an extremely powerful tool. And sorry if any of this has already been chatted about. I was a little bit late, but my question for you is, and this doesn't seem like something we can force, right? We can't just all say, hey, like, we need to do more of this, so let's all get out there and start posting. So my question for you is, what would you say the current state of, like, HSP and PLS social media looks like, and what could be done to help promote more of that? And I'm sure this session would be something along those lines, but I don't know, anything else we could do to help spread the word and help get more people motivated to be more active on social media and sharing more of their stories?
I think that's a great point. One thing, I mean, it doesn't have to be on a big stage, right? And so even if, and I know sometimes this is hard because one of the struggles I think a lot of people can, relate to is, how do you come out and tell people, right? Especially if it's something that's new. How do you tell people about your diagnosis? Or, I was just talking to someone right before this and saying, at work, a lot of people see me virtually. So I'm on a meeting, and so they don't know that I, can't walk. And so what I usually do is I tell them ahead of time if we're going to meet in person, or I'm going to a dinner or an event, or I'm, for work sometimes I speak at conferences. And so I tell them, hey, I have a disability, I use a scooter, so that they're not surprised, right, when I go out there. But even if you don't go out on a big platform, I think sharing it yourself, I think brings awareness that, I'm just like you. I just have, this aspect of my life is different. And normalizing it is really, I think, what I want to do, right? And so PLS, HSP probably isn't going to touch, many people, right? Other things touch more people. But the disability part of it, and what do you do, right? Do you offer to help somebody? Do you make comments about it? Do you not talk about it? Do you say, people don't know what to do. And so I think sharing that, educating people is one thing, right? Even if you're out in the world, people ask me all the time, do you want help? And I'll usually say, if I do, yes, because I think that's, if someone wants to help me, I need help, it's a win-win, right? I need you to open the door. You want to open the door, that's great. That doesn't make me unable. I could figure it out, but hey, why not just take advantage of it? But, if somebody does something like, for example, when I was using a cane, if I were to lean on the door, and I'm trying to open the door, I have all my weight on the door. And if someone comes from the other side and pulls it open, well, hello, I'm going with it, right? And so what I'll do is I would take the time and say, hey, if somebody's opening the door, they have their weight on it, like, you need to be careful and not, don't do that. Or if somebody makes a comment, sometimes people do make comments that they mean well, but if you see that person often, sometimes I would say to them, that type of comment, like an example I gave to somebody was I was walking into church and I was using my rollator, and it took me a long time. And when I got to the door, the greeter goes, "Yay, you did it," you know? And I thought, oh my gosh, like, and I know that she meant well, right? I think she was trying to be supportive and encouraging, but you think about that and it makes you feel, right, like a good child, you know, or something like that. And so, you know, things like that, educating people on what's the right etiquette. It's okay to ask people if they need help. It's not a bad thing. And the worst that they can do is say no. You know, but you know, things like if you're out in public and someone's talking to the person with you and not directly to you, right? I'm sure you've all experienced that. I experience it. I do a lot of work travel, and I'll be with coworkers, and I'm trying to, you know, gate check this, and they're talking to the coworker, and I'm like, "Hello, I'm right here." Like I can talk. But you know, things like that, people don't know, and so educating people on it, I think. Even if it's a small circle, right? Even if you only affect or touch 5, 10 people, not 2,000 people, it does make a difference. And, you know, I talk about the podcast with my daughter and stuff, and there's some things that she says in there that friends of mine and people who have listened to it, it was very eye-opening to them about, you know, things that she's seen happen to me that she's like, that wasn't okay, or that wasn't right. And they're like, I never thought about that, you know? So I think sharing it with people, coworkers, you know, and just being present and being out in the world is even, you know, not necessarily having a social media account, but being out in the world and showing people, I can do all these things, right? I'm not gonna hide in the house, I'm gonna go out and do things, right? It may be different, you know, but I mean, I go to my kids' sports events, you know, it's not easy. There's challenges, right? There's things that I have to do to accommodate, but I think it really shows people a day in the life, right? And that's powerful.
Anybody have any other questions? Come on, hands. Come on. You know you want to be secret podcasters. Come on, because I know I want to do.
It's a lot of work, I'm telling
Yes.
you. But also, I think kind of to piggyback off of that question is, you know, even if you don't, support those who do, right? So encourage them. I get back to the question about the trolls and things like that. You know, I am blessed that I don't get a lot of negative commentary. But even if somebody just writes, you know, I post a lot of videos of me doing modified workouts. You know, my balance isn't good. So, but I still want to work out because use it or lose it, right? So I do a lot of workouts like on my knees, or I modify different things. I can't go as hard as I used to. And, you know, I've learned to accept that. But if somebody even if they just post like a muscle or, you know, something like that, it makes my day, right? Because I'm like, okay, you saw me. You know, it's making a difference. So even if you're not the one who's out there putting yourself out there, encourage those who do, because it does make a difference. I know there are some folks out there that put out disability content, and they've made some big changes with like airlines and how they, you know, with wheelchairs and mobility aids and things like that. It does make a difference, and bringing awareness, right? Like, so encourage people who are out there if you can, and support them, because, you know, if I were to post content out there and nobody liked it or nobody said anything, I probably would stop, you know, because it's kind of like just talking to the wind. But, you know, the encouragement, even if it's just a like, even if it's just a emoji, right? Like, hey, good job, or, you know, keep going. That means the world to those people. And so those people can reach more people, and that's what that ripple effect of change.
Question. Hey, what? No.
Oh, hold on. He's going to come to you.
You got to raise the hand, then you got to wait.
And I want to share. So she has one called Falling for Fitness. So you should also follow her. She's amazing.
Well, thank you. I appreciate it. And she has really great content also, by the way. And it is, she's very transparent, which I find very valuable. And you've shared so many great tools and things that you've learned. And I've definitely, like the Cadence shoes, I hadn't heard of that until your Instagram post. So then, of course, I went and investigated and all that good stuff. And she's posted some really great supplements and some different things that, you know, I've looked into just because she posted. So, yeah, there's definitely benefit there. But my question was, I know a lot of us are interested, well, I have an Instagram page, but for anybody that's getting started too, are there any tools that you use on social media that help you, like with your posting or like managing those posts, anything to that effect?
That's a good question. Well, I have found, for me, TikTok is very easy to use. I know not everybody loves TikTok, right? It's very easy to use and edit videos there. There's also CapCut, which you can use, so you can use your video and you can add music or effects or things like that to it. So one thing I really encourage people, a lot of people don't want to speak or put their face out there. They want to kind of just have pictures. I think it's so much more meaningful when there's an actual person behind it that's relatable. So I know it's hard. I just honestly, like 90% of my videos is just me sitting in my office, or I have in my backyard, I have a hammock, which I always post myself in the hammock and, stuff like that. But it's just me and my phone, and I'm videoing myself and just kind of winging it. I usually think about, kind of the night before, sometimes if it's going to be content that's like I do Motivation Monday stuff, and I'll think about it Sunday night, what do I want to talk about that day? And so there's not specific tools. I think if I was, there are tools out there. I just kind of wing it. And I find personally, those are the more endearing videos, right? The ones that are overproduced sometimes, it doesn't feel as authentic. But just somebody talking into a camera about, here's what happened, or here's how I feel about things, or today was a rough day. I mean, there's been times I've gone on and cried, and it's just like, well, should I post that? I'm like, might as well. And, people will, support. And it was a bad day. We all have bad days, right? And I don't go on there with the intention of going, okay, today I'm going to post something about me crying. It's, this is how I feel, and it's raw and it's real. But yeah, there's a lot of editing things that you can use. I find TikTok is easier. You can do Reels. You can do, CapCut's a good one. But sometimes, just adding music is good. Sometimes, talking is good. It just depends. And I'll tell you one thing that is something that I had to learn the hard way is if you do start doing it and you don't feel like posting, just don't post. You don't have to. And I used to feel like if I don't, like I'm letting myself down, or the people. And I thought, nobody cares, right? If I'm having a bad day and I'm tired, or I don't want to add more stress to my life, right? This is something that's supposed to be fun and relieve stress for me. And so if you're not feeling it, don't put pressure on yourself to do it. Just do it when it feels right, because that's where that transparent authenticity comes from. Is when I do it, it's because I have something to say, and because I feel, good about it, and I feel like it's important to say it. And when I don't, I just don't, and I move on.
Any other questions from anybody? I've got one for you. Oh, sorry, but I'm going to do mine first. You talk about music when you do your videos and stuff like that. What's the copyright issues with those? Any copyright issues with using music, on your podcasts or whatever you need to do behind it for licensing, or any issues like that?
So if you use TikTok, it depends. So if you use TikTok, usually it's already, it's kind of a free-for-all. It depends. It can be banned in some countries. So I've had messages where it's like, in Russia, your video can't be seen, or something like that. But then there are times, like I know Taylor Swift had one where recently any video that had her music in it, it took the audio out, which means that all the audio, including what I might have said, let's say I use it as background music, everything is muted. So there is a risk sometimes with using that. So what I try to do, it depends on the video, but if I'm actually making content that I want to last, I will do generic music, right? Like happy music, or, things that aren't artists creating. So that way it doesn't get blocked. Because if you do use like Taylor Swift or something like that, it can get blocked, and your whole video does get muted.
Sure.
So
good.
Yeah.
Good answer. Thank you very much. Here's a question right over here in the front.
So this isn't really a question, but something that I noticed. I feel like a lot of this stuff is like not just for people with disabilities because, you know, everyone has a bad day, everyone has struggles. But I also think that it can help people without a disability because it can put it in perspective that, you know, what you're dealing with probably isn't that huge of a deal whenever you step back and look at it. And like normalizing it is like helpful, and like the compassion thing, like seeing the hard days will make you understand more, and, you know, just make it not so huge of a deal whatever you're dealing with at the time. So
You're so right.
I think it could be helpful for everyone.
You're right. So that's my son. And yeah, and you know, we talk about that all the time, is that everybody's going through something, right? Everybody. And it's all relative. I was talking to someone earlier about this. I said, maybe your hard day is you have a bad hair day, right? And that's based on what's going on in your life, right? So maybe your biggest struggle today is my hair didn't turn out the way I wanted it to. And maybe my biggest struggle was I face planted this morning getting ready for work, right? So, you know, everybody's experience is different, but we all share that, and to put it in perspective. And that's one of the blessings of this is, you know, we get to have those conversations of when something bad happens, we do talk through it and go, okay, in the grand scheme of things, where does this fall, right? Is it the end of the world, or is it just a little speed bump, right, that you have to get over? And I would say for me, you know, probably 80% of people who do follow me are not part of the disability community. They're just people who have found me through motivational things and things like that. Because I try to mix it up, because I would try to do funny stuff and, you know, different things just to kind of get a wider audience. But it's relatable because we're all going through something. And so, and that to me, I love, because if 80% of those people, you know, if 20% of the people have a disability, I can help them to deal with it, right? And then 80% of those people, I can teach them what it's like to walk in our shoes, and how can maybe they treat people differently, or what should they do, what should they not do. So I love the fact that I'm probably more heavily followed by people who don't have a disability, because then they learn something, and then they share, and then they share, and then they share, right? And hopefully, and that's always the thing that, you know, if I could change the world, you know, I would love for the world to be more accessible, and I would love for people to learn how to treat people who are different, right, in general. And so I feel like for me, you know, like Evan said, it's relatable to everyone, because we're all going through something, and it's all relative to your life circumstance. So your story, even if it's, you know, whatever it is, it impacts people far more than you know, or that you can even imagine.
Great. Any other questions? Because I'm going to ask a 10-part question.
10-part?
10-part with subsets.
He
in
will
there.
too.
No, it's okay. Hey, here's one for you. We talked about your ability to help the members of the Spastic Paraplegia Foundation, right? All of our members, to help them tell their stories. We'd obviously want to have hundreds of Kim Sanchez's out in the world talking about HSP and PLS and reaching a broad audience and stuff. How do you help them reach an audience? Obviously, you were talking about an 80-20 percentage breakdown over an audience. How do you generate that audience? Where do you find that audience? And how does the podcasts generate to people? How do they come and find you so that you can tell your story, or our members can tell their stories to get the word out to a larger group?
So the strategy that I followed initially was I mixed it up because I wanted to get different types of people. I think, you know, if you post it, right, the people who are looking for it will find it. The people who aren't looking for it may find it by accident. But I think having a foundation like SPF to be behind you is huge, right? Because then that can help to, you know, even, you know, on social media, on our Facebook page, right, I was seeing people going, oh, I didn't know there was even a conference, right? Which is so sad, because I thought, wow, we did such a good job marketing it, you know, but still people didn't know about it. And so I think having a big organization like that, or partnering with like Cadence and things like that, right? That having those folks is going to give us a bigger reach. You know, I think from a personal perspective, right, it's mixing up your content so that the people find you that aren't necessarily looking for you. But then you also include things like the hashtags and the keywords for people who are looking for it. So I would put, you know, spastic paraplegia, or I'd put primary lateral sclerosis, or, you know, whatever the keywords are. So if they're looking for it, they're going to find you. But I think having the foundation is really, really helpful. And then, you know, I think if you're new to it, I think, you know, someone like me who can ask you questions, it's a lot easier, right? Sometimes it's hard to just think about, what am I going to talk about? But, you know, asking some of those hard questions and kind of being interviewed sometimes, if you're new to it, is easier. And it gives you that confidence to think, oh, I can tell my story. Where if you think you can't, you know, someone asks you some questions, like, you know, my first question to everybody was, what's your favorite part of the conference? And that would open up, you know, the next thing that we talk about, right? And, you know, so you kind of go through that and you realize, oh, I can do this. I can share my story. So I think that's a key is, you know, interviewing is a lot easier than just creating content.
Wonderful. I think we have a question right here in the center.
Just a comment, Kim, I'm not on social media, don't do near as much of that as Norma does every day, but you mentioned hashtags, and I know she's been using for the past couple of years hashtag HSP and PLS. That's simple and easy for us to remember, so if you want to get started on social media, that's a good hashtag to throw into the mix.
Yeah, it is.
Any other questions? Well, wait, no, nope. You gotta wait. You can't raise your hand and then disappear. Here you go.
Thank you. This isn't a question, it's more of a comment. For people that are my age that aren't really savvy about social media, please explain to us what a hashtag is and what it does.
So I'm older too, in a way, but basically what a hashtag does is it will, I don't know, my kids could probably explain it better, but it's a way of organizing things. So if I put hashtag disability, if you were to look up, let's say in the search, you know, on TikTok or Instagram, if you put in disability, it would find anything that has that hashtag. Number sign, yeah, the pound sign, right? If you've ever used a phone that wasn't a cell phone, it's the pound sign. But yeah, so it's a way to kind of organize things, and then you can also follow a hashtag or a pound sign, and you can follow it, and then anything that comes up with that. So let's just say I follow hashtag disability, anyone who posts content with that, it will come up in your feed. So on Instagram and TikTok, it will automatically come up. So that's great because, you know, that, and you have to find the right ones. And honestly, one of the easier ways to do it is you can Google, you know, top hashtags for disability or top hashtags for mobility aids or whatever, and it will give you a list of things that you can put in there, and then you just copy paste them and you see what sticks. And I've kind of gotten to a point where I know which ones are going to attract, you know, the people that I want to attract to my page, without attracting the people I don't want. And so, yeah, you can look that up and it'll give you ideas. If you don't know what hashtag or pound sign or tic-tac-toe to use, you can just Google it and it'll give you ideas and you can start there.
Great question, Tina. Any other questions? I'm scanning the room. All right. Hey, I've got another one for you, though. When you develop your audiences, I mean, you obviously create content for a specific, not only for your benefit, but to benefit other people. Yeah. And so we talk about using hashtags and keywords that can find that, which is, you know, basically how the internet works and how it has its logarithms or whatever to make things come together. How does your audience expand just naturally with people that follow you, that because of the keywords and also the content and do they stay with you for a long period of time? And then here's the seventh part of the question is, can you guide and direct them to do things for you, such as fundraising events and things like that? and also to inform them and information and to send them information. Is there an upscale of information that comes up to, do people reach out to you and say, man, I need a neurologist, what do I do? Does that come up in those kind of conversations?
Yeah, it does. That's a good question. Gosh, that was a long question, Tim, so now I'm forgetting all that you said. But yeah, so organically you can grow your audience through the keywords, right? And so that's part of it. You can partner. So I've partnered with some people. Gary's in the back. He and I have done duet videos together. And so his audience and my audience will both see it. And then you can get followers that way. I can tag like at Spastic Paraplegia Foundation. And so then people will see that and they can click that and find the webpage. I mean, you can spend money to promote your website or your social media if you want to. I haven't done a lot of that, but you can spend money to promote it. You could partner with companies. I haven't done that either, but you can have, sponsored content. You can have ads that run. So there's a lot of ways you can boost it. Mine, for the most part, I've just kind of organically grown it, and people will, especially on TikTok, things come up on your For You page based on algorithms, so things that you look at, right? So you guys know, as soon as you talk about something, your phone is like sending you ads for it, right? So if I'm thinking about, I need a new scooter, right? My videos may show up in your feed because you're thinking about buying a scooter. So that's how I've done it. But you can definitely partner with other people, other content creators, companies. You can tag them, which is just that at symbol, and then you put the name. Things like that. That helps to get more visibility. If you have other people, they can share it on their story, which then will kind of link back to you. And that's where I have found a lot of my folks that I follow for disability content is through stories, where they share the story, you click on the story, and then you get to their page. So there's a lot of different ways to do it. You can pay money, or you can just kind of organically grow. My goal is not to get numbers. I'm not doing it to make money. I think people who do it to make money obviously are going to spend money to do it. Mine is just kind of a hobby for awareness. So I let it just kind of grow just by people finding me. And some people do. I mean, it'll give you insights if you want to. You can see how many new followers you have, how long they watched your video, where they found your video, do they stay. For me, I mean, at times I'll lose a follower here and there, it just happens, right? But my numbers have been pretty consistent, and so.
Great. We got one more question for you, right back here.
Well, actually, it wasn't a question. It just another comment about that is, because all these things a lot of them have algorithms. So when you first start, get your friends on board. Get your friends online when you are posting, so it raises the number so that the machines, whatever, can recognize and promote it more. It's just my suggestion, reach out to all your friends and your Facebooks and so forth, whatever organizations you're in, to help you get the numbers up, even just so that it gets out there. Not necessary for money, even though that can be there too, but just to get it out there, that can help.
Good, yeah. I think that's one thing is if you, follow someone, follow them back, right? If someone who is in the same, disability community, right? They follow you, you follow them back, and it does help raise numbers. Yeah.
Wonderful. You've done really, really well, and we're glad that we are partnering with you to help us in this social media way. Everybody be on the lookout for her stuff. You should all be there clicking right now. I saw the phones. Look, Tina was a teacher for a million years, and whenever the lights were out during the movie time, everybody had their phones on under the desk. So we know what you're doing here. So make sure that you guys connect with Kim. Okay, you should all be doing it right now. You all have social media. You do. You all have that good stuff. Find her out there. Click on her page. Join with her. And then she's done a lot of work the last day or so, grabbing some stories here at our conference. And so we're really happy about that. We can't wait to see those when she produces those and put those all together. And will continue to help us spread our message around the world and into the universe, wherever the internet shall go. And so we're really excited to have you and to help teach us and teach our members that they can do the same thing, and we can grow awareness exponentially to many, many more people. We all meet hundreds of people every day, but through social media and the internet, we can meet thousands and hundreds of thousands and millions and billions. Maybe that's a little too much, but we're going to try. That's our goal. And it's all because of you, and we're going to thank you for that, and thank you for being with us. Guys, we're at that time of the day that we are finishing up all of our programs for today, and we're getting ready to move on to our evening here, so you're going to get a little break time and everything. Everybody give a big round of applause to Kim Sanchez.
Thank you.
All right, everybody, you're all done. Go ahead. You can take off. We're going to have a little dinner in a little while. We've got auctions and other kind of fun stuff to do tonight. Hang on to it. Yes, ma'am. With Jim, wherever he goes, follow Jim. Anybody want a picture with Jim? Follow Jim. I'm not. Where do you want to take your picture? Be up by the glass doors. Okay, so just go out with him and you can take a picture with him for the walk, roll, and run and stuff. Take a little time. No hurry, no rush. Okay, we'll get out there. We'll get that done. Thanks for being with us for the last day and a half. We still got a big evening planned. We'll see you soon.